And good morning everybody from the ROV Falkor, where we're currently located within the Great Barrier Reef Marine Park, some 130 kilometres northeast of the city of Cairns in Queensland, Australia. ROV SuBastian is in its final descent to a water depth of approximately 600 metres. The seabed is just coming into view. Water temperature 7.8 degrees at these depths. It's a beautiful day up on the sea surface. We've got sun in the sky. In the distance this morning we could see the breaking waves or the edge of the Great Barrier Reef. So we're positioned about a kilometre away from the edge of the outer reef of the GBR. Really exciting place to be right now on a Monday morning here in Australia. As we zoom in, as the ROV SuBastian settles towards the seabed, we're starting to get a view of clearly what is a pretty barren, from this distance, muddy to sandy seabed. My name is Scott Nichol. I'm from Geoscience Australia. I'm Chief Scientist on Leg 2 of the Reefs, Seamounts and Canyons expedition on the ROV Falkor, supported by the Schmidt Ocean Institute. This is a collaborative survey across a range of institutions who are, you can see listed on the YouTube background. It includes Geoscience Australia, the University of Sydney, JAMSTEC from Japan, Queensland University of Technology, and Queensland Museum. So we're here to discover the life forms, the geology, and the oceanography of this part of the Great Barrier Reef system. As you look at the zoomed in image coming right in close as the ROV pilots test the cameras for this morning's broadcast, doing a bit of colour checking there, processing some images, just getting the colour balance just right so we can get a close to real life true colour image of what we're looking at on the seafloor. There's the little crustacean lurking. Doesn't seem to be too bothered by the camera zooming in. Pretty cool. So just to paint a bit of a picture as to where we are in the GBR Marine Park, as I said at the outset, we're sitting a kilometre offshore from the outer reef system, what we call the Ribbon Reefs. It's Ribbon Reef number 5 of the 10 which make up the outer fringe of the GBR. And we're really interested primarily today in the upper reaches of a large submarine canyon system which has cut its way across the edge of the continental slope of eastern Australia over geological time, incising a canyon which in places is up to 800-900 metres deep. But today we're very much towards the headwall, the upper reaches of that canyon. Just watch that little critter crawl away from us at the moment. We're going to undertake an ROV dive which traverses about a kilometre across the seabed, starting where we are now and then move upslope to shallower water. We're starting at 600 metres water depth. We'll get shallower as we go. We'll track the depth, and then at some point we will hit a sheer vertical wall, we fully expect, of the edge of the Great Barrier Reef itself. That wall's, we're really excited to see this because this is a locality which is rarely seen with this sort of high resolution camera systems such as we have on ROV SuBastian and the sampling gear that it has in order to collect samples. ROV SuBastian is now just lifting off and we can start to drift on our transect. We're going to head more or less in a direct westerly direction from where we landed. Running along, and people are starting to see things on the seafloor. We have a team of scientists in the control room this morning, including biologists, benthic ecologists, geologists, sedimentologists, the whole spectrum of knowledge here in the room right now. As we just zoom in, that appears to be a very much a large burrow from an informal organism creating that. And there's something in it. Real beauty. It's an isopod. I'm hearing in the background. That is perfect. So it's getting a bit of clouded water there kicking in.
Y'all want to look at that thing or keep going? I think they want to look at it.
Yeah, it'd be good to have a look at that isopod and get a sense of if hopefully it hasn't scurried away. Clearly, as you can tell from the cloud kicked up here, these sediments are very fine-grained. For the most part, we're dealing here with a lot of sediment which would have been shed or fallen off the edge of the Great Barrier Reef from shallower water, transported off the edge of the shelf by tides on a daily basis and during higher energy storm events. You know, this part of the Queensland coast gets cyclones quite frequently during the cyclone season, so it's not unusual to have very high energy events setting up large waves, of course, which can transport sediment off the shelf down the steep slopes to where we are today. This is dive number 384 in the sequence of dive numbers that the ROV team operate. But for our survey, this is dive number 10 of the entire survey. And for us that are running leg 2, this is our third day of operations, having left Cairns last Friday. For those dialing in or tuning in for the first time, this is our third day in this canyon system. We've been tracking up the axis of the canyon, where we started a couple of days ago in 1,800 metres of water depth, looking at very muddy, fine-grained deposits, sparse biota in that locality. Yesterday we saw some really fascinating outcrop of rock midway along the canyon, which we managed to collect a sample of, which was a really exciting moment for the geological team of part of this party, giving us some really important insights into the long geological history of this canyon system. And today we're changing gears a bit, coming up into those shallower depths, where later on in this dive, which will run for a number of hours, we fully expect to see a lot of seabed life as we get to these shallow, what we call mesophotic depths, which are within the reach of sunlight. Just. So we're just being patient here while the ROV pilots try and position ourselves. There's an interesting fish going by. He's hanging around.
Got to get to this point right here, so I would say just kind of like that. Yeah, it was right here.
Yeah, more than anyone else. With shallow, so probably got steps.
Thrusters kicking up a bit more sediment as the guys turn the ROV around. The ROV is about the size of a small car. So there's a set of thruster systems which you can use to move in all directions really delicately and approach features that we're interested in. Okay, here we go. Here's the seabed coming into closer focus. Whether we manage to find that burrow with the isopod in it will be. We've moved on. Thanks, guys. Yeah, kick on. Okay, let's just travel along our transect, and at the 100 metre mark, I think we'll start our 500 metre transect if we're Yeah, just move up to that waypoint. That'd be great, J-Rod. Thank you. We've got J-Rod and Adam as our pilots this morning on ROV SuBastian. We've worked really hard in the last 24 hours to deal with a few little tech issues we've had, but fantastic to be operating again.
Yeah,
We really haven't lost that much time. Okay, there's those isopod burrows. Not the same one, I don't think.
So is there not much current down here? Just coming out of this that goes.
I think you're dragging a little bit on that floor.
So we have to be careful with the stop or it goes down. There'll be very much current. Yeah.
Okay, I'm hearing from the background there's not a lot of current at the moment in this, whether that's the stage of the tide we're at or just the nature of this locality at the foot of the canyon wall. Okay, this is looking clearer now for everybody. Get more interesting. Certainly a lot of small fish dashing about. And the lights. Remember, we're down at 600 metre water depth. Typically this is pitch black.
All right, shall I jump in there and we'll do this chat?
Okay. I'm going to hand over to Dr. Rob Beerman, who will introduce a guest from on shore today. So over to you, Rob.
Hey Jody, how are you?
Hey Rob, how are you? I'm great.
Yeah, can hear you loud and clear from the control room on the Falkor. I can see you just above my panel. I've got the science camera right in front of me, the science camera vision, and you've got the same. Yeah, it's good to hear. Good to hear. You in Sydney, and I'm out on the ship. But oh boy, hasn't it been it's been a long trip to get to this point. Shame you couldn't be on board, but you can virtually anyway.
Yeah, indeed. I'm as we sit here, I'm actually in the bowels of the Geosciences building at Sydney University in our Geo Reef lab. So it's quite amazing to be talking to you and also looking at images and footage of the seabed from the SuBastian. It's quite spectacular. It's an amazing. It's quite a disconnect and disembodying experience to be in a number of places at once.
Well, are you talking about your workshop where you've got that 210 metre core from Ribbon Reef 5? Now, if that is the case, that's only about 3 kilometres away from where that core was taken.
Yeah, indeed, Rob. So actually, sitting on the bench right in front of me is some of the Ribbon Reef 5 core, a couple of core trays, which we are working on actively in our lab. And the Ribbon Reef 5 core, as you say, the actual modern reef is just very close to us right now, and we're sort of heading towards that. But yeah, this core that we've been working on is really quite spectacular, and it was collected some time ago in the mid-90s, but it's quite unique in the sense that it penetrates about 210 meters, and what's really special about this core is the high quality and the high recovery. So it's really a special core.
Yeah, no, I've been rereading through all the literature, and right, it's one of the longest drill cores through any of the coral reefs on the Great Barrier Reef, and yeah, literally we're just sitting about 3 kilometres from where that was taken. But we're now in the head of this canyon, shelf-incised canyon that butts up really hard against the edge of the Ribbon Reefs. The Ribbon Reefs being this barrier reef system right on the outer shelf of the Great Barrier Reef. And you know what's really quite amazing about this trip is we're able to look from the seaward side at that wall, which hopefully will kind of match up that drill core that you've got sitting in your workshop at University of Sydney. So we're actually on what we call the middle part of this canyon. So we're down at about 600 meters, about 1 kilometre long, and at the very end of this ROV transect, it's going to go very, very steep. We hope that will be vertical wall that will show the layering that you've seen in your cores. We'll see it almost like an X-ray view because it's a sheer cliff, like, you know, a quarry face. So look, this is really exciting because it's kind of joining that sort of two-dimensional core view against the three-dimensional landscape, which to be able to see all those different layers.
Yeah, indeed. It's been a dream of mine to be able to really see the front of Ribbon 5. So to really try and get a better sense of exactly that, connecting what we can see in the drill core, what we might be seeing at the front of the reef, it'll be really, really quite spectacular. And, you know, in the 80s, they did a submersible dive, much, much shallower depths, in the vicinity, but of course they didn't have anywhere near the imaging capacity. Of course, they had no way to actually sample those outcrops. So I'm really, keeping my fingers crossed, and I'm hopeful that we'll get some really stunning footage of that four-reef slope or that cliff, and then that we'll be able to sample that material and then hopefully compare what we're seeing at the front to what we have in the drill core.
Yeah, so we're actually taking a core sample, a sediment sample at the moment. This is part of our systematic comparison for all of these canyons along the front of the Ribbon Reef. So we're, for every one of these ROV transects, we do a standard 500 metre run where we don't stop, but at the start of it we take a push core, and so at the moment the manipulator arm is pushing literally just an empty tube into the sediment and lever it out, and it's been very successful. We've got a few other toys up our sleeve here. The ROV team have created this this drill bit that allows us to actually bite into hard rock. Now, yesterday we had our first go at this, and this was really quite amazing because we're at the middle part of this canyon, right, this Ribbon Reef 5 canyon, around sort of 1,600 to about 1,200 metres depth, so it's about 1.5 kilometres down. And it was really steep. The mapping that we had was showed that it was quite steep, and we deliberately put the ROV down right in the axis of this canyon. It was literally V-shaped. Sides of the channels were just sheer rock face, and I don't know if you saw this yesterday on the YouTube feed, Jody, but that was something else. We were actually, it was hard rock, and more than just look at it, we're actually able to get a sample. So we actually had a drill core bit, and they cut a little plug of rock out, and this is actually the basement of of the Great Barrier Reef. You know, the reef has grown on top of a bit of continental rock that's jutting out underneath, and we actually have a piece of it now. So that was something else. Very exciting day yesterday.
Yeah, I was watching that actually. It was obviously Sunday morning, and my two daughters, Lily and Scarlett, were actually watching as well, as the ROV team was attempting to sort of drill in, and successfully able to do that. And that was quite amazing to see that chunk and the actual drilling in action. And I noticed actually that they seemed to bend the drill bit. So I'm assuming that the magicians on board are now to sort of fix that up. So hopefully we're ready to go to try that drilling system further up the slope.
Yeah, for sure. I mean, they're quite remarkable. The ROV team. There's a big workshop out the back here. It's just filled with ways of fixing problems and the like. You know, the SuBastian is. It's one of the world's best science research ROVs. It's about the size of a large car, and it's about 4 tons in weight. They've got all these ways of solving problems. So the drill bit did bend a little bit, so it's been shortened overnight. So there might be a bit more torque power. Yeah, so I mean, the ideal world we'll get to the end of this transect, and as it starts this steep rise up the wall, where we hope to see the different layers of ancient Great Barrier Reef. If at all possible, we'll be able to put this drill bit in and actually cut out a little plug, and maybe you can tell us what you might do with that plug, because there's so many things, so many different analyses you can do.
Yeah, so what we're hoping to get is some fresh limestone material. There's still a lot of debate as to how old the Great Barrier Reef is. We're not 100% sure. Maybe 600, maybe 400,000 years. It's still a controversial point, in part because this material is very, very difficult to date. By that I mean using geochemical methods, geochronological methods, to get unaltered fresh material. And we're actively working on that with the Ribbon Reef 5 core that we have in the lab. Hopefully we can get some additional samples today, and we can do a number of things with those samples. Firstly, we can have a look at that material and see what it's composed of. What sorts of corals, what sorts of algae, what sorts of larger benthic forams, other sorts of reef biota, and then we can, by comparison with modern, we can then reconstruct what sort of environment the reef may have been growing in at that time, and then hopefully if we can find some unaltered skeletal material from the corals, we can use some techniques, dating techniques, to hopefully get a better age and actually understand the timing of the inception of the Great Barrier Reef, and better understand the conditions that led up to that. So that's really a grand challenge that we're still trying to work on.
Yeah, and I guess yesterday's dive in the middle part of the canyon really just talks to the long development and then erosional history along the continental slope in front of the Great Barrier Reef, because the Great Barrier Reef is just a kind of relatively thin layer of limestone, multiple successions of limestone growth as sea levels cycle up and down, but it's grown on this great long build-up of the continental shelf, the edge of Australia, since the breakup of Gondwana back in period. So where we have the shelf, as it builds up, eventually becomes this hard rock basement. So we've been mapping this area, you and I, for well over 13 years, trying to build up that three-dimensional picture shape of the seafloor, including all these really complex canyons. But to actually put a camera down there and see that rock first time, that basement rock of Australia, the Great Barrier Reef is growing upon. That was just amazing. And also the rest of the shape. In fact, I don't know if you can see this inset. If you can see this inset, the mapping, right, so this is a three-dimensional picture of the shape of the seafloor, the mapping that we've done. So, of course, it's artificially coloured. We've just coloured it rainbow colours. The shallower portions are red, going through yellow, orange, green, into blue and violet in the deeper area. But 2 or 3 nights, and so this has given us the three-dimensional basis from which we can then choose the ROV dives. But you can actually see the bit that we're on. This is this red line at the very top of the canyon. This is the stretch that we're on now. So 2 days ago, we're at the very, very base of this canyon. It was nearly 2,000 meters deep, very, very muddy. But certainly lots of marine life there. We had lots of holothurians and sea cucumbers and the like, but I guess beyond the origins of the reef, this voyage is also trying to understand what's living down in these canyons. You know, as I say, you and I have been mapping this area for, just think of our first voyage back in 2007. It's 13 years ago. First opportunity to put really good cameras down in these depths.
Yeah, indeed. So it's one thing, to get these multi-beam maps, which have been, just transformative in terms of our understanding of the nature of the seabed and, revealing the structure of the shelf edge and the canyon system and all the different types of canyons and slope features that we've seen. But yeah, it's another thing entirely to be actually see the seabed and to be able to sample areas that we weren't able to sample before. So yes, fingers crossed, we can get some good imagery and some good samples.
Yeah, so just talking about the marine life that we're seeing now, I mean, it's worth talking about that because this is right in front of us, and the SuBastian is now drifting over the seafloor. I think we're on our 500-meter transect. This is a standard transect that we do on all of the ROV dives, and allows us to do this comparison between this particular canyon, which it's a shelf-incised canyon. The head of it comes up hard against the barrier reef system that we have here, what we call the Ribbon Reefs, and another canyon we're going to sample later in the afternoon, which is shelf-connected canyon, where it's actually open to the Great Barrier Reef shelf itself, where we expect lots of water movement and organic matter to come down. And I'm just looking at the seafloor here. It's clearly sandy. I often talk about these reefs as carbonate factories. They're just pumping out calcium carbonate, which is the limestone that makes up coral and the algae that grow on these shallow coral reefs. And then some of it ends up on the shelf itself in lagoons, and some of it ends up on the seafloor here, in the deeper seafloor in front of the shallow coral reefs. And it's certainly muddy, muddy sand, and there's actually a fair bit of life down here. I don't know if you can see these. There's lots of burrows. We've already seen one of these giant isopods. It's like a big pill bug. It's about as long as my forearm. It had these are these large burrows. Now they're about 5-10 centimeters across. So we've already seen one of these giant isopods sitting in amongst it. Another thing we're seeing is eels.
There's a lot of life there.
Yeah, and so there's quite a few of these small demersal fish. They're triangular in shape. They're very silvery when the light's on them. They've been dashing all over the place. Lots of crustaceans as well. Like you can see. Here's an isopod. It's coming into view now. This is a giant isopod, probably about 10 centimeters across. Benthanomus is its genus name. They're found all over the deep oceans, and there it is. It's just skimming into view. So this is a crustacean. And it's got a hard exoskeleton. It's just dropped on the seafloor now. It's got a kind of a pinky purple colour, and these are the guys that are making these fairly large burrows that we're seeing scattered all over the place. Pretty cool, because I've actually seen a fair few burrows, but not necessarily with any occupants, and so it's great to see them. They're just excellent diggers. There we go, like little jet planes, scooting over the seafloor.
You can really imagine them turning over a lot of sediment that way. Really reworking it.
It just shot off to the left there. And actually down on the bottom here, I'm seeing, this looks a bit like a brachiopod or bivalve. We're just going past, in fact, it looks like we might have a benthanomus. Jeremy, we've got a couple of things here. We're just going to zoom down on the, what is either a bivalve or a brachiopod. So it's dead, it's upright. No, I think this is more a bivalve. Okay, so we are in fact close to less than a kilometre away from shallow coral reef itself. So it's no surprise we're seeing, all kinds of, I guess, shallow water material, perhaps bivalves. Just, they fall off the shallow coral reef and fall down this slope. It's a gentle upward slope. And Jeremy's next to me. He's our black coral expert, and we've got something in that burrow. Just going to have a bit of a zoom in here, Jody.
Right.
So you might be able to see these two. Yep. It looks like we have our another isopod. A bit hard to see. It looks like it's the front. It could be a mantis shrimp, even.
Oh yeah.
Could be a mantis shrimp, and it's making use of the isopod hole. Yes, and it's a squat lobster. Yeah, squat lobster. We're getting another view from a different one of the cameras. There's about 10 cameras on top of this SuBastian ROV, so you're just getting the science cam view. We actually have other cameras looking down from above, and we're actually looking in on top of it, and it's a squat lobster. It's making use of the burrow that's there. So just really spectacular. So we're getting the deep time geological story through looking at the canyons, both the deeper parts of the canyon, the middle zone, and now the upper part of this canyon, and also all the marine life that's over it, and that's just been spectacular. But what we might do is leave it at that, Jody, and we might join you later in the dive in about an hour or so, where we've moved through our 500-metre transect, and then we'll be a little bit higher up that transect, closer to the coral reef itself. At that point, we'll see a real change in gradient, we hope, and I expect we'll see coral rubble and sorts of things that have been described in the literature, but we just have never had a view of it before.
Sounds good, mate. No problem. I'll see you soon.
All right. Well, I'll sign out and hand back to the other members of the Falkor party, and we'll keep you in touch through the chats and everything, so you know where that's coming up, and then we'll join you then, and we'll just see what we find at the end, eh?
Excellent. All right, mate. Thank you.
All right. See you later. See you later, Jody.
Something coming up.
Okay, Scott Nichol back online here with the real-time narration as we continue our dive on dive 384. Sitting in water depths currently of 570 metres below the sea surface. running along one of our standard 500 metre sampling transects, across which we have a team in the background working on specialised software which allows us to systematically record the biota and any other features we see on the seabed as we go along. So we have a team sitting behind me in the control room who have got software called Squiddle, which they hit buttons as we observe features such as those what look to be the holothurians, I think, perhaps, sitting on the seabed, sea cucumbers. But for the most part, they'll be describing this as a barren, sandy to muddy, flat seabed with a lot of bioturbation. Those small mounds we can see as we cruise along at a consistent altitude above the seabed of a couple of metres. Each of those little mounds is the burrow. You can also make out a lot of trails or feeding traces along those little wobbly lines across the seabed. They're, for the most part, narrow troughs or furrows in the seabed created by those isopods that we saw earlier crawling along. I'm also starting to see a hint of some small ripples on the seabed formed of those lighter coloured materials, which would be most likely slightly coarse sand. So that's a bit of evidence for a degree of current activity, very weak, gentle currents on the seabed, but still water moving about nonetheless to help separate the sediment out into those ripples. But today, the current strength we're experiencing with SuBastian is very, very weak, very low energy. You can get a bit of a sense of that too if you look into the water column and look at the particles floating down. They're all sort of just kind of drifting gently. In some of our other deeper dives, those particles have been rushing past us at a couple of knots. So the currents here are pretty low energy, pretty weak. Which kind of makes sense because what we're looking at is for the most part, a muddy to slightly sandy seabed. And you do need those low energy conditions to allow that mud to settle out through the water column from 500 metres above to settle on the seabed. So it's a consistent story starting to emerge in terms of the energy conditions at this site at this present time.
Okay, so Jeremy's
We've got cucumbers.
They almost look bed form like. But
yeah,
Jeremy's money's on animals, but how do you end up with almost like burrowing? Not burrowing. Ploughing is probably the word. But how do you create animals like this? It's too random to be.
Yeah, we're having a bit of a debate in the control room here as to whether with these myriad lines running across the seabed, whether it's the origin of those is from bioturbation, from organisms crawling along, which in some cases they clearly are, and or we're looking at a bit of ripple formation. We're sort of potentially in that sort of transition between the two. So it's, you can have cases where a burrowing organism creates a micro topography, maybe just a centimetre or two high, which then a current can slightly rearrange to form and sort the sediment and remove the fine grains perhaps and leaving behind the sand. So these are some of the clues we need to take on board and decipher as we continue to try to understand the dynamics of the seabed in this place. Looks like a fish sitting motionless on the seabed there, twitching a bit as we go over him, trying to... So Currently the jury's out as to whether we've got a clear ripple field or a biturbation field or a bit of both. Good morning to everyone who's on the chat this morning through YouTube and Facebook. Great to have everyone along, joining us on the ROV Falkor from the Schmidt Ocean Institute, part of dive 384 here in the outer reaches, outer limits of the Great Barrier Reef Marine Park, offshore northeast Queensland in Australia. Okay, something coming into view. Looking at an urchin, we're hearing in the background. Very nice. So just a reminder, we're on a standard 500 metre transect where we're scoring or describing, recording through our Squiddle software what's there. We're not stopping to zoom in or collect samples at this point. We'll do that later on the cruise, on the survey transect this morning. Brittle star, quite large. These are very common.
Yeah, look at this. Just look at that. And that there, you know, so how about if you start with
major mound structures and burrows there. You start to get a sense of the three-dimensionality of the seabed too, as we, in this view, at least as I'm seeing it on screen. Those lighter lines, I'm putting the money on being coarser grain sands, undoubtedly carbonate, shell fragments, perhaps halometer flakes from the reef, which have settled down. Current water depth 540 metres. Correction, 533. We have a couple of readings here of depth. We'll stick to the one that you're all seeing.
You feel it maybe at home meters, but not at this depth. This is ocean. It's the only way that. But we do know that there's just a little bit. You think it's really sharp. You know, canyons be directing.
Is waypoint 3 the end of our 500 metres?
What's that?
Is waypoint 3 the end of our 500?
No,
not yet. Okay. Got a ways to go. Okay. Yeah. So we're not quite halfway through our 500 metre transect. Looks like 325 to go. Thank you, J-Rod.
Oh, there's a seabed with the lights making.
Sea pen coming into view. Yep, sea pen, thanks.
You want it, Jeremy?
No, we'll keep cruising. We need to be strict with ourselves on these parts of this transect. There'll be other opportunities to collect.
There's no contour rights around for a start.
Just to the inset coming in on screen at the moment is our 3D view of the seabed that we've mapped using multi-beam sonar. You can get a view at the moment of our track line, which is that purplish line running up the slope of the upper reaches of this canyon system as we head towards the edge of the Great Barrier Reef. Our transect line this morning is a total distance of 1 kilometre across the seabed. So we'll be on this for several hours. Currently on our 500 metre standardised transect where we don't sample, we simply observe. There's another one of these deep isopod burrows we'd expect. Yep, something moved into it as we flew across. Yeah.
Actually, we have a little ROV in the channels. Yeah.
What have we got here? Some form of it's a tube anemone, I'm hearing from our biology team in the background. I'm sure a lot of people online will recognise that as well.
Black corals were so closely related, we created an order just for them called the surrounding area. And after realizing how distinct they are, then they broke it up into 2 different orders.
There's a fish we're on the transect. We'll just keep it this height. But the nice little fish. Googly eyes there. Really is a lot of bioturbation on this seabed. Perfect habitat for these isopods and other. I'm sure there's a whole range of really highly diverse infauna within the sediment, crawling through the upper 10 to 20 centimetres of the sediments. Those who've been following us online for the past few days may have seen us taking short push calls in these soft sediments. What we're finding as we've been bringing those back on board and opening the calls up to have a look at them, there's a clear what we call a redox horizon within the upper 5 to 10 centimetres, where we go from a oxygenated surface layer of sediment, which is, we identify that simply by its colour. So in these environments, it's typically a light brown to faintly reddish in colour, as indicative of the presence of oxygen within the sediment. And then we pass into, below that, a grey anoxic oxygen pore layer, which would continue for many metres below the seabed. So we've got an active layer which has been turned over with oxygen introduced into the sediment, and there's no question that the bioturbation behaviour of these infauna contributes to that oxygenation of the sediment. That living layer, you might call it, living with people, living with organisms and living with oxygen.
Do you want us to stay on that ridge? It's a little bit deviated from the path a little bit, but it's kind of a good picture. You're getting that whole kind of...
Yeah, sure. Yeah. Thanks.
Now we got a big rock, by the way, that you're supposed to have.
Something coming into view here, which it looks kind of lumpy. Okay, here we go. We have what I'll first call a large block, and we're looking. This is rock. This is unquestionably a large block of hard material. This is not mud. It's mantled by mud. Just a reminder, we're in a submarine canyon, so this is not unexpected. Just gently fly past it. A seabed slope is quite steep here too, so my initial thinking is we're looking at a loose block, a rock which is
This is where animals come to hide.
whether it's a direct outcrop from the underlying rock or it's a...
This is Echia and worms.
Okay.
So you've got this classic T-shaped proboscis. The actual worm is inside.
Yeah, okay, it seems to be, it's clearly buried into the distance, so we just saw a face of it there. So they're a little hard to separate between whether that's a transporter block from higher up or whether it's a direct outcrop in place. We'll keep moving. Yep, thanks guys.
So these are a couple of metres long, easily. That might be 2 metres long.
And just
pulling it in.
clearly I was focused on the rock there, but there's obviously a lot of biota, epibenthos using that hard surface as habitat. Stars, urchins. So back now to that relatively featureless muddy to sandy seabed with all those squiggly lines running across it, which were clearly some biological fingerprinting of the seabed with burrowing organisms. A bit more detritus around. So a question online, are we set up to do rock cores again? And the question is, yes we are. Those online yesterday would have watched us collect our first rock core with the small drilling implement the crew on board manufactured for us on board during the last couple of days. We managed to bend it a bit yesterday in collecting a rock sample, which wasn't that surprising. But overnight the crew have adapted it, shortened the stem. That'll give us more torque as we plan later on this dive to ideally sample some of the exposed limestone, which will form we expect will form the vertical face, which will climb up later on this dive. Yeah. So yes, asking whether we could use a bag to catch it. That's an idea, because yesterday we dropped our rock sample, managed to find it after a bit of a search.
First living corals, deepest, series.
So could you use two arms to unhold the bag and one? Yeah. Okay. So yes, just to fully answer you.
We'll try to set up somewhere where we can have something underneath.
Yeah. Okay. So the J-Rod, our ROV chief pilot, is saying yes. We'll try and be a bit smarter today and have two arms involved with the rock drilling. Because if we're 200 metres up a cliff face and drop something, we don't have any chance of finding it. So we'll have they've got ideas about what we'll do there. Have something underneath held by the second arm. Maybe it's a little bit of board or something. We'll see. So not a lot of change as we continue our 500 metre transect, up the gentle slope, relatively gentle slope, 490 odd metres water depth. There's our inset again of our canyon viewed from a bit more of a distance, and in the background there in that inset diagram is the Ribbon Reef number 5, and you can just get a sense there as you start to zoom in how this canyon has just started to nibble in the edge of the continental shelf of Eastern Australia, what we call a shelf-incised canyon.
It'd be good to have a zoom in on that if you get the chance. It looks like just a bigger urchin.
You don't want to go back to the urchin?
No thanks, we'll keep going. Thanks guys.
Well, we've seen two big ones, and then 10 centimetre.
Yeah, and I mean, it takes it down like 3 minutes.
You could probably say the number of our biotubation mounds has increased in the last little while. Yeah, it's interesting that the density of these mounds is something that you got to start to wonder about. You know, what's controlling the? Is there a limiting population's kind of density for the presence of these in fauna in a particular environment, or does it not matter? Is there enough nutrients and accommodation space, if you like, for a higher population density? This is some of the denser burrow mounds we've seen on this part of the dive. Seabed dropping off. So right now we're tracking along a section of the canyon along a gentle ridge, so it's now dipping down to the right or to the north as we look at it on screen. So that's quite a nice little cross-section view right now of the edge of that little ridge down onto a lower beach, but really no change in habitat with that minor change in topography and aspect as we drop down. Still favouring high density biotubation in fauna. Bit of a cloud of sediment there from a fish trying to ignore us.
$150 meters to go.
150 metres to go to the end of our transit. Thanks, J-Rod. 15 metres to go, Alex. 150, I should say. Takes a lot of attention from the team in the background, focusing in on these images as they see them. Putting dots on the screen basically is what they're doing, which records the position and time of observation of any biota. A lot of concentration, and gets harder and harder with a greater density and presence of stuff on the seabed. One thing we're aiming to do with the video, which we collect using the ultra high definition 4K video record we have of flying over the seabed like this, is to build up what we call a mosaic image, which we can create an image in 3 dimensions of the seafloor. So really helpful for analysis back in our labs later on as a reminder of what we've sampled of the general environment from which we've taken samples. There's something burrowing in. I didn't quite get that. Urchins. A question from Facebook, I've just picked up on, about what happened to dive 379 to 384. Really good question. That's occupied us for the past 12 to 15 hours as a series of aborted dives. We had some issues. Sorry, we're just going to. I'll come back to that in a moment. Yeah, Jeremy's commenting on some biota.
All right, that's great. Let's just continue. That's fine.
Okay, we'll continue. Yeah, as I was saying, we had a series of aborted dives from late yesterday afternoon where we had some tech issues with one of our key science cameras, and the ROV team had to troubleshoot that one, and they've managed overnight to come up with a bit of a fix through some recabling, I think, was the solution. So fundamentally, we were losing yesterday afternoon. We were losing our video feed. So these things happen when you're at sea, but we've got everything on board to fix the problems. Got the people with the skills, we've got the gear. You never go to sea without that sort of ability to fix stuff. So yes, an aborted dive is recorded as a number in the sequence. So we had, just go back to my notes. Yeah, so 379, 38, 381, 382, 383 were all aborted dives, and they included a couple of quick test dives. So any time the ROV goes in the water, it's recorded as an event, recorded as a dive, even if we just put it off the side of the vessel. Because even then, from an engineering point of view, we're collecting data around, in this case, around the performance and the condition of the ROV SuBastian.
What's that? I think this is. Are we done?
We're close. We are
80 meters or something? I mean, we're so close. This is a branching black coral. It's super deep.
60 meters away.
That's really it's interesting.
So just hearing, we'll slow down a little bit, as you can tell.
I guess we're just going to transect.
Yeah, I'm hearing branching black coral from Jeremy, our coral expert on board. But we'll continue.
Right here and come back to it.
Continue.
We're so close. We'll just finish. And then
Yep.
focus on.
We don't really have the time to ask
Yeah.
on this one.
No.
Yeah.
So everything
We're going to continue.
Yeah. That's okay. But it was very long. It was very long branch, but not complexly branched, which is odd. But there's a few. If the black part there, I couldn't see. It just looks odd. Odd is always good.
It's a tube anemone, I think. As we get closer to it, yeah. Attached to the seafloor. Here's another one of your corals, I think, Jeremy, coming up.
You're like teasing that.
Yeah, we're teasing Jeremy.
Yeah, it's so interesting.
Yeah. How close to the end of our line are we?
We done yet?
Almost there.
80 metres.
Just keep going.
We'll keep going. Since we've seen two, we'll see another one. We'll continue, guys.
We can see a lot.
Yeah.
It looks like
We're pretty confident that will be the last one of those we will see for Jeremy to sample. Famous last words, probably.
yeah, sure. That's how it works. That's how it works. It's different. It's much more densely branched. Is that a beard? It could be an octocoral. They tend to have this one stem and then lots of branches coming up.
Human impact. That was my business.
Maybe it's a full beard.
Oh, this is sad. This is sad.
You want to clean it up?
Yeah, let's grab this. We don't want to see rubbish.
Great move, but it's a great song.
It's a great northern beer. That's a relatively new. We can date this relative terms. This is a relatively new beer type in Australia called Great Northern, and so that tells me that's probably been dropped in the last couple of years. They're pretty fresh. So we've cleaned up the ocean, which is a excellent thing to try and do. Thanks, guys.
Australia Day, I think this week. I think so. Yeah, I think so.
We're doing our work.
It was our Clean Up Australia Day effort. But we hope we don't do more and see more of that.
Bit of chatter in the background. Thanks, Rob. In Australia, we have a thing called Clean Up Australia Day, where everyone in the community gets out with empty bags and cleans up the streets and parklands and everything. So that's our little contribution with Clean Up Australia Day. We think sometime this week or this month.
we're going to look at these branching so good.
Stars in around the rocks. There's quite a few of them actually. 1, 2, 3,
Wow, look at that.
5. There's at least 5.
There's a microhabitat there of sea stars sitting on that rock. Okay, distance to end of transect?
Less than 20 meters.
20 metres, counting down.
Oh, sorry, we're going to add those. Yeah, I messed up on that before as well. 60 meters.
60 metres, correction, counting down.
I'm sorry.
You notice more of these class, these rock.
Yeah,
pieces starting to-
yeah, I'm seeing
And we should see more.
Well, more of this,
closer
yeah,
to
that-
the shallow.
yeah, that's right, Rob.
Reef, but
Yeah, gee.
And you picked up before, you know, that animals are using them as little habitats. So, but we're just starting to see more of these. You know, could be just coral on a rock that's been knocked off,
Yeah.
and it's
Yeah,
down slope of the shallow reef.
that's right. Yes, we're just chatting here. You may have heard that. We're seeing more of these very small clasts, as we call them. Or small rocks, perhaps.
Oh, is that a nautilus shell?
Yeah, it looks like it might be. Yeah.
Or is it living? It could it be a sleeping nautilus?
We'll soon
We've
see.
had them. We've found them
wake up.
sleeping on the seafloor before. We are
That is a
within their
nautilus.
zone.
Yeah.
I think that's a living. That could be a living nautilus just having a sleep.
It's
Yeah,
alive,
it's alive. Yeah, do you see the little siphon squirt?
Cool.
It's not very big.
We'll leave him alone.
Can we zoom in on him?
Yeah. We can. Whoops.
Hooray! Nautilus is a sleeping nautilus. So this is the zone they live in during the daytime. They're down this between, let's say, about 400 to 800 meters, and at night time they vertically migrate into the shallower waters. This is quite a small one actually. Maybe less than about 5 centimeters across. It's quite young.
See his eyes there.
Yeah, and we have seen them before. They're sleeping. They basically close up their operculum, which is this sort of leathery top that closes in. And when this is open and when they're actively swimming, you can see the eyes. In fact, you can just make out the pinhole eyes on the left. You can just see that little dark dot. That's its pinhole camera eye. But just underneath that flap, which is a kind of leathery, and it's got these sort of soft, like little spikes on top, there are about 90 individual arms. So this is a really remarkable animal. It's related to ammonites, which died out mostly at the end of the Permian extinction over 400 million years ago. But at one time, these were like the top predator in the world's oceans. And now what we have is this sort of, well, you could probably call them relic. They're long distantly related to ammonites, but they're a type of cephalopod, so they're related to octopus and squid and cuttlefish. And here we have them living in the Coral Sea. And for this voyage, this is the first living nautilus that we found on the edge of the Great Barrier Reef during this expedition.
Yeah. Okay.
And
That's
it
pretty
is
cool.
very likely sleeping, right? So
Well, not anymore.
there we go, and he's starting to wake up, and he's realises something big out there. But yesterday the guys were saying they stopped for Dumbo octopus, but we also stopped for nautilus, and we love looking at them.
Yeah,
There we
look,
go,
it's taking off.
starting to move. All right, so they have a little siphon. And they have this really odd swimming behaviour. Now they can be positively buoyant, and they use the chambers inside the spiral shell to change the density of the water. This is a really young nautilus, like it's only about 5 centimeters across, and you can see that beautiful fresh coloring on the outside. But they have a siphon which they can squirt, and they do this kind of weird rocking motion. But the chambers, the internal chambers, are really interesting in that they actually use osmosis to change the density of the internal chambers and make themselves more light, so they become positively buoyant. They rise up into the water column, and at night time, right? So they feed in the shallows at night, and then they vertically migrate down into these depths during the daytime. So at this depth, which is 450 meters, if the lights were off on the ROV, it would be pitch black. But we believe they do most of their feeding at night time, and that's the reason the vertical migration. But we have seen lots of them in the previous ROV dives.
That was great.
Very cool.
That was great.
Thanks, gents.
So we'll, I think that's the end of our 500 meter transect. Almost. Another. We'll just resume that. We'll finish it off, and then we'll go into sampling mode.
Yeah, we might see a lot.
And I'm sure we'll see a lot more of that sort of stuff.
All this stuff.
Yeah. Oops. So we're still live. Got distracted there. And as we look at the, just coming to the end of our transect, and there's more of the corals. Jeremy is itching to collect one of these.
Or just see what it is.
These corals, or at least have a closer look at it. Yeah. So we're continuing to come up towards the end of our 500 metre transect.
So just talking a little bit more about the Nautilus. A colleague of ours, Andy Dunstan, did his PhD studying these Nautilus, mainly out in Osprey Reef, which is about 100 kilometres due east of where we are now, and we're going to visit that during this expedition. But one of the projects he did was with Greg Barrold, who's from the United States, and they came out here with a small vessel, and they had funding to put down Bribus baited remote underwater video systems, and they laid them all along the front of these Ribbon Reefs where we are now. And it wasn't surprising, they actually found lots of Nautilus attracted to the bait. And then the bait they liked were chicken bones. So these are clearly scavengers, and they seem to like the smell of chicken. So the cameras actually recorded quite a few Nautilus right here where we are now. But just, I guess the differentiation here is we have the beauty of the SuBastian, and we can actually follow these animals around. And we may see more of them in this dive, but what we have now is an opportunity to do some sampling, and we've got Jeremy, he's very interested in black corals, and so we're going to try and do some sampling now that we've finished our transect.
Okay, so we've just come to the end of our 500 metre transect, and just by nice coincidence we've got a couple of corals which we'll settle down to sample and keep Jeremy happy. It's an important thing to do, is key player on this voyage. Key player. Yep, just carefully settle SuBastian onto the seabed. Undoubtedly kick up a bit of the mud, so for those looking online it might go a bit cloudy on screen for a bit. But then we'll just patiently wait.
Yeah, this looks like a Salopathes black coral on the right, with the little crustacean hanging on at the top of it. If you remember, on leg one we were seeing a lot of these, and we haven't seen one yet since we've gotten to these canyons. And if you remember, there's a a worm run that runs from the bottom to the top of the coral. And what the worm does is it creates this tunnel, and it modifies the skeletal spines that we use to identify the species. So every one we've ever collected has had these worm runs, and we still don't know what the spines actually look like. But given the opportunity of having an ROV in the water, we can look at the colonies and find one that's perfect condition, maybe young enough to not have been taken over by a worm, to describe the species and separate it from closely related species. So it's a bottle brush pattern. So it has repeated branches or pinnules that go around the stem. Look at this guy.
We just put it.
They just placed it there. We're going to collect it. It's the same one
We've
as
just did the
before.
crawl for the last 20 minutes.
They'll box it. Off you go, mate. There we go. This is defending itself. Back off.
You're in auto right now.
I'm in auto.
If that thing gets backwards,
I put it on. Now it says auto.
so try it now. If it doesn't work, let me know.
Nope.
All right. Did you click it again?
It's off auto now.
Yeah.
Yeah. Okay. So the pinnules have some subpinnulation. There are little branches coming off of these primary branches, which is one of the features that we use to describe the species and separate from other species. And you can see it's got tissue. It's feeding. Okay, we got all we need. We could put it, it's about 40 centimeters, so we can put it in BioBox 34. Okay. Yeah.
BioBox 34?
It's even smaller than that. You could probably get it in
Two.
one of them.
Yeah.
Well, even one.
Definitely get it in quad.
Yeah, if you want to put it in 1A or B or
Let's
something.
put it in 2, so the rocks are, yeah.
If you look at his jaw is this big, so it's only this high.
Okay. Okay then, put it in A.
It looks like it's attached to a coral
also think
clasp.
about-
Oh yeah, if you could grab the rock.
Yeah. Also think about where you want that can to go.
or the Nautilus shell.
Nautilus shell.
No big deal.
So isn't it interesting? It's used the rock as a base,
Right.
So we've
Yeah,
just
yeah.
found that little hard ground for almost
Wow.
decent sized rock.
So potentially BioBox.
That's good.
Should still fit the
Getting
quad.
some geology as well, which is nice.
Yeah,
You want it
that's a
in
good
quad?
picture with the Great Northern in the back.
Maybe.
You want it in the quad or in BioBox 2?
No, is it okay with you? In A, to put it in A, or rather not. Okay.
Yeah. This one is good.
Yeah, you put it in A, and it'll stick right up.
All right, off you go.
Yeah, he'll
escape.
He's got the biggest claw.
Might have been drinking that Great Northern. Pretty confident.
Quite a advertising system.
We don't want this.
It's beautiful. It's a great colony. Every time we get one of these on the surface, within a half hour, the tissue, it's so thin and small, it kind of just goes away, and I have a really tough time getting viable tissue into a vial to preserve the DNA to extract it later. So I have to be really quick with the specimen when I get it on the deck. Awesome. And so I'll just take this opportunity to explain why we're collecting this full colony and not just a little piece. So as I mentioned, the skeletal spines are what we use to identify new species and separate between closely related species. And we'll just look at that coral next to it now.
Sure. Put the can in with that.
So what we need to see is the skeletal spines along the corallum or along the colony. And there's often features at the base of these corals that are also indicative of
different species. So if we were to just collect the tip of a colony, it wouldn't tell us what it is. And we are keeping track of how many of each species we're collecting. All right, now we get the chance to look at this. So it's sparsely branched. It reminds me of a genus, if it's a black coral, Allopathes, which it looks something like this, where there's multiple branches coming from the base of the stem and very long branches, although these seem a little thin to be Allopathes. Can I just zoom in?
Yeah, sure.
All right, so we're gonna see what this is. So yeah, it looks like a black coral, and it looks really weird. It's odd because there's a stem, so that central stem, and then it has two branches, and these branches are really long, but there's no other branches, so it's very sparsely branched. So we would put it in the family Antipathidae, which is considered the taxonomic dumping ground of black coral species, because when you don't know where to put it, you put it in Antipathidae. Why don't we grab this one before the sand kicks it up? So I really don't even know what genus to put this one in. So it's going to be a really interesting specimen to preserve and try to identify later on. It might be in the family Antipathes. But this is a really, really interesting specimen. And we'll try to grab it at the base and see what it's attached to as well. See what the basal plate, the attachment point of the coral, is attached to. Probably a rock. Probably a small rock, or it could be a big rock. And if that's the case, it'll probably pluck off near the base. But we could be looking at a new species here.
Deep down in there.
You want to go straight forward with this, Jeremy?
Sure. Yeah, we probably broke it off near the base, probably not at the base. I don't see a basal plate. But maybe if we see one more along the way up, we can try to grab one more.
How deep do you think it was down in there?
That's
my jaws are in the mud line.
Yeah, because, I imagine the sediment piles up pretty quickly. If it's an old coral, and these black corals are slow growing, very old, it could have been on the surface when it grew and just grew upwards. And as you can see, it's not branching, it's using its energy to grow straight up. So it could be being constantly buried and continue to grow upwards.
Interesting.
Okay.
Yeah.
That's interesting.
Yeah, we don't know much about the sedimentation rates here, do we? But
No, we don't.
it's probably
Probably more the
slower
thing
end.
is the biotubation that's
Yeah,
happening, right?
yeah,
So
that's right.
It's the turnover just from the
Yeah.
biology rather than the sediment.
Yeah, yeah.
We do notice that from cores more down in the deeper parts of these canyons that the cores are only several meters long,
Yeah.
and yet you can be down in last ice age sediments within 30 or 40 centimeters
Yeah.
of the sea surface. So I would say that we're not talking about massive sedimentation rate, but clearly, if you're in maybe in the top 5-10 centimeters of that sediment, it's just turned over constantly because of all the animal life that's constantly burrowing and eating and turning it over and pooing and
Exactly.
It looks like a very high turnover area.
Amazing.
Okay, thank you.
Fantastic.
Thank you, team. That was great. Oh, we're end of transect, aren't we? Yeah.
Yeah, we're 10-15 meters out.
Okay.
You want to take them or the
Put on the cliff. Yeah. Let's try that, yeah. We'll push up, yeah. Let's push up to the base of the cliff. We want to
Super weird.
Just get
maximise our time on that cliff face.
Can I just jump in there and get some shots? So this is Robin Beeman from James Cook University in the audio seat here, and we're just going to get an inset of the 3D map of where we are. If you look in the bottom of the screen, we've got our Fledermaus 3D perspective. This is the mapping that's been done by the Falkor over the last few nights, and you can see the little yellow cube is actually the ROV. It's just a little symbol, little 3D model, and you can see where we started in the very axis of this canyon, and now we're in what's called the head of the canyon. So when we describe canyons, you've got a head. This is the origin, the very topmost, shallowest part of the canyon. And you can see how it's kind of like a bowl, and it's gradually getting steeper. We started on the southern steep sidewall of this canyon, so it's like a big valley, big broad valley. We went into the channel axis, and we're basically going up the gentle gradient towards the actual shallow coral reef. Now this is the shallow coral reef is Ribbon Reef number 5. It's a barrier reef system. You can scuba dive on it, and there's the part that we've mapped is this sort of pink zone right at the end of the ROV transect. So that's our aim today. That whole transect length is about 1 kilometre, and Nelson's just zooming out. You can see the Ribbon Reef 5. It's about 6 kilometres long, so it's got this sort of almost teardrop shape. So in north-south distance is about 6 kilometres long. It's about 0.6 to 1 kilometre wide, just to give you a sense of scale. And you can see it's growing right on the edge of the Great Barrier Reef. This is the outer shelf of the Great Barrier Reef. Now the Australian mainland is about 50 kilometres further west from here, and I think the closest main town is Cooktown, which you can visit, and it's not far from where I live in Cairns. But the whole goal of this is to climb up this what technically called a shelf-incised canyon. And right now we're about a third of the way up it. So we've still got a little ways to go. We expect to be in the water for at least another hour. We've completed our 500 metre transect, which we are doing systematically in every one of these canyons. And we're just going to keep climbing up, looking at the seafloor. And so for us, it's really exciting. This is the first time we've ever had, really systematic vision taken in the heads of the Ribbon Reef Canyon. So this is really, really unique. And of course, we're in the deeper part of the canyon below us, in about 1,400 metres yesterday, and the day before that we're in nearly 1,800 metres, so nearly 2 kilometres deep. So we started in the deeper, the mouth of the canyon, and we went to the middle zone. That was yesterday's dive, and now we're at the head of the canyon. And the goal really is, I guess the culmination of this transect will be to climb up the very, very steep vertical face that's directly in front of Ribbon Reef 5. So if you're listening to myself and Jodie Webster speaking earlier, Jodie and I have been working in this area for well over a decade, mapping, and we've put autonomous underwater vehicles in front of this particular reef, Ribbon Reef 5. We did that back in 2007, and they were complete robots, untethered, but it only went down to around about 100 meters back then. So this is the first time we've had cameras down at this depth before, and currently we're at 400 meters. And so it's just been quite fascinating to see the marine life that is all over the seafloor here. If not the animals themselves, certainly the tracks and trails that they leave behind. And this is this German word, life traces, called Lebensspuren, and I guess it's been a feature of all the canyon vision that we've taken so far. Everywhere we've been where sediment accumulates, there's lots of marine life that lives here, and you can start classifying it into different types of Lebensspuren. So just in the lower left there are burrows. They look fairly old to me. But other animals start using those burrows beyond the original inhabitants. So we actually saw some isopods before. These are giant isopods. They look like a pill bug. They're about the size of my forearm. And I would say a lot of these very large burrows are made by these isopods. And then other animals start to use them. And we have some rock. Okay, this is great. All right. So the question is, what kind of rock? Is it basement rock like we saw yesterday, which is this very, very hard mudstone, probably many millions of years old? And this is quite interesting. So we're just going to have a bit of a look around here, and there's all kinds of marine life attached to this. So if we look in here, we can see feather stars. These are Comatulid crinoids down on the bottom left. We've got beautiful fish just hovering there, a couple of other kinds of fish. Unfortunately, we just don't have too many fish experts on board, but we do reach out to anyone watching from ashore to help us identify these animals. And I'm just going to zoom in and see if I can make out what kind of marine life we got here. We've definitely got lots of corals. Now there could be, looks like lots of solitary corals clustered in around on the wall. Yep, they're looks like individual cup corals. Some of them dead. They're the more white ones, and we have. Oops, sorry, I'm driving the joystick here, and it's not going all that smoothly. But gee, we've got a real collection of stuff here. But looking at the rock itself, it appears to be stratified, and my gut feeling here is that we're looking at some kind of reefal substrate. So this would have been originally part of the coral reef, and I'm hazarding it may be just a large boulder that's broken off and has now tumbled down slope. But we do have lots of marine life. I'm just going to zoom out so you can get that bigger picture again. Looks like we have soft corals over on the right hand side. We've got a feather star, yellow feather star down the bottom. Lots of soft corals. I'm just going to zoom in on some sort of large, almost oyster-like animals. A bit hard to make out there. They could be bivalves attached to the various layers, but it definitely has this layering effect. Now this is typically what you see in these old coral reefs, when they've been eroded away. And if you scuba dive, certainly in tropical areas, if you're swimming up the steep edges of these coral reefs, this is kind of the very typical limestone layering that you see. And it also has this sort of dark iron oxide colouring. So down at these depths, we're in 400 metres of water. We will see this oxide colouring still, but as we get shallower, it'll slowly disappear. Now I'm just going to zoom down. This is an Anthomastus soft coral, Gorgon's head coral. We see lots of them in the Coral Sea, and they have this classic sort of spouting, budding look about them. These are an octocoral. So that's the pink one right in the centre. And then we got looks like a cup coral over on the right hand side. So I'll just zoom out again and see what else we can see. But yeah, beautiful little habitat. Lots of individual cup corals. These are these sort of white, almost flower-like, individual flower-like animals just sitting out from the rock. Yeah, and nice to see James Reimer is watching. James is from Ryukyu's in Japan. He joined us yesterday, so nice to have you with us, James. And we've got Jeremy, who's guiding some of the sampling. And what are we sampling here, Jeremy?
So we're going to suck up some cup corals.
Okay, we're going for some cup corals. So, okay, these little white ones below the Anthomastus. Just slowly turning right here, and you can see just above the laser beams the cup corals. The tentacles are extended, and you've got this very nice pink Anthomastus soft coral just to the left of them. Oh, look at that! It looks like an anemone up the top. I'll just zoom out a bit so you can. No, it's just beyond the top of the screen. Oh, there we go. That's the one we're after. You can see the coral lights. These are the septa separating the different parts of this polyp. Really distinct septa, quite thin in size. It's probably maybe only one centimetre across. And there it goes. And we got the entire thing. Thanks very much, gents. We've got J-Rod and Adam on the controls at the moment.
Do you want a group or individuals?
Grab just like whatever comes in with the screen.
John?
I'll try to get one at a time. That's okay with you?
Yeah, this is looks like it's budding out from it. Jeremy, is it?
The one that they're attached to.
No, it's okay.
You can see the vacuum working. It's just a little slurp, and here it goes. So this looks like it's got polyps budding out from the stem of the larger colony, which is quite interesting. And of course, corals can do this as asexual reproduction. You know, they're like clones of themselves, and they just bud off. They also have sexual reproduction where they spit out, sperm and eggs. This is part of the mass spawning event that you have on the Great Barrier Reef. Talking about the shallow water corals here. These are deep water, cold water corals. And I actually know nothing about their reproduction down in these depths, but certainly if you live in North Queensland, you have the opportunity to be part, to go and watch mass spawning event. I've done it many times, where you go out at night time, usually the full moon in October or November and possibly December. At night time, it's a nighttime dive. You can actually see this mass spawning event. It's one of the great wonders of the natural world, where many of the different species of coral all spawn at the same time over just a number of nights, and the water column is, like they describe, an underwater snowstorm, and you just get billions and billions of eggs and sperm all being pushed out into the water at the same time, and they generally they're positively buoyant. They drift up to the surface, and to be scuba diving in the water column when this is happening is quite something. So just zooming out a little bit here. Yeah, okay, so Adam's taking charge. Yep. Yeah, and just to the right, now this looks like some kind of bivalve. You know, we have oysters attached to rock, they're a bivalve. The cemented half will go onto the rock. That looks bivalvish to me. So these are really, really small little corals. We're just nudging them. There we go. So
it's into that cave. You see that big one underneath the cluster? Yeah. See the tentacles coming out?
So cup corals, Jeremy? Yeah, okay. So small cup corals. And it looks like an anemone just behind the tube. It's just hanging in that cave there.
So back now?
Yeah, there's a soft coral there.
That's an Anthomastus, just Gorgon's head coral, very close to where the tube is at the moment. And we've seen some really big ones of these out in the previous expedition out in the Coral Sea. We're heading back out into the offshore Coral Sea area after we leave the Great Barrier Reef margin. But at times we've seen really big colonies of these clustered on rocks. Very, very distinctive. These are an octocoral. You can actually see the tentacles now retracting. Would go, Kathy. Yep, no worries. And we're doing some sampling for Kathy McFadden. Yep, and so she's one of our octocoral collaborators. So doing that here, see if we can do it. You know, it looks like the base of these are really quite attached to the rock. So a bit of a nudge. Got that vacuum pump going. There we go. Whoop. And it's just held on by a little thread of flesh. There it goes.
That works.
Yep, thanks very much.
Is it still going?
The one's okay. Yeah, the one's okay. All right, that was a good rock.
Yeah, and Anthomastus. ANTHOMASTUS. So just look it up on the internet. We have seen lots of these. Very classic, beautiful octocoral, mainly because the tentacles, you know, the polyps are really large. You know, it's very obvious. You can see the whole animal quite clearly.
Yeah, we don't want too much sediment.
No, that's why I'm just barely catching it.
Okay. Thanks for letting me grab this.
There we go.
Yeah, this will go directly towards sequencing.
Oh, I love it. Okay.
Okay, that's all good. So we've done a bit of sampling so far. We have to keep a move on. We've still got a ways to go. We're not quite halfway along this transect with probably a lot of sampling ahead of us. As we get higher up the head of this canyon, as things get shallow, we're most definitely going to see more of these this exposed rock, and of course all the animal life that's attached to it. And of course, from a geological perspective, we are very, very interested in seeing in situ growing, well, actually, you know, fossil reef that was once growing in these deeper depths. Now, that would actually point to the origin of the Great Barrier Reef. We may even have a chance to take a sample, a rock sample, and Jodie Webster can do all kinds of geochem magic, I call it, on those rocks. You can start, because these living animals like coral lived within the water, and locked up in the skeleton is that geochemical signal of the ocean at that time when they were alive. And so you can do things like, you can look at, well, certainly their calcium carbonate content, their aragonite content. You can start doing things like looking at their water temperature that was recorded when they were alive. Now, that's really interesting to know if we are talking about the origin of the Great Barrier Reef. But, I did think this was actually a big boulder, but in fact, what we are seeing looks like big outcrop, a big outcrop of limestone. So this could in fact be the very, very base of the Great Barrier Reef.
400 metres, so that's feasible.
We're at 400 metres. It's a bit deeper than I would have expected, because from a seafloor shape perspective, from the mapping that we've done, I wouldn't have expected to see something like this straight away. We might just reach out to Jody Webster just to get his take on this. We're just having a discussion in the control room here, reaching out to our to Jody Webster at University of Sydney to get a sense of what we're looking at here. It does look like what we call framework coral reef. This is, if it is, that means this is the deepest exposed part of the Great Barrier Reef, and really significant. Much deeper than we probably would have expected. But if it is, we probably should get a rock sample of it. So we've got some other.
Break off a piece of it, or-
Yeah, I think.
Yeah, we're just having a chat with Jody.
Not entirely sure, but thinks it's worth trying for a sample anyway.
Okay,
We'll go for a grab.
we're going to try and get a piece. And like I said, we want to find the deepest part of the Great Barrier Reef. Now, all we have from the evidence so far is from this long coral core that Jody was talking about. This is the Ribbon Reef 5 core that was taken in 1995. They basically put a jack-up drill rig on Ribbon Reef 5, which is only about a kilometre west of where we are here, and then they drilled down in right through the middle of the reef, and they pull out this great long 200 metre long core. And so Jody's been working on this for some time, and he was describing it from the workshop where he is in University of Sydney. He's actually got that core laid out in front of him. So now we are seeing what appears to be limestone. It looks very old. It's quite eroded. Lots and lots of holes and things, but this is kind of typical limestone, old limestone environment. And what the pilots are going to do is just use the manipulator arm to find like a thin area and see if they can kind of twist a piece off. So you can see there's quite a bit of pressure.
Screen from questions. Right here.
Can you follow my
Away from the script right now. Yeah, no worries. Yeah, so Jody's going to be he's watching through YouTube and the like. But anyway, we're going to try and we might be able to try and get Jody on at some point so he can talk about what we're seeing here. Anyway, going to try and break a piece off. Now this would be really significant if we can get a bit of coral here. And of course, the rock itself is covered in all kinds of cup corals and lots of marine life here. But this is really quite amazing to think that There we go. We have a piece. Let's see how big it is. No, we missed that one. Yeah, and if you look in the, there's all kinds of life here. We got some little hermit crab right in the foreground. Cup corals as well. You can actually see the white of the rock that's been broken off. Got brill stars in the background. Perhaps another black coral. Keep Jeremy interested. He's not into rocks himself. He's more the black coral guy. But anyway, we're going to persevere here. Marty, are you able to just? Do you know if has Jody replied at all yet? Has he replied on Slack?
Yeah, so we're not 100% sure at the moment if this is an in situ outcrop or if it's a big block that's come down.
It looks too big.
It looks in situ.
It looks too big for a boulder. I mean, look at it. It just seems to be reaching off into the distance. So this is really significant. It'd be great to get Jody on here.
Operate the camera, sorry, for J-Rod.
Oh, sorry.
Yeah.
So, sorry, coming down. Oh, wrong way. Sorry, gents. Coming down and over. Oh. Coming over. Is that okay?
Yeah, we're down.
Yeah. Push it down. Sorry.
Try to get you back down.
There we go. Well done.
I'm going to push it out some more.
Okay, so we've just broken a piece off. Now this is the
B.
B.
B, yes, thank you.
There we go, having a look. It's fairly eroded. If we need that face, that's probably what we need there. Now that's important because bioerosion can completely change the geochemical signature of rock when you're doing age dating with carbon-14 and some of these other age techniques. But what we might, let's see, I wonder if Jody can join us.
Am I clear to drop it, or do you want to look at it?
You can drop it, thanks.
So we're just having we're just seeing if we can reach out to Jody. Yep. I'm just going to step off for a moment to grab a headset. Okay, back in the audio seat, this is Robin Beeman from James Cook University, and we've got a very full control room. And we've got Chris on controls with J-Rod, and we're trying to reach out to Jodie Webster at University of Sydney, who is our other colleague. Unfortunately couldn't join us for this voyage, but Jodie and I have done a lot of work together over the years, including well over 10 years of really comprehensive mapping in this area. So this is the first time we've had cameras down at this depth, and for Jodie it's been really a long-term goal to understand the origin of the Great Barrier Reef. And
Just another rock.
one of the really great things we can do with this voyage is to I guess look at these canyons, this extensive complex system of canyons that lie in the deep Great Barrier Reef to seaward of the shallow reefs, and drive up them until we go from bedrock, the ancient Australian continent upon which the Great Barrier Reef grows. And we saw evidence of this yesterday. We got a piece of that rock. It's probably very, very old, millions of years old. And this is the first limestone looking rock. There's a beautiful fish swimming past now. The first, what we think is framework reef. And it's kind of unusual. We're talking at 400 meters depth, and which is a bit deeper than we expected. But let's wonder if we can get Jody to join us online so that we can speak to each other and just talk about what we're seeing here. And I think we've got Jody.
Jody, can you hear me? Testing 1, 2, 3, 3, 2, 1.
Jody, can you hear me? Testing 1, 2, 3, 3, 2, 1.
Hello, hello.
Yep, I can hear you, Jody.
B, which is the same one.
Good job.
Can you turn the speaker off?
All right. Clear.
I can hear you loud and clear.
One on the B1.
B.
Yeah, thanks, Jody. We're just getting the audio set up here in the control room. And glad you can join us, because I've been talking about the fossil reef, what we expect at the end of the transect, and here we are. We're at 400 meters. So we thought this was, we thought it was originally a boulder, but it is far too big to be a boulder. Something that's just tumbled down slope. This is, to us, this is a large outcrop.
Hello.
Can hear you. Can hear you loud and clear. So we're just getting our audio systems sorted.
Oh, hey guys, can you hear me?
But it is far too big.
Hey guys. Hello.
Boulder, something that's just tumbled down slope. This is, to us, this is a large outcrop. Can hear you.
Oh, Rob, sorry mate, can you hear me?
Yeah.
All right, mate, thank you.
All right, there's a delay.
Yeah, this is quite amazing.
We haven't quite got the audio sorted yet. Sorted.
Oh, hey guys, can you hear me? Hey guys. Hello.
Jody, can you hear me?
Hi, Rob. Yes, I can hear you now, mate.
Okay, that's good. There was a bit of feedback going on there, mainly because of the delay.
Yeah, sorry, yeah.
Okay, sorry to bring you back a little bit earlier, but I think this is pretty exciting. This is really clearly quite different. This looks like stratified limestone, but we're at 400 meters, and I can't quite get my head around this.
Yeah, it's a bit deeper than we would have anticipated. I guess there's a couple of possibilities. As you say, one possibility, it is a very large block that has fallen off and obviously settled thousands of years ago, where we're seeing that some exposed surface of the block. Another possibility
this is not a block, Jody. This is not a block.
Well, then we have some sort of in situ deposits. Yeah.
Okay, but when you think,
looks like there's structure.
Yeah, so when you think of modern reef morphology, there is always a scree slope in front, and so maybe we are looking at the layered scree slope that is now cemented and solidified, and you can see the layering. But this is definitely not basement. This is definitely not boulders. This is outcrop. This is in situ outcrop. But I don't know if you notice, it does look almost layered, like one layer on top of the other. So that might speak to a kind of a scree slope effect in front of the old, the Great Barrier Reef as it is. So we're still going down. Looks like the base of it now. So the ROV guys are just kind of dropping down because it's covered in marine life. It's just really quite spectacular. Got a lovely little lobster in front of us.
I saw you guys got a couple of pieces of rock you're able to break off. Is that correct?
Yeah, we did just before you joined us. And that's important, of course, because this is all about sampling. And just tell us what you can do with that rock, Jody. I mean, this is really important.
Well, one of the questions we're just posing here is it in situ framework? Is it material that's been redeposited and transported down slope? We may be able to see indicators within those rock fragments, and whether we're seeing framework material or jumbled pieces of rubble with, for example, lots of finer matrix. So there may be indicators within the rocks themselves which can tell us in combination with the morphology of the outcrop, that can tell us the origin of what we're looking at.
Yeah, well, you've got the same view as I have. You can actually see this kind of layering. I mean, it is very bio-eroded. There's lots of little cavities and things like that. I mean, if this is ancient Great Barrier Reef coral, I mean, we're talking, you know, several 100,000 years of age create this. So, what sort of aging techniques would you use to try and get a sense of how old this is?
Well, one of the techniques is uranium thorium dating.
Is it octopus? Sorry, mate. I think we got an octopus hiding in a hole here. Can we just zoom in, guys?
Yeah.
Yeah. Oh, it's lovely. All right. So, sorry. I'm in charge of the zoom camera. Let's see if I can do it. Well, sorry. And I'm just going to try and drive forward a little bit. I'm sure my son could actually do a much better job than me. Here we go. I'll just hold it still. You could just see its movement. You know, I guess this is the beauty of, we're getting that geological story. We're getting this beautiful biology story as well. I could just see some movement under the rock, and you know, it was more too fluid to be a so it's. So I can. Whoop. A little bit more.
Oh, there we go.
Gonna zoom in. Whoop, sorry everyone. It's just. There we go. I'll kind of hold it there. It's just moving slowly along underneath the this overhang. I wonder if we're looking at the eyes here. These little white. Yeah, probably the eyes. The main body is actually coiled up underneath.
Up there.
Yeah, okay. Okay, the pilots can do it much better than I can. I tend to overshoot a little bit. The other thing is, we have got a lot of overlying sediment. That's this, you know, rain of carbonate sediment that's coming down from the shallow coral reefs above. So as soon as we hold still, it tends to throw it up a bit. But you know, just getting back to what this could be, Jody, you know, it does look layered. You know, what could create?
Yeah.
I guess this
layered and also inclined. So you're getting quite steeply inclined layering within the outcrop. So I guess if I was to guess, I would say one of your points about it being a cemented sort of proximal scree slope would probably be a good bet at this stage.
Yeah, you know, like if you scuba dive on a modern reef, you know, in the shallow waters, you can always see this rubble that's kind of accumulating at the base of the slope where it just starts moving away from the edge of the reef. So, you know, this could be a much older version of it. But you know, here we are at 400 meters. It's too big an area to just simply be one boulder that's rolled down the slope. Because that's important. If anyone's thinking about, you know, if you're a geologist, you're actually looking for outcrop. You're looking for something that's in situ, not something that's just tumbled down slope, because you get the whole message. Your interpretation is entirely wrong if you're making all your geology on some boulder that's tumbled down slope. Oh, actually, this is a bamboo coral. You can actually see the nodes on it, right? This is living bamboo coral. Really quite pretty. You can see all the different polyps up it. But, you know, if you bring your eye in, you can see the little nodes. They're several centimeters long, and each node's joined together, almost like, well, bamboo, you know. So when they die on the seafloor. So, Jody, if you recall, back in 2007, you know, back in 18 odd years ago, when we didn't have the benefit of cameras, but we put dredge down, you know, we put a big heavy rock dredge down on the seafloor in the Queensland Trough, you know, oh, look, great view, guys. How's that? This is the octopus. And you can see little photophores. So and little protuberances around the eyes. Can you see almost like little tentacles? So so an octopus, this octopus have the ability to change the shape and colouring, the pigments within the photophores, these tiny little like cells all over the body. It also change the muscularity of the cells all over its body, so they can assume the shape of sorts of whatever is they're hiding upon, right? So if it's a flat seafloor, they can flatten themselves, can make the colouring exactly the same as what's around them. And we are in this kind of area. See the beautiful colouring of the cells and protuberances. So we've got we're going to have to move along, I think, guys, but this is spectacular both geologically and biologically. There's your broad view. This is not boulder, this is really big outcrop of some kind of cemented limestone, lobe-like.
Is there
lobes?
Rob, is there? Can we see like is there an expression on the multi-beam data of this?
No, not really.
Is there anything that
Yeah, okay. I'll just reach out to Viet, who's over in the controls. Viet, are you able to bring up the 3D view, and we can just have a quick look at where we are in the transect according to our 3D perspective, and that. But I guess the short answer is no. This is not what we in our 3D model. We're just having a bit of a play with the inset views here. And we're going to have to start moving up slope, but still have that very big steep... here we go. Thanks, Viet. Okay, so you can see the inset view there now. We're at the head of that canyon, and we're working up to Ribbon Reef 5, where you got that big 200-metre long sediment core right through the middle of that reef. And we're about halfway along it now. You just see sort of some bumps. Do you see some sort of bumps? Now, that could be what we're on at the moment.
Yeah.
So if those
sort of
bumps...
midway up the slope.
Yeah. Okay, so we might have a bit of a surface expression of what we're seeing in our camera view at the moment. Okay, well, that's good to know. We've got a nice looking... looks like a bannerfish, I think. Bannerfish, Dean? Oh, it's not a bannerfish. Sorry. Oh, and we've got a coffin fish on the left. This is a coffin fish, and we have seen them before. And I'm just going to come left a little bit with the joystick. I'm just going to see if I can zoom in and coming left a little bit more. Fairly sedentary. They don't actually move around much. I've seen them with some beautiful coloration. One's mostly yellow with... Oh, we lost it with the... There's quite a layering of just fine sediments draping over this this hard rock limestone. Zoom back out again. It gives us that sort of broader perspective. So I hope people are watching and just picking up all this amazing stuff. Seeing too, all kinds of surprises here. But geologically, very, very curious that we're finding down at... we're just past 400, we're just a bit shallower than 400 metres. Rock is covered in these little white cup corals, and we're seeing fair few fish. So coffin fish just sitting there quietly, and they do not know the name of this other fish that's sort of swimming near the laser beam now. It looked like a bannerfish to me. A fish expert, we have to reach out to our Army colleagues to name to a face here, but I do know this coffin fish because we've seen quite a few of them out in the deep coral sea. We have one now. They just sit there quietly, but they had this. Their pelvic fins look almost like hands, and when they start moving, they kind of do this sort of walking along the seafloor. It looks almost human-like. Pretty bizarre. There we go. The guys are much better at driving the camera controls than I am. grumpy fish, not very happy. At his morning coffee.
It does look grumpy.
So Jody, just talking about ahead of us. Okay, so let's just assume this is like for as a hypothesis that this is a cemented scree slope. It would be very, very old. It is certainly hard and cemented. You can see it extends quite a fair distance. So it's not a boulder. And then we're back into this white sand draped sea floor. We can assume that the rock that is that let's say call it a scree slope is underlying this, right? So this is now a layer of soft sediments over the top. But as we get towards the end of the transect, I expect we're going to see the gradient will rapidly increase. So it's going to go from gentle rise to suddenly really, really steep. It'll probably be around about, I think that's about the 200 meter mark. And, those early geologist descriptions call it the wall. Maybe you want to talk about the that garage-built submarine that just explored this area. That's quite a tale.
Yeah. Yes, indeed. So in the 80s, scientists called Charlie Phipps from the University of Technology, Peter Davies, a very prominent professor of geology here, and also worked for Geoscience Australia, and in fact Professor David Hockley, again a very, very prominent reef worker in the 80s and 90s, but really did some of the absolute foundational work in the 70s, 80s, and 90s and beyond on the Great Barrier Reef and understanding its evolution. And they were here with a manned submersible, I think, in 1984, around that time. It was called the Platypus, obviously kind of lower tech than what would be available today, and so it was pretty heroic. And they did a series of dives off the front of Ribbon Reef down to about 200 meters. So we're obviously now, and have been for the last hour or so, looking at completely, undiscovered seabed. But they did a series of dives up the wall and made some really important and basic observations of what we know, and of an almost sheer cliff populated with some really complicated layering and stratigraphy, and also some really prominent caves, or what they described as caves, at about 160 meters and 120 meters, which they had interpreted to be the remnants or evidence of previous lower sea levels during the last ice age. And so with a very, very complicated history that based on the drill core which was collected subsequently, we know that this part of the reef is at least less than about 700, 800,000 years based on the work that has been done, and the actual main reef section, probably 4 to 5, 600,000 years. As I said, we don't know exactly how old it is, but we've had this really complicated history of high sea level, low sea level, high sea level, and it's thought that steep wall grew through this phase of rapid reef growth during subsequent sea level rise and then fall. It looks like we're seeing some nice biota here.
That's right. We've got Jeremy here, our black coral expert. He's having a close look, and just because we're doing live from the control room on the Falkor from the ROV team, they wanted to reach out and say a big hello to Diane watching from ashore, and of course to all of our people watching ashore on YouTube and Facebook. It's great to have you joining us. To let people know, we're edge of the northern Great Barrier Reef at a place called Ribbon Reef 5, and it's about 170 kilometres due north of Cairns, northeast Australia. At sea now for about 3 days. This expedition is 2 weeks long, and we're spending this first week just working on the edge of the Great Barrier Reef, the deep Great Barrier Reef in these canyons, with the idea to do systematic sampling and vision within the deeper parts of the canyons, the mid-zone, and now the head of canyons. And the head of the canyons is really important from, as Jodie's been describing this, we're looking for this exposed older barrier reef. And he just to sort of explain some of the terminology, we mentioned you mentioned framework before. Can you just talk about what framework means from a stand?
Yeah. So when we're looking at thinking about what we see in the cores and outcrops, reefs are built by a combination of what we call the framework builders, and that could be the corals, the algae, and other forms of sediment that act to bind the reef structure together. So you could think about my fingers as branches encrusted by crustose coralline algae, and then that might die. Then we might get another coral that then settles out as a planular larva and grows on top, and then it grows again and again and again. And so over millennia, that builds up a limestone that is framework or corals, stuck together in essentially growth position, as opposed to where we might get what's called detrital material, which is made up of the breakdown products of sand, gravel, and rubble pieces, which may be then cemented and built up and then forming another type of limestone. So we have framework types and basically detrital broken pieces which might be cemented together. Different types of reef materials.
So Jodie, I was reading that paper that you wrote with Humboldt back in 2017, and we're talking about that great long sediment core through the middle of Ribbon Reef 5, which is less than a kilometre away from where we are. And you were saying that you found distinct reef. The reef sequence really turned on about 130 metres down in the bottom of that coral core. But you found limestone reefal material deeper than that in the core, but around about that 130 metres down in that coral core that you saw the reef turn-on sequence really start then. Do you want to talk a bit about that?
Yeah, if we take the 200-metre section, what we know is there's a clear reef coral dominated section, and then what we call a non-reef section, which is more skeletal material, cemented sand stuck together. And the really important point is about 137 metres or so, where we see the first appearance of the really framework building, branching, in fact, acroporiforms, the ones that we see today that are dominant on a lot of outer reefs, building the really branching, complicated fabrics. And we see those turning on, or at least starting from that stratigraphic level. And then from that, we see about 6 or 7 different cycles of different reefs with a repetition of different community types as we go up core.
Yeah, you're watching this, Jodie? Yeah, sorry to interrupt. We're just...
Oh, yeah.
Everyone's having a bit of a cheer here. Of course, all these are sessile animals, these corals, they need to attach to something, and it looks all like a muddy environment, yes. But in fact, you're a little bit, your eye is that it just thinks that there's this great thin layer of, it's all sediment, but in fact it's not. We are actually looking at a lot of rubble, hard rubble, with a veneer of soft sediments over the top, and so these corals, these living corals, can actually attach to them. Did you see as their claw closed, you could just see there's rubble all through here?
Yeah, there's a lot of coarse material, and I guess, the biota, the living biota, those living deep water corals are profiting and growing off those hard substrates. So I saw you actually pull one of these up earlier in the dive with the rock attached. So this is really a bonus for the bio team and also the geo team.
Two samples together. But talk about the sequencing a bit more. This repeated, this multi-cyclic sequencing from growth and demise and growth and demise. This is fascinating. You and I are quite familiar with this, but if you try and explain this to people to understand, the Great Barrier Reef has actually grown and died maybe 6 to 8 different times as sea levels rise and fall over, that Pleistocene period.
Yeah. So, driven by, natural orbital cycles, which, run at about 100,000 year cyclicity. We've got the world is transitioning between glacial or cold and low sea level. And then also, as sea level then rises and the ice sheets decay, the world warms, sea levels rise, and we move into the transgression or rising sea level, and that floods back over the previous position of the shelf, and we turn the reef on again, and another cycle grows. And of course, that goes for another 10,000 years or so, depending on how long that cycle is, and then sea level falls as the world descends into another glacial period. And the reef in that place is completely and utterly wiped out and destroyed, and the system migrates from that position out to the edge of the continental shelf. And in fact, we're actually now hopefully heading towards one of those spots of where does the reef go during these lower sea level periods. So we know, based on the multi-beam mapping that we did, and that there's a distinct terrace, or what we think is a paleo reef, at about 50 meters and in fact 70 meters, and they represent, you can see it further up on the red. That represents the time and the place where sea level went and where the reef went during some of those lower sea level periods.
That's right. We've got the inset in there now. You can see the end of the transect. It goes almost vertical. So this is what Jody, what you're talking about, Jody, with this that steep part. That is where sea levels had dropped during these glacial periods, where global sea levels drop around about 100 meters, and of course they rise again.
118 meters.
118 meters. Yeah, that's right. We're quite accurate about this now, just from your IODP work.
Yeah.
So,
and it's worth to note, actually, that we actually had the IODP for the audience at home and the people on Facebook. That's the International Ocean Discovery Project. So this is a different program to what was done to drill the Ribbon Reef 5 core, but this was actually bringing in a large drill ship to drill the 70 meter and 50 meter structure that we're hopefully going to visit. And we did this just a little bit further to the south off Ribbon Reef 4. And we recovered corals that were growing about 30,000 years ago, and 15,000 years ago, and 10,000 years ago, which were drilled from that shelf edge area.
Yeah, look, it's just fascinating to think that all of this work that we've been, over the decade or so, the mapping, the drilling, the coring that's been done, to try and understand these fossil reefs. It's all going back to, I guess, trying to understand the responses of the Great Barrier Reef through each one of these cycles, and that it gives us context about, the sort of current state we're in. Here's a brittle star. Oh no, sorry, it's a squat lobster having a bit of a crawl across the surface here. But it is all about giving context. Looking at corals as a library story of our sea level change and climate change, so we can just put into context where we are at the moment.
Yeah, that's exactly right. And the corals, particularly, we've discussed how they can be used to reconstruct the position of past sea level. They're fantastic dipsticks, to see where sea level is, but they're also really excellent recorders of sea surface temperature. Based on some of the drilling work we've done we've been able to collect well-preserved corals, and my colleagues have looked at the ratio of different trace elements within the skeletons and different isotopes, and then they're able to reconstruct sea surface temperature. And what they've been able to show is that during the last ice age, it was perhaps 4 or 5 degrees colder about 20,000 years ago, and so you've had about, again, 4 or 5 degrees of warming over this period from the last ice age to when the Great Barrier Reef turned on, and about 9,000 years ago. So the reef has responded not only to large sea level changes, but also quite large temperature changes. But this is in the context of the modern situation, where we've had maybe 0.7 of a degree, maybe a degree of sea surface temperature rise in the last 100 years or so, compared with 4 or 5 degrees in the last 10,000 years. So it's really that's very fast, relative to this geologic sort of baseline or context, and that's one of the values of looking at these fossil systems.
Yeah, you're talking about those past temperatures and that. You get this directly from the coral skeleton itself, can't you?
Yes. So the ratio of, for example, strontium to calcium within the skeleton. So, my part of the IODP science team published a really interesting paper a couple of years ago now that reconstructed the temperature over that period. And in fact, there's another really important paper that's just now in revision, which is sort of putting some more and more accurate constraints on those temperature data. So that's really important.
Yeah, so we just got Jeremy here. He's just driving in. We're having another look, another black coral, potentially, because this is a big sampling exercise as well. It's not just about vision and mapping. We actually do want to actually take samples. So, Jeremy, we've got a fairly full set of boxes already, but in fact, the end of the dive, the top of this canyon, which is, for us, is really, really exciting geologically, will also probably be quite amazing biologically, because as you get shallower, it's quite, well understood that, you get more biology, more biota as you get into the warmer, more sunlit zone. But I think the thing we need to keep repeating here, Jody, is the head of this particular canyon ends at this vertical face. We hope to see actually the base of the Great Barrier Reef, this framework coral, the stacked strata, and I hope we can actually take a sample. We have this drill that the guys here have built in the last few days, and we've already tested it out. Yesterday we actually drilled a plug of basement rock, like the ancient Australian continent sticking out in the middle part of this canyon. Yeah. And what we want to see is the limestone on top of it, growing on top of it. So we hope that will be in this vertical base that we'll find at the end of this growing up.
Yeah, and I would really love to try and use that drill system on the 70-meter feature and possibly the 50-meter structure on the substrate itself, because we weren't able to come here with the IODP drill ship, because we actually had some technical problems, and we were left with, I remember distinctly, we were left with a choice. We had very little time left in the expedition, and we had some quite catastrophic technical issues, and we had a choice of, well, okay, we have to go further south to the last site of Cairns. And I remember, because it was all about coming to the front of Ribbon 5, because what I wanted to do was, we had the big Ribbon Reef 5 drill hole. I wanted to put a whole series of these smaller drill holes in the 4 reef slope. And I remember driving away, almost crying, thinking, oh, I'll never get to come back. And so it would be fantastic to be able to see those shelf edge mesophotic reefs and be able to sample them if we can.
Yeah, that's right. We have sought out these big vertical walls because it's almost like looking at an X-ray. Like there's a couple of ways you can look into the deep Earth. You can use seismic, like geophysical sonar to actually look, internally into the earth. You can use cores, as you've described, or you can come up to a nice big steep rock face with a world-class ROV and an amazing research ship and drive up the wall and have a look at it that way. But, actually getting round to this point is such a long road. People may not realise just how much effort it has taken, Jody and I and everybody else here, to get to this point. So just to take some background here, Jody and I got our first expedition together in 2007, Australia's Marine National Facility vessel. This was the Southern Surveyor, decommissioned, and it's been replaced by the RV Investigator. But then 2007, we were fortunate to be ship time. We got nearly a month on there, and it was in September 2007, Jody, and we arrived on the Ribbon Reefs. We had Stephen Williams and Oscar Pizarro from the Australian Centre for Field Robotics. We had their new Sirius AUV, the autonomous underwater vehicle. It's a true robot. It was a really bold expedition to go out and look for these drowned reefs, these ancient drowned reefs. This place we came to was out here. Of this previous history of this long drill core that was taken in 1995. So we keep coming back to this Ribbon Reef number 5 because it's probably the most studied reef in all of the Great Barrier Reef as far as deep geological time, and so it makes sense to keep coming back to the same place. But I just have to talk that background a little bit more. While we were actually trying to find those drowned reefs, which is a bit shallow up from where we are now, Captain wasn't going to do any mapping to the reef at night time. And so we were told, right, so come up with a mapping plan that's out in this deeper water on the continental slope in front of the Great Barrier Reef. And so our very, very first night, very first multi-beam line, we went across the continental slope here, and instead of being this big flat slope, which is what everyone thought was there, covered in canyons. Mind-blowing. I'll never forget the excitement of those first pictures, those first 3D pictures coming back of the deep Great Barrier Reef. We're talking sonar mapping here that generates these beautiful three-dimensional pictures, which we're now using to help us work out where to put our ROV. But we only did this back in 2007. That's only 13 years ago. Prior to that, we didn't even know these canyons existed. Of course, it's just generated all of this research, Jody. I want to talk about some of the work that Angel has done. That's really impressive.
Yes. So as you said, sort of almost as a byproduct, really, of that sort of not being able to work close to the reef at night. We were able to just map more and more of the deeper slopes, revealing this incredible network of submarine landslides, as well as the deep canyons. And we came out here to actually, as you say, study the cluster reefs and also the mesophotic reefs. But as a wonderful sort of byproduct, we were able to see the seafloor for the first time in the deeper margins. And some decades before, people had taken deep sediment cores in the basin and the slopes, but they really didn't have a good map really to put those cores into any sort of context. So you could see the connections, for example, between the shelf and the slope. And so working with our very close colleague, Dr. Angel Bernabeu from Granada, who's a fantastic marine geologist who's just been doing incredible work investigating the morphology of the canyon systems and the slopes. Also, we've been doing lots of work on trying to understand the delivery of sediments from the shelf through the canyons into the slope. So going back to some of these old cores that were collected by previous workers decades before, that we can now place into a really good spatial context. And that's revealed a lot more about the margin.
Yeah, but just getting these big ship expeditions up has been, it's a real challenge. Like, people may think that we just do this every second week, but in fact it's nothing like that. Most of these expeditions that Jody and I are talking about have been developed over many, many years, and we've been fortunate to get a number of these larger ship expeditions up, but generally they're years in the making. And the thing is, you can get out here and the weather could be bad, and captain doesn't want to go anywhere near the reef, or you have equipment breakdowns, and there's some real technical challenges to doing this kind of blue water research. This is the ship, like the Falkor here, is it's 80 meters long. Other vessels we've been on, like the Investigator, that's over 100 meters long. These are like little floating cities, but working out in the blue ocean here, everything's working against us. The weather, the wind, the waves. It's either tropical storms, cyclones, and then you finally get on site and have technical issues. So it's very much a patience game. You can't rush these things, and if you have technical challenges, I'm thinking of the ROV we have here, incredibly technical. There are 10 cameras on this thing. There's a live feed from the seafloor. There are fibre optic cables. We're putting high voltage down into cameras that's being streamed onto the ship, and then has to go through satellites ashore. Incredible technical challenges to overcome. I think we've been very, very fortunate, Jody, to have, achieved what we've done. But, there are a number of times we've got here on a vessel and we've had to leave. You're talking about sailing away and that IODP voyage. It's an international enterprise, bringing people in from all over the world to take part in this quest to understand the deep Great Barrier Reef. And when you finally get here and something happens and you can't do it, and you just have to say, all right, it's a long game. We are playing the long game here. Yes,
indeed, a very, very long lead time for a lot of these things. In fact, I was a graduate student when the plans were hatched to drill, for example, or put in the proposals for the IODP drilling of the Great Barrier Reef margin. So this was 2 decades ago. So those IODP expeditions can take a very, very long time to come to fruition, and even some of these other expeditions. And of course, this is in a sense what makes what's happening right now. Obviously, the other major challenge that is going on is the global pandemic, which has affected all facets of life, but also research, with many expeditions cancelled. Our own expedition cancelled, hopefully to be rescheduled soon. And
that's right. That's right. So, you and I had that Halimeda voyage, Halimeda being that green algae, and we were going to study this Halimeda ecosystem on the Great Barrier Reef shelf, and I think we put our first proposal in, over 3 years ago. We were granted the ship time 2 years ago, and then it's taken, like a year of really solid planning to get to, it was nearly April, wasn't it? And then that pandemic kicked in, and that was all cancelled. So it's a miracle we're out here on the Falkor. Falkor must be one of the few research ships anywhere in the world that's actually working right now. So everyone should be very, very proud of effort. Huge effort by the Schmidt Ocean Institute to get out here, because pretty much no one anywhere else, certainly in Australia, they're doing research like this. And I have to pinch myself. Here I am sitting in the control room, and we're flying up those canyons that we first mapped in 2007. And you're not here, but you're virtually here. So great to you're with us, Jody. Amazing, mate. yeah, just talking about the seafloor, it's getting a lot more rubbly. Seeing a lot more rubble.
Yeah.
Clearly, so I'm going to say there's sediment over the top of it. But it looks like you don't have to go down far, and you're basically hitting rock rubble and the like.
Mm.
And we got some
It's a really interesting
Go on.
sort of slope. Yeah. Oh, I was just going to say that, one of the goals that one of the outcomes of our work on the canyons and Angel's work, our Spanish colleague, is to really characterize the different types of canyon systems that we have. We have these canyons that go right up to the top of the shelf and that are connected to the shelf. We have similar shallow canyons like the one we're in now that go right up to almost the modern reef. And we have another style of canyon that we'll hopefully get to later in the expedition, where the canyon only reaches to the sort of upper slopes, and they're much, much deeper. So all these different styles of canyons, and to understand, the different types of sedimentation, like how active are they, what impact it has on the biota. So it's a really fantastic sort of natural experiment that we're doing.
Yeah, that's right. I mean, going back, we've now mapped well over 100 of these canyons along the northern Great Barrier Reef margin. There's still more to be done. My job is not finished by any means. I feel like, this is my role in life, is to map these canyons, and while there's still some yet to be mapped, I'm going to keep working on it. But yeah, so north of here, off Cape York Peninsula, we know there's canyons there. There's been one or two ship transects through, but there's still a lot of these unmapped. But of course, it's not enough just to map them. We will need to describe them, and this is what you were just talking about, how we've actually classified them. Now, there are shelf-incised canyons, ones that are very mature. The heads of the canyons are right up hard against the Great Barrier Reef, the shallow reefs themselves. Like, you could stand on the edge of those reefs, look down into the heads of the canyon, and this is one of the ones we're going up now. This afternoon, if we get enough time, we will mobilise to a different canyon. It's between Ribbon Reef 4 and Ribbon Reef 5, and so it's fully shelf connected. The head of it is the channel between one reef and another. So we may expect to see different kinds of marine life there, because the sediments that are coming directly off the wider shelf behind literally drain directly into that canyon. So this afternoon we're going to have another look, but on this one now we're well over halfway along our transect. See, the rubble is picking up. Lots of benthic life. We're seeing brittle stars, sea stars, lots of echinoderms, fish. It's definitely becoming more coarse, more rubbly. But eventually what I hope to see as we get near the head of the canyon, the gradient will sharply drive up. You can see the inset view. The very end of this transect, it will rise up steeply. This is probably around about that 200 metre mark. Right now we're at 330 metres, so about another 100 metres vertically up. We should see a real steep vertical rise. That's where we hope to see this, I guess, the ancient stratified layers, the framework, the X-ray view of ancient Great Barrier Reef. So what we might do is I might just stop there for the moment, Jody, and let someone else take over the audio, because we still probably have a good another half hour or so before we get into that steep vertical zone. I'll just have to look at this. This is really interesting. What do we got here? Jeremy, what are we looking at here? We got a tube anemone on the right. This is a cereanthid, and there's something quite strange in front of us here. I might just use my own. I might just do my zooming in myself. Is this a harp ctenophore? Is this a harp ctenophore? We got any YouTube experts watching in here? Or is it just simply some kind of weird holothurian number?
How large is it?
It's
There's
good
the
size.
lasers.
You can see the lasers. They're right in the center of the screen. It's 10 centimeters. This is a good 20 centimeters across. What are the people ashore saying? It's a harp ctenophore. This is a benthic ctenophore. So if anyone's been listening in before, we've got ctenophores. Generally, they're a pelagic. They're plankton. They've got these beautiful rows of cilia, and then there are these benthic ones, and they have this kind of harp shape. I'll just try and see if I can drive the view a little bit more. Pardon me if I kind of overshoot a little bit. Sorry, I'm just heading that, and I just turn right. Yeah, it is big. We have seen them before out on the offshore Coral Sea area, attached to coral rubble and the like, but not as big as this. This is really large, probably a good 20 centimeters long. But until someone told me it was a ctenophore, I wouldn't have believed them. This is really a strange looking animal. It's attached to the seafloor.
It's attached to some rock underneath the thin dusting.
Yeah, possibly. You can see the base of it, and it seems to be spread out. I'm not sure if people are sure, like on YouTube and like, can describe, like, can these things detach and move to another area if if it's unfavourable? Well, that's my question to the YouTube chat people. Really quite spectacular colour. I mean, good 20 centimeters long. Semi-translucent. Really quite a beautiful looking animal. So the pilots, we've got Chris. He's just next to me. He's zooming in. We're having a real close look. In fact, you can actually see almost like little like hairs running down. Ribbon, on the outside. So any word from ashore, guys? Can they detach? That's my question.
What a strange creature.
Yeah, I'll say. Yeah, so much weird marine life here. The ones I like, the things that I can't describe, like I just go, I don't know what that is. And, I've been looking at these ROV videos for a while now, and, I'm starting to get my eye in for various things, but, this is quite amazing. We have seen them before.
Holes in the tips?
I don't see...
Is it like sucking water in? Like there's sort of like a...
It doesn't look like it, Jody. Yeah, okay, so just for anyone listening, benthic is just anything attached to the seafloor, right? Pelagic, something swimming around in the water column. And of course, we've got Dougal Lindsay, he's part of the collaborators, and he's a plankton specialist looking at this pelagic in-water animal life. And then we have benthic. This is the opposite end of the animals that are living on the seafloor. So most of what we've seen today is benthic attached to the seafloor, and this is a ctenophore, and it has a cousin which can swim around in the water column. So really quite bizarre. So there's a real close view of this animal, like ribbon. Filaments running along the outside sides of it. We're going to have to keep moving. We've still got another dive this afternoon, but thank you very much, guys. This is our team. We've got Brandon and Chris on the controls. So what we might do, we might sign off here, Jody, and have a break, and I need to grab some lunch, and I'm sure you do too. We'll get someone else in the audio seat, and then you and I can join again for the climb up that steep vertical face. How does that sound?
Sounds great. Excellent.
All right, I'll just say goodbye. So this is Rob Beeman signing off, and I'm going to hand over to Jeremy, our black coral expert, and he can talk you through the remainder. So I'll see you later, Jody.
See you soon.
How's it going from your end? That's good. Some more footage. So there's some editing there.
All right, Jeremy here. Taking with Rob while he grabs a bite to eat. And we've spotted a sclerotinia cup coral. So we've collected quite a few on this dive, but they all seem to be look a little different. So it's important to collect, within a group, different looking organisms which could represent different species. And this could be flabellum, maybe. So, would you put it in B? What do you want to keep? Yeah.
We don't have the camera stacks. We can't put it there.
Why can't you grab what it's attached to?
I'll just go and grab it.
Yeah, I imagine it's attached to a rock, which we could probably grab. And next to it is what looks like a pencil urchin.
I can do whatever you need. Alright, push me away so you're going to get further. I don't either, but I didn't do it.
Yeah, this is an interesting looking cup coral.
Porch back in, I guess. Alright, porching in. Give you some more space. There you go.
Just thinking, how can we squeeze in two? I think so. Best one more hour on this to get into it. One more hour. There you go.
All right, so it seems the coral was attached to, I assume, a larger rock, which made it pretty easy to grab.
You want to put it? B. Yep. Porching out.
You got some rubber with it? Just a little bit of the rubber.
This is a good one.
It could be crazy, and we don't want to go in, or just okay. I want to see the players put some surfaces already. Yeah, so yeah, okay, that's
So we're slowly filling up our bio boxes. We're seeing a lot of really amazing things. Earlier we collected 2 separate black corals that they were branched, but sparsely branched. And I've seen species like that described in the literature, but it was described before color images were a thing in scientific journals. So it was hand-drawn, and it was similar. It was a stem, and then 1 or 2 long branches, but otherwise unbranched. And this could be the first time we're seeing this species since it was described in the early 1900s, I'd guess. which, if it turns out to be one of those previously described species, it would be the first time we have tissue of that species. And we would assign this specimen as the new type of that species, because of course the original holotype isn't in a museum. And if it looks different than what was previously described, then it's likely a new species. And that's why we collected 2 of them, because they look similar and they're different than anything else I've ever seen in a museum, and really unlike anything that's been described in the literature. So, pencil urchin, anemone, little sand dwelling critters.
Did you see that? Huh? Did we just hit a
Thermocline?
Yeah, we just did.
Yeah.
Look how fast it tracked.
What was the temperature just minutes ago?
14, now it's at 16.
So we just went through a thermocline where at 320 meters it was 14 degrees, and at 315 meters it's going up to 16 degrees. And you might have noticed it, the thermocline on your screen. It looked like the screen went wavy. So thermocline is when the temperature, a cold body water and a hot body water mix. So that creates a.
It's not an octopus.
It looks like there's a mixing of water, makes it blurry.
Are you on drugs?
Yeah. So we're now at 312 meters deep, and the goal is to go all the way up to the top of the canyon, which would be like 60 meters deep. If we can get to the top, that'd be fantastic. As you might have noticed on leg 1, when we were doing these large bathymetric surveys from 1,000 all the way up to the surface, like in Cairns Seamount, you saw these shifts in the assemblies. So different animals living at different depths. And it's due to many things. It's due to the substrate, the temperature, the salinity. And so we expect, over the next 150 meters to be a very distinct change in the life that we're seeing. We have an anemone here. Just gonna look up a little bit more. Some more anemones. So a whip coral here passing us on the bottom left of the screen. And so now you're starting to see rubble. So must be an area where corals from the shallows are dying and breaking off and tumbling down. This is probably a plateau-like feature that allows things to settle. If we can look closely while we're moving, maybe we can see some things living in the rocks. Got something up ahead.
take a look at it?
Sure, why not? I'm not sure what we're looking at.
Nice and close. Alright. Calm down.
Looks like some sort of mollusk.
Give it a go. No, pretty far.
Yeah. Push core, yeah. Alright, let's do that here, actually, 300 metres.
Alright. Real good. Just good.
You can see a little crab going inside of it. See what happens when he enters. Siphon. Yeah, maybe.
You want one or two? See how the first one looks?
Peanut shell.
Two, potentially. Thank you.
I'm not sure. It's interesting. So while we're sitting down here, we're going to get some push cores. I'm just going to zoom out to give them some view. And we can together come on a consensus on what this is we're looking at.
What? Typical. One or two are already taken, so 3 and 4.
Correct.
Alright. Get some review of this. 40's so sad amongst all the big ones, now, isn't it? Where will it die? Yeah, it was very loose.
Anywhere.
Pretty hard.
Oh, you got it. I just have to break it over there.
You got it.
And yeah, it's okay.
Getting some great push cores. Oh, why did I say it?
Why did you say it?
Man,
It's very rocky.
Yeah.
Extremely rocky. And what I try to do is get not as many rocks at the top.
maybe just behind this shell, this mollusk.
You reach it and you go. Once you have that, I can push back in and get you a little closer if you get it on the back side of that. What do you think?
Good fortune.
I fortunate.
Good there.
Happy with that? Alright.
Okay, do it for me first.
So we're looking for this site without too many rocks.
Yeah, so the question is what is up, like if you want to get the representation of the seabed, then we want that kind of stuff.
Yeah.
Which we can
Yeah, as in a scoop, maybe.
we can do a suction sample of the sediment, or we can do a
Yeah.
So
sediment.
let's continue trying to do this before.
I'll say it there.
I think it's alright.
Yeah, if that stays in, that'd be great.
Yeah. 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 Yeah.
All right, you need to fortune out. Fortune out. Quickly as probably can. There you go. That's all I got, buddy.
All right, we got seconds. It's starting to come out.
It's all right. It's all right. You got it. You got it. I don't see anything moving.
Yeah.
Water level off there.
Going.
No, it's good.
you're right.
It's good.
Nice.
Shove it on in there.
Victory! Thank you.
Forward just a touch. There you go.
It's all in there.
Yeah, that's great. Let's just do it.
It's gonna be so much fun taking that in.
We're doing another one.
One will be fine with that, yeah.
You want to do some
Little suction.
Yeah, suction.
or
Where does that go in the
Into one of the jars.
Go ahead, Rachel.
jar? Yeah,
It cause any issues for me to keep vulture to starboard, so we're heading to 100.
Could you mind coming down a little south if you do that?
Yeah, can do.
All right, that's fine.
Great.
So yeah, I'll give you the waypoint, and then you can,
Okay.
or you can turn the head, whatever.
Thank you.
Yep. I'll bring them down here.
So you don't need the layers, you just want what's on the top.
Fortune in a little bit for you. Right there.
Yeah.
The janky angle on it. Is jar number 2 okay?
Yep.
Alright.
Now we're going to suction some sediment.
Oh, got some movement on it. There you go. Solid. We're on, sir. Like,
It'd be better to go into 8.
Oh,
With
yeah,
more
sure.
flow.
Probably grabbing lots of 4Ms here. So you can see what the sediment's made of here.
I think it's quick. I gotta move a little bit. And not doing anything. Load it. Up, up.
Suction's off.
Suction is off.
Rotate.
Nothing.
Turn around and look back. Yeah, look back at the suction sample. Dead base.
We will not be collecting the mollusk. It's not one of our targets for this expedition, but it is a beautiful mollusk, whatever it might be.
Keep working. Work. 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 Suction on each other.
So now we're just checking to make sure that the sediment went in the proper tube.
Keep it.
No, for this expedition, the biology that we're collecting are sponges and corals. Soft corals, hard corals, black corals.
Are you gonna nudge it?
Also jellies, of course.
Suction off. Suction's off.
Sorry, Dougal, I always forget.
Sit down. Other way. Yes, other way. Angle and touch.
Okay, just got a nice push forward. Thank you, sir.
Are you rotating? I'm not rotating at all. This is straight which way?
I am going to push forward.
Are you going to push forwards? So I need to hold on a second, stand by. Don't do that yet. Good. So getting this way. Alright, because he's forward away from you.
Keep an eye on
Okay, you're clear. Clear.
So here we are now.
So the Halimeda, essentially everything that falls down from the shallows to this depth becomes substrate for other living things to grow on. You'll notice that there aren't that many corals, really for the most of this dive, and yesterday for that matter, where you don't have a lot of suitable of hard substrate. It's hard for most corals to grow. So what you are seeing is when there is a coral growing, there's usually a rock underneath the sediment, and that rock used to be on the surface. So what's happening is there's a rock, a coral larvae is able to attach to the rock and grow, but then it starts to get buried because the sediment keeps raining down. But if the coral is growing faster than the sediment that's building up on the seafloor, it'll survive. And that's why we're seeing these really long corals with real vertical growth.
But we do have some materials.
Yeah, we definitely got some material. But we won't be able to use that much anymore until we get back on deck and figure it out.
All right, so we're gonna continue upwards and onwards. We'll say goodbye to this mollusk.
There's a big fish behind me.
Oh, there's a fish just behind the ROV. Can you inset the video? We're gonna try to put up the video for you guys to see. It's right in front of our light. It's still there. We're just trying to get there. It is. So this guy is right in front of the camera. Anyone able to identify the fish? I think Kai's gone. So the fish is liking the spotlight. All right, the fish has gone away, and so will we. Okay, so we're at 300 meters now. It's 16.6 degrees Celsius, and as we head shallow, the temperature will start increasing at a quicker rate. It'll get pretty warm to the top, probably near 25 degrees Celsius. So I'm just gonna look.
Oh, there he is.
Oh, the fish is enjoying the spotlight.
Is that crunchy?
Maybe he likes the light. Maybe he's using the light as a means to find prey. Big fish.
The other thing is like this line driven light to makes it really drops off.
So one more second, then we'll carry it.
No worries. A yellowtail amberjack,
Good eating.
maybe. All right. So we will continue to look for specimens to collect and keep an eye out for a shift in the assemblages as we continue shallower. What's this? What is this thing? Of course, this coral's on a rock, and it looks like a Gorgonian by the polyps. Yeah, we'll take a. I'll just zoom out so we can get closer. That's interesting.
Where is it? Oh, there it is.
Yeah, I would say it's a Gorgonian.
You gonna want it?
No, it's okay.
No? Oh, what's in front of it?
Yeah, the polyps seem to be protruding, out. So the black coral tends to have polyps. You don't see the entire polyps kind of sticking outwards before the tentacles come out. Yeah, it had the feel of a Gorgonian. Yeah. Oh, well, crustacean? James said it could have been a zoanthid. Yeah, I did a bit of reading yesterday about the difference between zoanthids, anemones. We're seeing a lot of really interesting anthozoans that, you don't commonly see in the shallows. So it's a steep learning curve to learn groups other than what I study. But I'm slowly getting there.
You're doing great here.
Thanks. Yeah, at this stage of my career, I should be able to name the common genera of soft coral at least. I've yet to get that far. Cup coral lying on its side. Looks like the last one we collected.
What's going on over there?
What are we seeing? Shenanigans.
Weirdness.
What's this? Stop for a second here. We're just going to stop here. Something interesting. So, I was able to spot a small colony. We're a bit shallow for. Let's see what it is. We got James online. Maybe he could help. Let's see. If it's a. Sorry, we'll just settle down. Yeah, I'm thinking hydroid as well, James. It has the morphology of a deep sea black coral, but when we look closely, if the branches are, sort of zigzag. Let's see. I can't say for sure one way or the other. The tentacles are hard to see. You know what? I think it's a black coral. And can we turn the lights down a little bit so that there's less? So if it's a black coral, this is really, really shallow to see this type. So it looks like an alternatopathes, which is in the Schizopathidae.
Don't do that again.
It looks like a black coral. The tentacles, the polyps look like black corals.
How's that? Go ahead, Bridge.
Yeah, let's try to focus.
Starboard, does it make sense to be on the other side of the line now?
Yeah, if you're gonna keep, how much do you think you're gonna be turning?
Probably another 10 to 15 degrees.
We'll just yeah, we can set you right now. If you're happy with that. How's that?
Yeah, it's perfect.
Okay.
Cheers.
Grab it and take a close look when you bring it up to us. Yeah. Is that right? Okay. Good question. How big is it?
It's not that huge.
Not too big?
It's been a small quadrant or.
We're expecting a lot more rocks. We are at the shallows. So let's put it in BioBox and B. 2. You know, the 2, yeah. Yeah. Yeah, it's not too big. It's maybe 15 centimeters in height. Yeah, correct, Dougal. It looks like a black coral. It's really hard to see the polyps. They're very thin. It's just hard to get a clear view, and we're a little too far away to get a close-up shot. So what we're going to do is we're going to pick it up and bring it close to the camera, and then we can zoom in. I think it's a black coral, and I don't think the genus that I'm thinking of, I mean, so the polyps are alternating, right? So on either side of the stem, it's one pinnuole, then the other pinnuole, alternating all the way up. So it looks like a Schizopathid. Schizopathids are deep black corals. They're usually found, you know, beyond 1,000 meters.
Do you want that rock too?
Yeah, sure. Looks like a brittle star is in the rock. Yeah, it's extremely odd to see if it's a Schizopathid. It's just really odd to see it so shallow. So I won't say much until we look at it in the lab and confirm what it is.
This is a very, very good.
Yeah, we've collected some really amazing specimens today. We could have 2 undescribed species in our BioBox right now. That's a nice big rock. I think Marty will be happy that we're collecting rock in addition to coral. Try to get a bigger bite. Yeah, let's see. What's it attached to? Yeah. Looks like a rock. Thanks, James.
Obviously, by the replying the whole time.
Just be careful of the coral bending. Zoom in. Nice. Awesome. So we'll just take a close-up view when it's in front of us. Just confirm it's a black coral, and I mean, I'm just surprised that we're seeing this type of black coral so shallow. All right, so we're looking for 6 tentacles. Oh, and there's a little crustacean hanging onto it. We often see them on the black corals. Look at that. So, yeah, it's a black coral. It's got alternating pinnules, simple, non-pinnulate. it can't be in the family Antipathidae. It can't be Maropathidae. It has to be Schizopathidae, and it's just really shallow to see one of these. So we'll see what it is when we sequence it. All right.
All right. Abby?
Yeah.
All right.
So Biobox
Biobox
2, yeah.
Biobox 2. Fortunate.
So, Alex, you worried that we're dropping?
We'll have to pick that one up.
Okay. No, what are you doing?
Oh, portion in. Portion in.
It's on the lid.
Oh, is it on the lid? I didn't know it was on the lid. Sorry, I thought it was on the ground.
Chris? It's on the lid.
Relax. Oh, I see it.
Nice. Sorry, there it is. So it dropped right on top of the bio box. Couldn't wait to get inside.
That's on the ground.
Yeah.
We need to.
I mean, in the records, there are shallow schizopathids at this depth, but often there isn't a coral to go along with the record, so you kind of assume that it's just a bad record, a misidentification. So we'll physically have the coral and, of course, the lat long in depth.
Yeah, happy to do that.
Wednesday, something we're doing.
Anyone play the back Russia?
It's supposed to be a pretty porous rock, not very hard yet.
The hard piece was in my hand.
Right. So it's like, is it like clay?
Feels like clay. Yeah.
Okay.
All right, open it.
The coral fell off. So,
Don't open up that
I won't open up that one. I'll open up 2 only.
and you don't care about this rock.
No, I don't care.
You could toss this rock in the
You don't care about the rock?
back to the sea.
Get that.
All right. In the end, we got the coral, and we can now put it in. Well, really, we could put it in A. There's a bunch of stuff in there that I could do. Yeah, so we can put it in A. I try to get it from the base if possible. It's getting knocked around, but we still have. Well, the crab is still holding on, so that's a good sign. We still have the basal attachment and the whole colony, so that's the important part. And it's difficult to do, but we're trying to grab it from the base as to not lose any of the branches, or in this case, any of the pinnules. Nice. Beautiful.
Can you kill your own anything?
And can you do it associated species? Well, find it in the box.
Open up box 1.
Yeah.
You want to go into B?
1A is fine.
1A?
Yeah, there's a few things in there, but it's okay. They're different.
I'll go just touch one.
That's correct.
So what's the news on this? Yeah, it's good. It's an awesome one. All right, we got it. It was a bit touch and go. Playing with our emotions. But that's science. Awesome. Thanks, Cody.
It's right up.
It's like those little corals he was asking me to get the other day, and I was like,
All right, so we're gonna continue back up.
And where's the J-Ray?
So there was a comment on YouTube about the worms in black corals. So in some genera, like in stylopathes, they have these worm runs. There's worms that modify the skeletal structure and create these tunnels along the stem, and they ruin the spine so we can't identify them. And usually it's one worm that lives, I guess maybe one or a few worms that will live in a colony. And often the worm won't even be there. So I'm not sure if they stay there their whole lives or if they come and go as they please. I'm not sure. But one thing is for certain, you always see these worm runs on this genus of black coral, and you wonder why this genus, So we have another whip coral. We've collected enough black corals for today, I think. We've done pretty well on. But having said that, I'm sure we'll collect one or two more. Especially as we get up into the shallows, we see families that we don't see in the deep, notably Antipathidae and Myropathidae. And of course, we're looking for scleractinian corals that don't occur in the deep. Leptoceres. We're looking for deep Acropora, which is a branching hard coral. And so we're at 270 meters depth, and it's very steep. So at this stage, we're going to start getting up the wall pretty fast. 17 degrees now. Remember, it was 14 degrees just a little while ago. I'll just look up as the slope increases. Got some sort of fish down there.
Oh no. This was remembered turtle.
Wouldn't that be something?
Something.
Huh?
Well, that's your food.
Like something for real, something for real? I don't know.
Yeah, you're calling it that.
So we're starting to see more whip corals and larger rocks scattered along the sand, which has become less muddy, as you can see from the push course.
Just talking about this. We're not going to rush.
Yeah, so we're seeing, so in this dive, we've seen a lot of really interesting corals, potentially some new species that we've collected. We've collected lots of cup corals and black corals. We also collected a soft coral. Yeah, you got to remember, this is the first time we're exploring with an ROV at this depth in this site, and so we're seeing things for the first time. if we're in areas with species that have very small ranges, this is the first time where anyone's seeing these species.
Cody, you make a comment, you make a judgment. Basically, he's saying, I don't know if I'd roll it off that short. You better watch it.
I don't think I'm talking about it. Jeremy, can Marty jump in there and talk about the halometer? You know, like I sit back and stuff.
Yeah. Want to put Marty on for you guys?
Hey, Rock.
Thanks, Jeremy. My name's Marty McNeil. I'm at Queensland University of Technology in Brisbane, and I'm also affiliated with the Geocoastal Research Group at the University of Sydney with Dr. Jodie Webster, who's been on earlier, and will probably join us again a little bit later on. So as we're climbing up this wall here, we can see that the sediment, the loose sediment, is getting quite coarse. And we took a sample earlier on, and what we were actually interested in looking at was, so a lot of that coarse sediment was actually halometer flakes. So halometer is part of the reef system. It'll be growing and living above us once we get up in the shallows later on today. Halometer is a genus of calcareous macroalgae. So it's a green, fleshy macroalgae. It grows in shrubby little tufts. And on the inside, it has got a hard calcareous or limestone skeleton. And halometer is a really prolific sediment producer in reef environments. And so what we're seeing in the sediment in those little white flakes that look like cornflakes, that's the skeletal remains of this halometer macroalgae. So in the Great Barrier Reef, or in the northern section of the Great Barrier Reef where we are now, up on the continental shelf, where it's much, much shallower than where we are now, there are vast meadows of halometer algae growing extensively, and they shed their skeletons over time and build up very thick packages of carbonate sediment. These mounds of halometer sediment can grow up to about 20 metres thick. And what we're seeing on the flanks of this reef slope that we're climbing up now is a lot of that halometer sediment gets shed off the reef and rains down the front of this steep reef slope. So when we see that in the sediment, we get quite excited because it's telling us that there's living halometer growing up in the photic zone, much shallower than where we are now. And we'll be looking out for that as we climb up towards the surface on our dive today. So these halometer bioherms are what I've been working on for my PhD project for the last few years. I first got involved in the project back in 2013, I think it was. I had got the opportunity to head down to the University of Sydney for a summer project, a summer scholarship when I was still an undergraduate student, and work with Jodie Webster on that project, and also Rob Beeman as well, who you've met earlier on the dive today. So that's how I first got involved in the halometer bioherm project in the Great Barrier Reef. And then I went off and did a different project for my honours, and then a couple of years later I was looking for a PhD project, so we came back to these halometer bioherms and been working on that since then. So, and in fact, we just had a new publication published today. So yeah.
That's keeping secret. So, yeah, very coarse rubble scree slope that we're looking at now.
So my co-authors on that paper are Rob, who's standing right beside me now, and Jody, who's online and watching from his office back in Sydney. And my PhD supervisor, Luke Notiff, who's probably tuning in and watching from Brisbane. And my PhD colleague, Nick Dero, who will also be sitting in Brisbane, maybe or maybe not tuning in. He might be working hard today on other thesis priorities. Actually, I think he's just submitted his thesis for examination, so maybe he's relaxing and having a holiday.
It's pretty steep. What do you think about it? Change the geology of the ocean?
So we're at 210 metres depth now. Temperature's coming up to 17 degrees. And this slope is quite steep. We're coming up quite quickly, and you can see all of this reef debris that is tumbling down slope from the reef up above.
I'm going to have to slow down a little bit to let the ship catch up. So, is there anything you want to look at?
Okay.
A rock?
Sure. We need to slow down a little bit here and let this ship catch up with us. So the ROV pilots and the bridge are constantly communicating with each other, and they need to maintain the appropriate position between the ship on the surface and ROV SuBastian down here on the seafloor. So sometimes we go a little bit too fast for the ship, and we need to wait for them to reposition and get into a safe position. So what we're looking at in front of us right here is, it looks like a couple of sort of platy corals that have broken off from up above and have tumbled down. And it looks like this one in front of us is possibly sitting upside down at the moment. A few thousand years from now, when this is all consolidated into a reef rock, and a geologist will come along and drill into that coral and try and figure out which way was up. And when the corals are all tumbled about and upside down, that makes it very difficult. So that's why we're always talking about, is this reef outcrop in situ? Is it the way that it was when it was growing and alive? Not rubble that's tumbled down. Because we're always trying to figure out what we call a younging direction. Which way is up? Which way the younger rocks or younger coral skeletons should be above and on top, and the older stuff should be below. So when things are all tumbled around and jumbled about, that can make it very tricky. When all we pull up is a core that has a diameter of 2 or 3 inches, we don't have a lot to go on. So it's always like a jigsaw puzzle, trying to put together the pieces of the puzzle. So what we're looking at here is not in situ. It's not one of these big nice outcrops that we're hoping to sample from. These are layers and layers of pieces of coral rubble that have come down from the reef above. This is still valuable and interesting information nonetheless. Because oftentimes we're looking at reef rock samples in cores that have been drilled through a fossil reef. And sometimes you need to use a bit of imagination to imagine what we're looking at in a core might have looked like when it was living and growing.
Marty, you might take over. Sure. Yeah, sure. Got us sort of set up for going live with Jody.
Great. Okay, I'm going to sign off for now. I'm going to hand back over to Rob, and we're going to continue on with this dive, and I think Jodie's going to join us from on shore. So I will sign off and enjoy the rest of the dive.
Testing 1, 2, 3, 3, 2, 1. Thank you, Marty. And just checking to see whether people ashore can hear my voice. I'm sitting in the audio seat here. Using different microphone, we're actually filming for a National Geographic documentary, so we're going to reach out to Jodie Webster shortly. Yeah, so just thank you again to Marty. Marty's excellent PhD student. We've been very, very fortunate to work with Marty, and it is absolute thrill to be out here on the water with her, given how difficult it was to actually get this expedition up and running through the COVID crisis and situation, how we get people across countries and across Australia just to get to here on the Northern Great Barrier Reef. And Marty had a very important paper published today, which will go into her thesis. So it's been fantastic to work with Marty over the years. So right now we're at close to the end of our ROV transect, but the end of the excitement for us, because we've got this steep vertical wall that's directly above us, and of course you can see that in the seafloor. The gradient is definitely increasing. We're seeing a lot of these rock clasts. These are parts of the reef that have collapsed and given way. We call it a scree slope. So it would be fairly unstable. Certainly there's this coarse sediment over the top that Marty was just talking about. Lots of this green algae, but of course we're also looking out for marine life. Just made a decision that we're going to stay on this ROV dive for the remainder of the afternoon. I had originally planned to do 2 ROV dives in a day, but we actually started a little later than we expected. It's just better that we don't rush things and we just take our leisurely time up this wall, across the scree slope. I'm bring Jodie Webster back in from the University of Sydney. This particular, also, for Jeremy and the other coral researchers, give them a chance to sample. We now have some time. Or we will go to another part of an area. Right now, we're just going to take our time going up this wall. Yeah, just sorting out that audio here. It's a bit of a spaghetti factory of cables, and just to let you know, back on shore, we're being filmed for a National Geographic documentary to talk about the geology and the biology, and just I guess how unique this is. Okay, so we're really at the base of this 200-metre wall, and we're going to try and get Jodie Webster back on a Zoom call from University of Sydney. And he can join us now. Jody's joined us twice already, and we talked about this wall. We're right at the base of it, and you know, really, it's important to get Jody in because he's a world expert in sort of fossil coral reefs. But you know, the instructions to the ROV divers, pilots, is to, if we do see anything that we want to sample, we will stop and do that. Yeah, good question about, you know, this much debris, and this is the cyclone zone, right? We have some fairly big cyclones come through here. Dean, what was the most recent cyclone through here? Cyclone Trevor, only a few years ago. That's right, it wasn't a big one, probably about a Cat 2, I think, Category 2. We are looking to do sampling here, so this is the base of this large rock wall. So just talking about cyclones, when you live in the tropics, this is all part and parcel of living there. And I live in Cairns, which is about 170 kilometres south of where we are now, and every year we prepare for the cyclone season. But the effects on coral reefs can be quite damaging, and you know, they can really tear up reefs. But funny thing about cyclones, you can find some parts of coral reefs that are, you know, quite badly damaged by the path of a cyclone, and others, you know, really just other sides of a reef can be very intact. But you know, if anyone's ever dived or swum across a reef after a damage, you do see a lot of boulders, a lot of fresh coral turned upside down, and yeah, it can be quite quite a thing to see. But certainly here we are at this really steep wall, and we're looking at a lot of rubble. But actually, if you look beyond the rubble, is what we are really interested in here. This is this basement exposed coral reef. And this is this framework coral reef that Jody was talking about, where it's in situ coral. It's not simply some boulder scree slope that's just tumbled down from above. We're looking for this framework. Now, what that indicates is coral reef that is grown in situ. Now, because the Great Barrier Reef is a very stable platform, that is, it doesn't subside much, and of course it doesn't uplift, then it's quite clear then what you know, what you're looking at is, actually I think we got a nautilus shell over here. Can you see the nautilus? No, don't worry about that. Sorry, everyone say don't worry about that. Okay, well I think we're lining up for a rock sample. Looks like we got a nautilus. It's a very old looking nautilus just lying on the seafloor there. Has a classic coil shape, but you know, it's not just about the geology; it's also the biology here, and we've got ourselves some interesting devices to do this. We've got like a coring device. So the guys here have a really well-equipped ROV workshop, and you know, for doing electrics and hydraulics and working, doing metal work. And we actually have a like a rock drill, and we've tested it out already on basement rock. This is much steeper down this canyon, around about 1,200 meters deep, I think, over a kilometre deep, and we actually drilled into the basement rock. Now, for us, that was really exciting because that's actually a piece of Australia, a bit of continental Australia, and it underfloors this Great Barrier Reef. So what we are now is the steep vertical face, and this is what we believe is the ancient Great Barrier Reef, the oldest part of it, and we're sitting at the bottom. So just simple stratigraphy, layer upon layer. The further down you go, the older that coral gets. Okay, that makes sense. So in the most shallowest parts of the Great Barrier Reef is the most recently growing coral. So here we are now at the base of this big steep slope, and we're trying to find this in situ coral, fossil coral, and that of course would be the oldest part of the reef. So it looks like Jody is going to be joining us shortly, and yep, so we'll have Jody with us pretty soon. Hey mate. Yep. Yep. Hear you loud and clear.
There's these clearly sort of upward projections, which perhaps imply some sort of upward growth. So, are we seeing algae on the surface there? Is that the red colour?
Yeah, that's right. Yeah, of course our screen's just a little bit clearer than yours, but here we are at exactly 200 metres depth, and there's enough light to have what's coralline red algae. So this is algae that we find encrusting over these kind of rocks, and they're using the the absolute limit of light here to photosynthesize. Now these can photosynthesize, but it does show the lower limit of light, and that's this red, almost like paint that you see on the coral rock here. So Jody, we really are at the bottom of this big cliff, and so it should just continue going up. Of course, there's a lot of debris. We've seen the layers, these big coral, like slabs overlying each other, and which we need to get past that. We need to get to this, I guess, more bouldery, exposed looking rock, and that's more likely this. This in situ coral growth that you were talking about before.
Yeah, that's right. So it's a case of, there's lots of debate about this steep cliff and slope. How erosional is it versus accretional? Like how much is cut back and exposed versus how much is the sort of outward growth of the reef? And these are all really big questions and ones we don't really have the data from because we need material. We need samples from exactly this area. At this same depth, at about 200 metres, 189 metres or so in the drill core, this at least, remember, a few kilometres further to the west, the material is within the non-reef part of the section. So it's this sort of what's interpreted to be a shelf sort of setting, very, very different. Often described as a ramp or slope with carbonate growth on it. And so, whether this material is the same or not, that's a big question.
Yeah, but we come back to this question that we keep getting asked all the time, Jody, isn't it? Like, how old is the Great Barrier Reef? And people think we just have this sort of number we can pull out of our heads and say, oh, it's this old. But the fact is, we don't really know. We only have these, these long drill cores that have been collected, decades ago. And here we are now with, like a world-class ROV cameras and an arm that you can actually pluck a piece of coral off that can help us answer this question. It's just this enduring mystery. How old is the Great Barrier Reef? And better than anyone just how hard it is to do this. I guess this is why it's so exciting to be here. And if we kind of translate it back to that drill core you were talking about, it's about the same length, isn't it? So imagine we're looking at this sheer face, like a big rock, like a quarry cliff, and we can start seeing these layers popping through. But I'm thinking now, we at this depth, we must be looking at the first bits of the Great Barrier Reef, the actually oldest parts. If we're talking around 200 metres of thickness, that's a lot of layers built up through time, and being at the bottom, this is the oldest.
Break that up and see what comes with it.
Yeah, if we were to project the stratigraphic layers out in a sort of horizontal fashion, that's right. This would be at the very base of the section, which is less than, about 700,000 years old. But it's a case of what the geometry of the margin actually looks like. How do those layers actually translate to what we're seeing on this outer part of it?
Yeah, so when you talk about composition, you know, I mean, that's a big word, but really the thing is, you're trying to actually talk about the species, genus and species of the actual coral that make up this limestone. And for me, this is quite amazing that you can look at this really old coral, like it's just skeletons, and you can work out what species and genus it is just from the fossil, you know, all that old fossil material. And you can even talk about the assemblages, you know, how they combine together. So I was just thinking about those sequences you talk about. You start with that Porites and Montipora and I think it was a Favid. You know, these are, you know, common coral genus types. But you're actually seeing this as fossil material going back, you know, hundreds of thousands of years. So I think that is amazing.
And so we're able to look under the microscope at the type of coral. It's not just the coral, it's also the algae within the tissues, the coralline algae, which is very important. So that's exciting.
Yeah, that's right. It's actually, yeah, you can see it was quite hard to break off. I mean, hard is good, which means that it's locked, you know, it's not just some rubble that's rolled down the slope. So that's important. We want a fresh face because, you know, to do age dating on rock, you know, you need that fresh face. And this is why geologists have their geology picks, you know, but it's quite amazing. I mean, the technology here, like with this ROV arm, I almost feel like maybe we should just stick a geology pick in the arm and do a couple of whacks on the rock and it will come out. But it's never as simple as that. You know, we're down at 200 meters and but we still have a few more tools up our sleeve, so to speak. We've got this rotating drill piece that we may pull out as part of this. So we've got our first rock sample from the base of this 200 meter cliff. Yeah.
And sponges and all sorts of living things, and they act to veneer and mantle the deposits, and also there's extensive bioerosion that goes on, and infilling and cementation. So there's a lot of things, a lot of challenges working against us to try and get at that fresh material.
Yeah, and speaking of fresh material, we've got we've got our biologists here who are keeping a very close eye on the living biota. When I say biota, I'm talking about all the, you know, living marine life that are attached to the seafloor. And so, you know, this expedition is not only really pioneering as far as the geology that we're trying to discover here, but it's the first time we've really had the ability to sample, you know, selectively sample the living marine life in these depths. And for some people this seems a bit funny, you know, it's the Great Barrier Reef. Surely we've looked everywhere, but the fact is, most of what we know about the Great Barrier Reef is what we scuba dive to, which is around about 20 meters depth, 20 or 30 if you're, you know, pushing it. So that's our limit, you know. Most of the coral reef studies are done within that top 20 meters. And so you just can't scuba dive to these depths, right? It's 200 meters, not safely, not without, you know, technical equipment. So you need this kind of remote technology, and it's been very hard to get that kind of technology out here. You and I, Jody, we've played with autonomous underwater vehicles, but even those ones, I think we had like a 150 meter, or maybe 200 meter limit. You know, it couldn't go. Well, we certainly never took them down to this depth. We were more flying those AUVs down to about 100 metres, but no deeper. You know, if we go back to 2007 when we last did that expedition out here. So, wow, can you see this picture now?
Amazing.
Yeah, okay, so we got some chutes. We got these sort of like in-cutted erosion chutes, and clearly the sediment is being, you know, it's like it's coalescing within those chutes. But we're definitely seeing this outcrop, like this framework coral. It's starting to be exposed. This is not just some scree slope.
Yeah, there's some sort of horizontal layers you can see on the bottom right. Yeah, there's definitely some stratigraphy there.
Oh yeah, love that view. Yeah, so it's pretty clear here. You know, we're probably looking at about 10, 20 plus meters of vision. You know, giving that view. So we're looking down at we're right at the very base of that cliff, and we're starting that kind of slow rise to the surface. So we're just on 196 meters now. It is pretty vertical. Yeah, we can. Yeah, we have. Yeah, we got a situation camera here because our ROV has about 10 cameras on board. You're just looking at the science camera, which is directly in front of us. The situation camera just shows it dropping vertically underneath us. So this is really steep. And then we've got our biologist having a look at our soft corals. Sea whip here, and you know the red we mentioned before is this. It's a coralline red algae. It's coating anything that's exposed to the light. This is the mesophotic zone, the twilight zone. And we've got Scott Nichol next to us. We've got a pointer here, so we're all pointing madly at the screen. It's really cool. So it looks like we're going to drive down to this this sort of more this vertical face here.
Jody, what do you think yourself? What do you think of this place as a potential drill?
Yeah.
Zoom into that.
Yeah, just about to ask you. This one that's coming up in the middle part of the screen. You know, it looks a bit boulder-like to me. There, Scott. Maybe we should move along a bit more. Okay, so it's important that we get something that's in situ that isn't tumbled down from above. So if it's tumbled down from above, you know, it confuses the age story. So we're going to keep moving on up, trucking on up the wall, and we're seeing lots of layers. You know, Jody, you spend a lot of time in your lab looking at drill core samples. It's something else entirely to look at it in a you know, the face of it, the reality of what it's taken from. Here's a nice face, Scott, coming up in front of us. Looks like a nice shell down here. Just looks like some kind of whelk shell, decent size, just sitting in that chute. But for anyone listening, we're looking for a nice, clean, vertical face that we can actually push our drill bit into. We actually want to cut a piece. Yeah, this is quite a nice gastropod, decent size, probably around about 10 centimeters long. And we're going to leave it alone. But we're looking for a really clean vertical face to drill into. And being so steep and so close to the reef, Jody, I was just out on the back deck. It is a beautiful day here, I've got to say. The Ribbon Reef 5 is, it's not even a kilometre away, it's probably about 400 or 500 metres away at the most, and it's low tide, and you can easily see the coral reef. You know, it's exposed, and of course just behind it is the lovely colours and the blues of the lagoon, and you can see the Australian mainland in the background there. So it's a very nice day to be out on the Great Barrier Reef, and even better to be on the outside of the Great Barrier Reef, because it's such a rare thing to be able to do.
We had a couple of nice days on the Southern Surveyor, but that was always a bit challenging to be that close to the reef.
Yeah, and I was just telling the story to the bridge staff a little while ago about how we nearly lost the AUV and all the dramas that caused, because we were, you know, it's the first time we were into this area, and we'd actually done some mapping with the Southern Surveyor, so we had a sense of what the three-dimensional shape of the seafloor was like. But what we hadn't expected was that there were quite a few caves, in that. Yep, we just got ourselves a sample. Yeah, and it's got that red colouring, so we just want to see what the broken face of that is. There we go, and they just bring it into the camera, and yeah, and that's good. We want to see a break rather than some simple rock just plucked off the seafloor. But yeah, just talking about that AUV mission. Yep. Guys, can you just twist it around so we can see?
We're just going to get it over the bottom.
Yep.
So if we do drop it.
Yep. Okay, so it's a delicate operation. Yep. Of course. Yeah, you think it's all very easy, you know, but you know, this is it's you know, it's a 1, 2, 3, 4, you know, it's a 4 or 5 degrees of movement here. Okay, there's our close up. Can you see that, Jody? Looks a bit Halimeda-like to me. Sponge rock. There's Marty's expert classification. Sponge rock. Scungy. Yeah. No, you got to have fun out here, and it is fun. You know, we're out in the Falkor. I don't think anyone else is. There's not many other research ships out in the world doing their thing at the moment. I mean, this is one of the few research ships in the world. Anyway, we're very lucky. We're having it here, working in our backyard. But yeah, just talking about the AUV mission back in 2007, we put the AUV down, and we were driving it up slope, and it drove itself into a cave, and it was quite safe. It was just saying, you know, all systems go, but I can't move. I'm in a cave, and you know, it's a pretty expensive piece of kit. And I remember Stefan, who owns the AUV, you know, I think we even went so far as to try and commission some commercial divers to come out and join us to swim down and free it from its prison. But eventually, you know, being a good robot, it figured it out. You know, it kind of wiggled its way out and popped to the surface, and there we were at night time, just off Ribbon Reef 5, and exactly where we are now. And it was, you know, the wind had actually come up by then. It was night time, and I just remember the very impressive piece of seamanship to recover that AUV, which was bobbing around in the waves, you know, and they got a loop on it, and we recovered it on board. And in fact, it was the only mishap for the entire expedition. It happened very early on in the piece, but from then on it just worked like a dream, and we got AUV transects along the entire Great Barrier Reef margin over about 1,000 kilometre distance. And you know, even to this day, we're still using the images that we collected from that AUV mission. So And here we are out in the Falkor, and they've just finished leg 1 with exactly the same AUV. So the Sirius came out again, and it's still going strong. We're not using it on this leg, but on leg 1, which everyone left about 3 days ago in Cairns, they used it extensively out in the Coral Sea. So, yep, the Sirius is still going strong.
Actually, and I remember it was all a bit stressful. I went to bed because I had been up for like over 24 hours, I think, and it was a bit unsure as to what was going to happen. And then when I woke up, everyone was very happy. It all sorted itself out. It was fantastic. And then we continued on, as you said.
Yeah, Jody, you're getting this picture now. There's definitely some, kind of more exposed faces here. Although, right just off the right-hand side, it looks a little bit like a scree slope, and then it kind of grades into this more exposed area where it doesn't look scree slope-like to me. So, are you seeing that too, Scott? To see how I think we're in a debris field here. So, from a sampling perspective, yeah, that's right. This is probably the better zone to be in on the right-hand side. Yeah, and you can definitely see the kind of the layering. Yep. Yeah, we're at 190 metres. The shelf break, the edge of the continental shelf, is at about 90 metres. So we've still got another 100 metres vertical to go on here. What's interesting now, I'm not seeing a lot of marine life, and I'm wondering if, like living marine life here, and I'm wondering if it's just the rain of sediment that's coming down from the shallow reef above us is suppressing the marine life that might live here. Now, we have seen this in other parts of, like the Coral Sea reefs, Osprey Reef, these steep areas where you get just so much sediment coming down. It's like a blanket. It suppresses the sorts of animals that might want to attach to rock, like corals. So you just wouldn't find living corals down here because there's so much sediment. But we have seen a few sea whips. Certainly the red colour is this algae, this coralline algae. It's grows like a crust, almost like a cement on the rock. There's plenty of that, but I'm not seeing a lot of fish, not seeing a lot of echinoderms. A bit further down the slope where the gradient wasn't so steep, there was a lot of that marine life, but this is somewhat different. But this also goes, to talking about the habitats, and so we have, habitat is the environment in which animals live, and part of this expedition is trying to describe those habitats. So I would describe this as a sediment draped fossil reef habitat with perhaps suppressed fauna. So fauna being, marine animal life. But we have, a team of biologists on board here they're looking for corals, sponges, and things like that. So we're certainly keeping our eyes peeled for anything that's worth sampling. So Scott's directing the pilots, Adam and Ben, to have a go. It looks like we're going to snap this piece off here. This looks like a little bit of rock that's sort of jutting out. And we'll see if we can snap it off. Get ourselves a fresh rock. You can see it's about 10 centimetres long. There's the laser beams at the bottom of the screen. There we go. Good grip. So for anyone listening, the ROV itself, SuBastian, is about 4 tonne. There we go, we got a piece. So it's the size of a large car, but in the water it's neutrally buoyant. And so this is provides challenges for when you're trying to lever things off rocks, or lever rocks, rock samples, because as you twist the arm, you're also exerting force against the ROV as well. It's not like it's locked onto the seafloor and you can simply, lever it off. So, it's all about those forces, the opposite forces exerted on the ROV. Oh, isn't that a great look? And we got a little cup coral too.
Algae that has built this deposit, and it's probably fossil in the sense that the inner parts of the crust are probably quite a bit older, and we've got obviously living stuff on the outside. We've seen this mantling some of the drowned reefs before, and that could be the fossil deep water deposits covering. But when we cut that open, there may be some coral inside. We'll have to see.
Yeah, okay.
But clearly there's algal structure.
So really we're the first people to see this here. I mean, this is a really unique expedition in that we can bring vision down to these depths and sort of make sense of the older part of the Great Barrier Reef. And here we are looking at, other people have seen like your cores, for instance. So lots of people have shared those cores around. But here we are seeing a piece of deep water coral, fossil coral, for the first time in front of this part of the Great Barrier Reef.
Like, if you can picture, like, the sea level is where it is now. We're 186 metres or so below present sea level, but during the last ice age, it was about 120 metres lower, so it was about 40 odd metres. And so that's actually a pretty good depth for certain types of algae to grow. And there's lots of debate about, how much of the last glacial maximum reef was growing on such a steep slope. Now, we have observed and drilled this further to the south on the International Ocean Discovery Program drilling campaign, but that was on a much gentler margin. So it's unclear whether you would get such significant low stand reef growth on such a steep margin. And we have to wait till we get up to 70 to 50 metres as the slope comes over, and that'll be interesting to see.
Yeah, we hope to get to that point. Of course, we're really quite close to the reef at the moment, like the shallow coral reef. Like you can stand on the back deck and actually look just over. It's not far away. So yeah, I mean that gradient is important, as we found, because you and I have been looking along the entire length of the Great Barrier Reef from, the northern part all the way south, which is well over, nearly 2,000 kilometres further south of where we are, trying to understand the variation across that shelf edge and where these drowned reefs grow. So I'm talking about those drowned reefs in that sort of 50 to 70 kind of metre depth zone. And again, it wasn't until 2007 do we have a real sense of where, I guess the spatial, like the distance and how far those things grew. But as it's turned out, those drowned reefs are, they run the entire length of the Great Barrier Reef, and I call them the proto-Great Barrier Reef. What was there before the modern reefs are, that we're familiar with and can scuba dive on and the like. But yeah, I mean, it's this sort of endless quest for trying to understand these fossil reefs has been really fascinating. And they're fossil in the sense they've stopped accreting, or they're much older, but there's always a veneer of living marine life on them, so they're not dead in that sense. There's quite a lot to see, and and some of these other fossil reefs we've looked at, where we've put cameras down, I'm talking about, say, 50 to 70 metres, they're covered in soft corals, living soft corals and sea whips, and I think of that work we did back in 2017, Jody, on the German RV Sonne. we had that one of the world's biggest research ships for a month. I mean, that was amazing. We put that drop camera down to, it was around 70, 80 meters, and the color that came back, all the living marine life that's growing, living on these drowned reefs was really, really amazing, wasn't it?
Fusion of life, so much, deep water corals, the sort of things that Tom Bridge, our colleague, really loves.
Yeah, I don't know if you saw that inset there briefly. It was just a camera view to over the stern, just to show you, well, the weather is just perfect. There we are again. Yeah, thanks. Who's on there? There's John. Thanks, John. So we got cameras all around the ship, and you can see how flat it is. It's 2:30 in the afternoon here, but that line in the background, that's Ribbon Reef 5, and there is a lovely view. Ribbon Reef 5, and you can actually see the Australian mainland in the background, so it's a really clear day, beautiful blue sky, and those are the mountains that run up from Cairns to Cooktown. And then you've got Ribbon Reef 5 in the foreground. So that's what we're slowly working our way up in this vertical wall. And again, a lot of sediment, a lot of rubble. This is like another rubble zone, so we have to avoid looking at that. And John, I wonder if you could bring in the 3D view. Yeah, there we go. Okay, thank you. So this is our three-dimensional mapping view, just to show you where we are, and we're really climbing up this steeper part of the rock face. So this is artificially coloured, of course. We've just coloured it red for relatively shallow, and blue and violet colours for deeper. But this is the mapping that the Falkor has been doing in the last 3 days, and you can see we're not quite halfway up that slope. But if you have a look in the background, this is the shallow Ribbon Reef 5. It's about 6 kilometres long, north to south, and about 1 kilometre wide, east to west. So that is the reef that you just saw in that inset, and we're now at 180 meters depth. Oh, talking about cascading, you can see the like little avalanches here, and I've talked about these reefs are big carbonate factories. Carbonate being calcium carbonate, and this is what limestone's made of. That's its chemical composition. But these reefs are just big factories. They're just growing and dying and just constantly shedding all of this material. And oh wow, this is interesting, Jody. Okay, we're getting a lot of layering going on here, lots of overhangs, big overhang here. This is interesting, and you can actually, just on the right hand side, did you see it's actually curving over, like you're getting like a bit of a mound. The strata is actually bending over. Yeah. Our first real caves. Yeah, and if you notice, there's a lot more marine life growing on it too, and I think that's probably because there's so much sediment raining down from above. When you're fully exposed to the sand, you're not going to grow too much, but as soon as you get a bit of an overhang, a cave, you can attach underneath. So if you look through, I can see all kinds of things. There's sea pens, and if we've got Jeremy here, we'll be probably looking for corals. Definitely a big change here. So we're 170 meters. Temperature is definitely warming up. It's 20.5 degrees C. That's the temperature at the ROV depth. So you could certainly be comfortable swimming here. sea surface temperature is probably around about 25 degrees C. So we're only just gone past winter in the Great Barrier Reef, and it's still very, very comfortable. You saw outside this beautiful blue skies, this classic tropical winter weather, and boy, are we lucky to be here now, because in fact this time of year can be quite blowy. We have these southeasterly winds which really blow consistently through the year, and if it was blowing like it has been in the last few months, we would not be here. So everything has to line up for us to be where we are now, and we're super lucky.
This depth in the Ribbon Reef 5, this 170, 160 metres or so, I think I was saying, was this really interesting non-reef section of the core. So there's coral, but not the big barrier building types of forms. And so what is dominated in this part of the section are these coralline algal nodules, or rhodoliths, that's what they're called.
Yeah,
so
okay.
Concentric rings, crustose algae, that we see very commonly at this depth in the core. So it'll be interesting to see if we can break off a piece, whether we see similar sources of mythology or not. But certainly the outcrop is different to what we saw below us.
Yeah, definitely. There's a lot of cavities in there too. Yeah, you know. So for anyone listening, if we talk when we use the word morphology, we're just talking about the shape. Okay, it's just a fancy term for shape. So the shape and texture of the seafloor change. Something's quite different here. There's overhangs above us, and we have this rock in front of us with lots of cavities and things. Now we're going to try and actually break a piece off. And Jody was just saying, at the equivalent depth, about this 170 meters depth in the core that he's studied, which, excuse me, that the equivalent rock type is this coralline algae, this rhodoliths, you know. So this is a particular kind of rock that we can recognize just from fossils. We just broke a piece off there. The guys are just talking about whether I think we're going to try and get a bigger piece.
Yeah.
I'm going to try again.
This one I can pick up another. Yeah,
I don't know which one they're aiming for. A bit of a target-rich environment, because you ideally want the biggest one possible, but biggest might not necessarily mean you can break it off. So we have to, you know, sort of choose carefully. Yeah, I mentioned before that the ROV, even though it's 4 ton, it's neutrally buoyant, right? This thing can bob around in the water at these depths, so you don't have that kind of leverage force that you can, because you know, like you do, your feet are planted on the ground through gravity, and you can reach out and pick something up. But you imagine if you're completely neutrally buoyant, as you lift something up, the opposite forces push you down, right? The main body. So it's always it's a real juggle between what you can pick up, you know, the size of things. It's still quite amazing though. No, I think we broke that off. Is that right, Marty? Yes, it was broken off. Yeah, in fact you can see the sharp face of it there. Yeah. Yep, thank you very much, gents. And it's Ben on the controls. You think of all the remoteness, that's the remote technology. I mean, we are remote. We're on a ship that's 200 meters above this, and Ben is using it. It's kind of an unusual joystick. It looks a bit like a pistol grip, and you know, he's just holding it quite gently, but he can actually do a lot with that handle. next to him is Adam, and he's keeping the. In fact, what they've done is they've sat the ROV down on the seafloor just to give a little bit of purchase here. Purchase being you can actually do a bit of levering. So that's one way of dealing with trying to pick something up, by just putting the ROV on the seafloor and letting it rest. Yeah, so the team here are just discussing where to put it. Our boxes, our bio-geo boxes are getting quite full here now, and you can see we've got a big team behind us, and they need to record exactly where they go, and we have people keeping records of that. So when the ROV is finally recovered and brought back on board, we can all just go in there. We know exactly what sample was collected when, which, and because all of these boxes are numbered. And we have quite a few things now. Lots of rocks. We have some corals for Jeremy, some soft corals for other collaborators. What else have we got? We got some sediment cores. Halimeda for Marty, is always interested in Halimeda. And of course we've got still 190 meters to go up, and we're going to spend the rest of the afternoon looking. We're heading into the mesophotic zone, this twilight zone, where the light is going to be increasing, and as we go up, we hope to see more and more soft corals, and eventually hard corals. Hard corals, photosynthetic zooxanthellate. Shallow water corals can only exist in those photic zone, but that's the kind of zone we're heading towards at the end of this dive. So we'll see where we go.
We're looking for the deepest photosynthetic coral ever collected in the Coral Sea GBR.
So from Jeremy, we're looking for the deepest ever collected photosynthesizing coral in the GBR. Now, this has been long-term work by a colleague, Tom Bridge, from the Museum of Tropical Queensland. He's always been interested in depth ranges for various corals, and he is a mesophotic coral expert, mesophotic being twilight zone coral expert, and he works at the Museum of Tropical Queensland. Unfortunately, Tom couldn't be on this voyage, but he is remotely. So we have lots and lots of collaborators, lots of principal investigators from all over Australia and the world, including Dougal Lindsay from JAMSTEC in Japan, your colleague Yusuke Yokoyama also from Japan, who would be Yusuke is a coral age dating expert, so we do need to bring in all these different experts to try and help us make sense of the material that we're bringing back. But Tom's particularly interested in these. There you go.
Yeah, Juan Carlos Braga, our specialist on coralline algae, is very interested in both the modern algae that we find, really understanding the depth distribution of these things, and also what it can tell us about the environments in the fossil outcrops that we find. So he's very interested to get his hands on some of these samples as well.
Yeah, so heading back into the wall now, Jody. I mean, just real change in shape, really layered here. But it's quite amazing to see. Oh, and some nice life here. These would be feather stars poking out. Oh, there's a beautiful soft coral colony just hanging down. This is quite a feature in the Coral Sea. These really big fan corals. And a lovely feather star hanging out underneath. Jeremy, what about this long feathery one? Is that a black coral?
Let's take a look.
Okay, we're going to zoom in here, Jody. We do have plenty of time now. It looks like there's a sea pen behind it, but
no, it's not a black coral.
What?
It's not a black.
It's not a black coral.
So it's soft coral. What I'm interested in actually is we haven't collected any sea fans yet.
Okay, so one of these big ones, these big gorgonians. And here we are. This is our first real deep one. No.
What the pygmy seahorse might live?
It might do. Maybe. Gents, could we just have a closer look at this this colony?
Yeah. This could be interesting.
So we're just having a look at the marine life here, Jody, attached to this this fossil coral. Depth is 170 meters. Temperature is 20.5 degrees C. Not so much sand on this lot.
You think you can grab while hovering? I can probably set up for a sample.
We have permits to collect this this kind of material, so
We don't need the whole
if Jeremy thinks it's worthwhile, we do have the time to do this. So we should do, you know, take a sample if that's important.
Yeah, we're seeing soft corals here as well.
Soft corals over on the right. Yeah, this is like our first very obvious soft corals as we're going up this wall. I mean, look at the rock here, Joe. I mean, it's really quite different to what we were looking at further down below. Very lumpy. Yeah, lots of caves, lots of caves. Yeah, so going back to the platypus, you know, I can't imagine these guys actually built their own submarine. I can imagine trying to do that at university now, and the paperwork you'd have to do for that. Oh my God! But these guys, they built it. You know, it was a different era, and off they went, charging over the edge of the Great Barrier Reef. They never took a camera with them. I don't recall any sort of imagery, but the description...
And it's really hard to see what's going on. And of course, they weren't able to sample. And if you go and read the earlier reports, they were ramming the submarine against the outcrops to try and break things up.
Pretty confident about their build.
Yeah,
literally ramming.
Yeah, I think massaging is the term. But yeah,
it's crazy.
That would have been. It was another time, I guess. But,
oh, absolutely, yeah. But yeah, you're saying the other day they nearly got bowled over by it was a bowler that actually came tumbling down on them. Is that right?
Yeah, my old advisor and colleague Professor Peter Davies, tells this wonderful story where they were, you know, down at depth, and suddenly, you know, the submarine starts rotating in a crazy fashion. It was apparently a block had fallen off and then just gone down and
Yeah.
But even we won't encounter any of that today.
No. I mean, yeah, we got the ROV. I mean, it's just way too much paperwork involved with submersibles, that's for sure. But you know, despite not having great photos or imagery from that time, the description was actually really good. You know, you can't fault that. And, you know, you can pull out these old classic texts of the Great Barrier Reef, and you can see the, you know, this profile that was drawn by probably David Hopley, you know, classic... a geomorphologist is the correct term. Like, he was a marine geologist, but you know, he's written great books on the Great Barrier Reef. You know, the older Great Barrier Reef, and the profile that he drew from that submersible dive in 1980s is I still think it's quite accurate. So they got about as far as 200 meters, and they started describing this layering that we're seeing. These overhangs, the caves, the wall. They call it the wall, and clearly, this is exactly what we're looking at here. Yeah. Yeah, yes, 4K vision. And, if anyone listening is sure, it's not just about the vision. We have a team of people here who are taking, doing event logging, so anything of significance, they basically take a still photo from the 4K vision. It's then logged onto the computer, and you can type text, depth and all that sort of thing, so you can actually append a whole lot of information to a particular screenshot. So we'll always be able to, we can then look at this post-voyage after the dive. We can jump straight to that animal of interest. And so we've got Stephanie doing that with us today, giving a wave. And we have Alex over in the squirrel seat. Now, squirrel, funny name, but every 5 seconds a still photo is taken from this 4K vision, and this is more about describing the habitat, the overall environment, how it changes all the way up along the ROV transect. So we've done that from the very, very beginning, 1 kilometre further down slope from where we are, and every 5 seconds a photo is taken. And what happens then is Alex can take that, she's got that still image in front of her, and then she can click on, let's say, a critter, and she can say that sea cucumber, write some text, or a particular kind of fish, and so she can actually annotate the actual photo. And when you're doing that so regularly and on images that have been taken every 5 seconds over a long transect like what we have today, well over 1 kilometre long, you get a really good sense of the change in habitat. So further down the canyon where we started this morning, it was there's soft mud, it was this sort of white sand habitat, lots of mounds and burrows and tracks of animals that are feeding on the seafloor. And here we are in a different habitat, rocky, more vertical cliffs, more in the mesophotic zone, and so we can describe the changes in the habitat, and importantly, where those depth zone changes. And so we're getting all kinds here. We're getting the deep geology story, we're getting the habitat, the modern habitat story, and with the benefit of all these samples, we can actually put names to all the animals that we're collecting. So, it's really, really exciting, this voyage. We're covering all bases here. And, we're also doing plankton, and we're doing conductivity, temperature, depth profile. So we're doing the physical water column values as well, like the temperature and the salinity. So we're trying to understand everything about this. It's all about trying to understand the Great Barrier Reef ecosystem from the shallow to the deep, and and how things move between the water column and also animals between the shallow and the deep, because it's all connected here. So just having a discussion what that was, Jeremy?
So apparently on there are zoanthids.
Okay, so possible zoanthids on stalk here. So we're taking our time to collect our coral specimens, and I don't think it's any surprise. As we get higher up in the water column here, closer to the surface, we're going to see an increase in the types of marine life that are attached to these rocks. Okay, onwards and upwards. We're at 170 metres. Oh, a fish over there on the right. but yeah, really quite a different looking rock type. Far more cavities and overhangs. So for me, I'm really interested in around that 120 metre mark, Jody, which is where the last ice age occurred. You know, that when sea levels dropped about 120 metres. I mean, this is something we've been looking for a lot, haven't we? Everywhere we go out in the Coral Sea, we're trying to find, you know, like a wave-cut platform where as sea levels drop to their lowest point during the last ice age about 20,000 years ago, you might expect to see ledges or caves. And so this is one place where we really, really hope to see that.
The last glacial maximum, so 20,000 years ago, this whole area or zone, which had been previously described in the previous profiles as the zone of caves, is also interesting as potential records of the previous glacial maximum, right?
The ice age before this one, yeah.
And
yeah.
margin over the 100,000 year period between each cycle, so they could be preserved at these deeper depths.
Wow. Wow, we just come out of this big cave at the moment. Oh, this is really amazing. Like, there's a big cave. You could just slot right in that one. Oh, and there's even light coming in from the back. This is hollow, Jody. You can actually see light coming in from behind. So this is like a big, like a, I don't know, like a big buffer that's pushed out, and it's so eroded, it's now open from underneath. So we're at 160 meters, and of course there's lots more marine life now. We're seeing a lot of soft corals hanging out underneath. This is lovely. You know, I'm a scuba diver, and I scuba dive on the Great Barrier Reef. You know, certainly shallower waters than this, but I love looking at the swim throughs. You know, just, you know, I just get into those caves and almost imagine the water had dropped and you could stand up in it. You know, of course, this is where Aboriginals could have once stood. You know, during the last ice age. You know, 20,000 years ago, there were humans, Aboriginals, indigenous people in Australia during this period. You know, from the last ice age, they could have been, you know, walking around on this, you know, amongst these limestone caves. Not at this depth though. This would always have been underwater. So I'm certainly talking about a little bit higher up in the water column. But it's those caves that really excite me. And, you know, as a diver, I love getting in them, and, you know, I don't do anything stupid like get into, you know, something too tight. But you know, we call them swim throughs. You know, particularly ones that are open at the other end, and you just swim through them. And yeah, I love doing that. But yeah, this is really spectacular. I'm looking at this situation cam too. It's a little bit a wider screen, and boy, are we getting this. It's like the big prow of a ship, you know, just kind of looming out of the gloom. Really, really amazing. Yeah, it is. It's looking a little bit higher up from the science camera, which is looking directly ahead. The quality isn't as good. It's not as sharp, but we're getting a wider view. And yeah, we're definitely seeing more soft corals coming in view. Yeah, okay. This is interesting. So you'll notice that there's hardly any sediment here. You know, there's definitely still some, but it's nothing like it was further down below. I'm just looking at these big caves. Wow. And these beautiful soft corals hanging down. And, you know, like the other ROV dives that we've done out in the Coral Sea, this gets very, very busy, you know, with fish and soft corals. And yeah, this is this dive is really exceeding my expectations, because all we really had was David Hopley's hand-drawn profile down this wall. Of course, it doesn't. It was a great profile, and that was, you know, kind of a first look at what might be here. But to actually see it in this beautiful vision is something else, you know. So lucky. Big sea whip in front of us. It's a coiled sea whip, well, a couple of meters long for sure. Yeah, it's getting quite busy now as far as mesophotic life, you know, the twilight zone life. I'm seeing green. Did you guys see this green? It could be green algae. Okay, so far we've seen a lot of red algae. Now it's green, very green colour. We're still out a little bit, hard to make out exactly what it is. So we're just going to have a bit of a zoom in. Ah, is this Calopa? I think this might be Marty. It's in, okay, well, there's a bit of a debate here. Some kind of, it could be encrusting green algae. This is the first one we've seen. 158 meters, pretty deep. You know, there's all different kinds of algae. Most of the red that we're seeing is an encrusting red algae, but this is the first real green algae that we've seen. And you know, we might have a bit of a look, see what's there. So guys, just we got J-Rod and Ben, the pilots on the controls here, and putting it down is not easy. It's like trying to sit on the side of a building, eh? Yeah, parking your car on the side of a building is not going to hang there real easy. So we're just going to kind of slowly inch our way in, because it's nice to get these real close-up views. We've got Marty, who's our algae expert, and we're going to be looking at her and Jeremy's on the zoom control, and he's having a look at this coral. What do you think?
It's a black coral.
Black coral?
Looks like in the family Meropathidae.
Meropathidae. So I would say we're probably going to sample it?
Yeah.
Yeah, absolutely. Yeah, we do have the time to sample, and that's what we should do. But for anyone watching it ashore, you know, just really look at all the detail that's starting to pop into view. So much. So, and it happened quickly too. You know, we went through.
Okay, both of these. So, two different families sitting right next to each other.
Yeah, Jeremy's saying two different families right next to each other. Definitely going to have a go. Guys, can we also get a rock sample while we're here as well? That would be great. And because the real challenge is you don't want to just pick up any old loose rock, because it could have just been some shallow modern marine rock that's tumbled down from above. So it's always about trying to break off a piece.
Should we get that rock that's attached to that coral in the back?
Yeah, that might work. Yeah, so we may be able to pick up this, the base of this black coral, which, you know, is a rock, and we might be able to pick that up at the same time. But yeah, if you look at this green, almost this encrusting green algae, I don't know what it is. I thought it might be Calopa, which is a bubble coral. But no, it's not Calopa at all. But maybe people listening ashore, any tropical algae experts, can help us with that one.
I think we have a red coral. You know, rumblum, precious coral. Red coral, you know.
Yeah, a bit shallow for that here. Yeah, Jeremy's saying, could it be precious coral? No, I don't think so. We do find precious coral in the Coral Sea, but this is a little shallow for it. We've typically found it down around sort of 600, 700 meters depth.
Like right there? Is that right? Or maybe it's a Garbonian. Oh, it's a Garbonian. It's moving.
Yeah. So bends on the arm controls.
Well, I guess at Biobox 2. 2, yeah. Our wet lab's going to be full today. Man, this is great.
And it's got a bit of a hitchhiker, which is just a feature of all marine life here, that the animals living on animals. We've got this we've got a brittle star just on the top attached, and we're just discussing where it might go. We've already got our bio boxes are getting quite full, and we've still got a ways to go yet. I've got 160 metres yet to rise.
Right, you want to make 130 for a rock sample?
Jody from the control room, would you like a rock sample here, or would you prefer to wait until you're up around that 130 metre mark? Yeah, we'll definitely aim for a 130 metre rock sample. Actually, we're good for time. We were going to do 2 dives today, but that was just not really practical. It's just better that we don't rush it and take our time working up the this really spectacular reef rock wall in front of Ribbon Reef 5. Yeah, we'll take our time. So we can look and sample. And tomorrow we're going to do another dive and different canyon system a bit further south as part of our comparison between different canyon types. So yeah, so we've got plenty of time here, Jody, and the weather is very, very good. It's flat. Everything's safe here. The ROV's going well. The team's going well. A well-oiled machine here.
While we're sitting and if they are able to snaffle a rock reasonably well, then why not take one?
All right, we're going to see with this one, because it's attached to a rock, we might be able to grab the whole rock and just lift it up. We'll see how it goes.
So, see? The rock's not too big. You can just grab the whole rock. That's what he's trying to do. Oh, okay. So this is a stylopathed. Just have to rotate. And Gorgonian as well.
Yeah, I think we're going to take a little bit of time here because there's lots of things to target. There's looks like a Gorgonian behind it as well. So Jeremy's saying that we're going to grab a few things here, Jody. So I tell you what, you're welcome to take a little bit of a break. We're stretching our legs here just as the biology is being collected. I'm sticking around for the ride.
Yeah, we'll take a quick break here, and I'll dip back in 5-10 minutes.
Yeah, that's right. Just keep an eye on the depth range on the screen. That's the most important thing. We're now at 158 metres. So 120 will be a really interesting bit, is what we're looking for, those caves. So yeah, that's still a little bit above us. So see you then. See you shortly.
Yeah, we'll do. And again, that in the core, as far as we can put the inception of the main reef building, is probably about 137 metres. So really, we want to see if we can see any kind of difference in the outcrops from about that point up to that 120 metres and beyond.
Okay, mate, I'll see you in a little bit. I'm just going to swap desks here, and Jeremy's going to take over the audio for a bit. So this is Rob Beeman having a break, and Jody and I will get back together in a little bit. So handing over to Jeremy.
All right. So what we just collected was a stalopathid next to a myropathid. So this is the depth at which we'd start seeing the shallowest two black coral families, Myropathidae and Antipathidae. And as you can see, we're starting to see lots of soft corals. We could see lots of gorgonians right here. Lots of whip corals of both the soft coral and black coral variety. We're going to keep heading north, shallow. And one thing we're trying to keep an eye out for is what will become the deepest ever collected zooxanthellae hard coral in the Great Barrier Reef. So it's been collected, I think, somewhere around 80-ish meters. And but on the first leg, we were able to collect it down to 130 meters. And so what that means is sunlight is penetrating enough for corals to absorb that sunlight all the way down at these depths, which is pretty amazing. Because if we turned our lights off right now, we would see almost nothing, but there's a little bit of light coming through. Is that an experiment we can do, or no? We want to see. All right, we're going to turn the lights off and show you what it looks like if you were a fish living at this depth. So, I mean, you could see light. You know, we can see the corals. So there's sunlight penetrating down to 150 meters. Beautiful. So you can see the rugose rock mountains going up to the shallows there, and you can see the sunlight at the surface. And so this is going to be where we start seeing many different types of corals that we weren't seeing down at 180 plus meters. Well, we could, you could see really fine detail without lights.
Good?
Yep. Aha, we could see again. All right. So you could see coralline encrusting algae, which is another indication of sunlight penetrating these depths. Lots of soft corals and black corals. So there's a question on the Facebook chat about how long it takes from a collection to be placed in a museum. Luckily, so sometimes it will take a really long time from when it first gets collected by the collector and then sent over to a museum. But I am affiliated with the Museum of Tropical Queensland, so right after this trip ends, I'm going to drive from Brisbane up to Townsville. It's like a 3-day drive with the corals in the back seat, and I'm going to deposit them that next day. So
Don't leave your car in the sun.
yeah, so really quick, and it's important to get them to the museum quickly because although we have ethanol in the boat and we're preserving the specimens, they're going to be in plastic bags that they can leak. So what we'll do is we'll put each coral in a glass jar filled with ethanol, and then it'll be preserved for the long run. So you're seeing lots of Gorgonian sea fans. So we're at 150 meters, which is where we found the Zooxanthellae hard coral on leg 1. So we're not breaking any records today with that, but still worth collecting. So, ooh, looks like a interesting looking soft coral. Can we take a close look at this furry coral?
Just don't pan.
Don't pan. It's beautiful. Yeah, so it's a bottle brush soft coral. I think it's worth collecting. I don't think we've seen anything like this. Yeah. Yeah. You can take it at the top half.
You said just the top half?
Yeah. So we're just going to situate ourselves. Where does it go? I don't know, you tell me. Is it?
If it goes in 3-4, it won't be confusing.
Yeah. Are we okay opening 3 or 4? One of the, either 3 or 4.
Yeah, should be fine.
Okay.
How do you want to just open it? Or self-fold?
Yeah, exactly.
Self-fold.
So I got a manual. Oh, there you go. Oh, beautiful. So, yep, I think I'm counting 8 tentacles. It's really fine branched. So each branch is sub-branched. Beautiful.
Okay, now you're good to keep going around.
We're good.
Good with images?
Yeah, that was awesome.
You just want the top, right?
Yeah, please. Yeah, I mean, this coral just stands out so much. How do you not get a sample?
About that much or a little bit lower?
little lower. Yeah,
And where'd you say you want this one to go?
3 or 4.
Is this enough?
Yep, that's good.
Oh, sorry.
Yep, it's quite a spectacular looking coral. Looks like fireworks. You can see how fragile it is. You can see little pieces of polyps coming off when it's moved. But got a great sample. Yeah, you could see that the branches go around the stem, and some of the branches look some sub-branched, so they're branches on branches. But not over, there aren't many orders of branches, maybe at most 2 branch orders. So it's fairly simple, but many rows of branches.
All right, number 3 or 4, which one do you want? 3. Is it like it? Yes.
Maybe a chrysogorgid. Thank you, Kathy. You took out the best part of the coral.
Serious?
No. Okay. Very good. So,
A little piece of it.
all right, we're gonna continue upwards. So there's a lot of interest in what's at the top of this canyon. We're getting there. It's an amazing opportunity to not be rushed and just to sample biology from the bottom to the top, which is what we've done over the last few days. Yeah, I don't think I'll get many opportunities in my lifetime to slowly climb this type of seascape. And you can see the diversity is changing, the assemblages are changing, and we're sampling it all along the way, and we're also gathering abiotic data, the water temperature and the salinity, and where you can use all that information to understand why these communities are changing when they do. And of course the substrate's changing. Not too long ago we were looking at mud and then coarse sand, and now we have this rock, this hard substrate, this with holes and little caves. Yeah, and you can see it's raining down marine snow.
Feather stars.
Yeah, feather stars taking advantage of the height of the corals to get out off the ground to feed and just stay protected from predators. Lots of sea whips. There's a black coral sea whips on the left. Little cave.
Like this.
Wow! Look at this!
Oh, that's soft coral.
Yeah, on the inside?
Yeah.
Chrysogorgia? No? Oh my god, look at that!
Can we look at?
Can we just slow down and just kind of have a bit of a look at
Yeah, just get my lighting.
huge colonies?
Yeah. So it all looks like one sorry, it looks like one type of coral, what they're probably a soft coral. So we're just going to take a zoom in. Oh yeah, soft coral. Look at the shrimp.
Oh,
Look at
big.
all those
All the
shrimps.
shrimp. That's incredible. Look at that. Wow. It's like they're having a dance. Yeah. Shrimp tea party. And look, there's more of them out on the right.
Yeah. Wow.
Okay, and these
look
are
at all this.
cardinal. These are cardinal fish in here. So forget the family name. Oh my God! Look at that. Now are these banded coral shrimp? Do you know? Does anyone know their crustaceans?
Seamounts.
Shrimp party. Yeah. So
Beautiful.
look, absolutely. And over here on the right hand side, full of cardinal fish. Over here in that crevasse.
And then we should take a look at this sea fan also.
Oh my God.
Yeah.
Give me a quick second, I'll just go around this.
Sure. Amazing. I think they're looking at us. Checking out what we're doing.
Just gonna do a little pan around this thing real quick.
Yeah. Yep, so we have Gorgonians really stacked up at the edge of this cliff. They must be getting nutrients coming around the corner here. And the shrimps are watching us watch them. That's great.
That's right.
Is this
You're
going
in
to be one of
Australia.
your favorite
Let's bring
pits?
put another shrimp
Make it
on the
yours.
barbie.
Make it a great one.
There's plenty here. Oh, it's, they're just sort of darting around, checking us out.
So we're at 137 meters and it's almost 22 degrees Celsius. So it's getting a little warm. As Rob would say, it's swimming temperature, not for me.
What would you like to zoom in on?
Well, let's try to take a nice image of these shrimp. I'll try to zoom in on them quickly. Let's get their faces.
Hold right there. Hold right there. Stop.
Yeah, shrimp tea party.
When I'm free flying, if you start panning like that, I think the ROV's moving. So I'm trying to counteract what you're doing.
So don't pan them.
Yeah, just zoom in
Yeah.
when I'm free flying. I'm just straightening it up right now.
Oh, they're all around. They're fringing all around the right-hand side.
Yeah.
Oh, wow. That's freaking amazing. Yeah. Yeah, you see the size of these gorgonians. The whole face is covered in them. Wow.
And see the fish in the back.
Raining shrimp. And cardinal fish in the background there, hiding as well.
Beautiful. Oh, they're looking away.
That is amazing.
It's National Geographic type stuff.
I don't have contact with Nat Geo. Look at that. Is someone talking?
Nope, someone's trying to get someone's attention.
All right, I'm going to zoom out. And could we just look in to the right? Just take a quick look for potential pygmies.
Which one's that one?
So
Yeah, you start slowly zooming in.
all right, we're just going to take a look at this sea fan. So potentially we're looking at some bamboo corals as well. Let's see. Anyone see anything living within the coral? Of the seahorse variety, maybe?
I'll have a look around.
Yeah, and they will be tiny. It'll be literally only
The
few polyps
Yeah.
long.
So J-Rod's has taken us around this fan.
Nice little squat lobster.
We're letting autofocus kind of pick the location to be in focus.
Can you back the lights off just a touch? Can you close the iris? I'll change the iris. Just one stop. Yeah.
It's a shrimp in the middle there.
Anyone see anything?
I'll come around the other side.
Sure.
Me.
Want me to zoom out?
No, you're fine.
There's some bryozoans there.
Yeah.
Just a touch.
Testing 1, 2, 3. Sitting back in the audio, thank you very much there, Jeremy. We're just having a closer look at this gorgonian because in other parts of the Coral Sea we found pygmy seahorse. Pygmy seahorse attached to some of these really large gorgonian sea fans, and we wrote a paper with Dougal Lindsay, who's our plankton biologist who is collaborating with us from Japan. We took his Picasa ROV out to the Coral Sea, and they found we found this tiny pygmy seahorse, literally only a few centimeters long, attached to, with its tail wrapped around the fronds of one of these big gorgonian fan corals. So it's what you're looking for is any kind of movement. And the colouring will be exactly the same as the gorgonian fan coral. Like they've evolved to colour themselves, camouflage, look exactly like it. So you're looking for anything that might move in amongst it. It'd look like a branch that's suddenly moving. So we're just having a bit of a scan here. Pretty amazing that we're 137 metres. We got this ROV the size of a large car, and we can literally dance, at this really fine scale along the edge of this gorgonian fan coral, which is probably a good metre wide. So just really fantastic. Of course, there is lots of other life here, and if you look closely, I can see feather stars, brittle stars, brittle stars entwined on the right-hand side. And these large gorgonian fan corals are their own little ecosystem in themselves. There's plenty of marine life just using them for food, or even just actually living. But I think these pygmy seahorses are really amazing because they actually have evolved with the fan coral itself, and we have found them at this depth before. Not seeing anything though. It looks like we've got a nudibranch here down on the bottom left. Guys, we've got this nudibranch just here. There we go. Excellent. J-Rod and Ben are driving. They're our ROV types. There we go. There's a nice close-up. So, we could assume this nudibranch is actually probably feeding on the polyps here. And of course, there's just this amazing variety of nudibranchs on the Great Barrier Reef. I've seen thick books written on them, just multiple photos of different nudibranchs. Really quite spectacular. You can make an entire career studying nudibranchs. And in fact, if you even look at the nudibranchs, it's got like little mites or something, little black dots crawling across the body. And, you know, yep, they're definitely like little, I don't know, little isopods perhaps, little crustaceans, who knows. You know, life upon life upon life. This is really quite amazing. We got this is this science camera. You can zoom in so well. Yeah, look, thank you very much. That was just brilliant. But just look at I love those big perspectives, you know, where you just bring in all of that life together, and we've got lots of big fan corals here. You know, and they're trying to capture all of the food that's drifting past. They're stuck on the wall. The food has to come to them. But along the face of Ribbon Reef 5, we can expect to see. What have we got in there? But it's probably geez, this is a close-up view. I'd say it's a brittle star. So these are the spines coming off the arm of a brittle star. Just wrapped its arms around the fronds of this large Gorgonian fan coral. That is just a great view. Yeah, exceptional piloting, because I believe it looks like they've got themselves locked onto one part of the wall. It's an absolutely vertical wall. I said before it was like parking your car on the side of a skyscraper. Like, how does that stay there?
My half camera.
What's that?
My half camera.
Oh, yeah, we're looking at this. Hey, Jody. Just describing this big Gorgonian fan coral that's in front of us here. We've got a big, big zoom in. This is what amazes me about this camera. You got that real big perspective. You can bring in that whole kind of environment, the big habitat story, and then you can zoom right in on the bristles along a brittle star's arm, which is attached to a Gorgonian fan coral, and see all of the individual polyps. It's just pretty crazy. I'm looking at some other camera views. I got the luxury of being in the control room. There we go. We just put that as an inset. This is the view, one of the side cameras. This is the inset. That's what's off on the right-hand side of us, okay? Yeah. That's a very big wide angle. So you can see the blue above, that's the sunlight. Filtering down, and literally you could swim to Ribbon Reef 5. It's just off the back of the ship. It's really, really close, but it's absolutely flat outside. It's a beautiful day, and that is the wall that David Hopley drew in that hand-drawn profile from the mid-80s, and here we are actually seeing what it's made of. So yeah, pretty amazing. But we're at go on.
Do the ROV guys think they can snaffle us a nice chunk?
Yes, we're aiming for something around that 120 metre mark, 120, 130.
If we could get something around here, that would be ideal. Because as I said, this depth, 137 metres around here, is the inception of who we are from the Ribbon Reef 5 core.
Okay. All right, no worries. From Jody Webster, would you be able to take a rock sample from around here? This is a pretty key depth.
So from the one that I just flew over?
So oh my God, this is so steep. Yeah, so we're 130 metres. This is kind of going into that last ice age where the sea level dropped to, and we have these really big overhangs directly in front of us. We just got to find the right place to be able to put the ROV down and see if we can break something off. So this is a big overhang here. Lots of caves, lots of overhangs. Of course, the marine life just make it even more spectacular. Huge Gorgonian fan corals draping down from above like great curtains, great colourful curtains. So Marty's here in the control room. She's working with J-Rod to try and find something ideal, because it isn't easy. It's so steep, and not ideally you got to park the ROV down, but it's literally like trying to parking on the side of a skyscraper. Where can you, what level floor can you kind of get the wheels of your vehicle into?
Wow.
Okay, Jody, we're heading into that really critical last ice age sea level zone. So, anywhere from about 130 through to 110 metres down. This is where global sea cycles have cycled, repeatedly through time. So let's say the last ice age, it's about 20,000 years ago. So sea level was down around about this level. And I guess this, for anyone listening, this is a really critical point that we want to make to people, that that we want to understand the corals that have grown here, because you've made a whole life of career of understanding these fossil reefs and how they've grown since that last ice age.
Yes, indeed. So it's really really important for climate scientists and people that study ice sheets to understand the timing and extent of the maximum extent of ice on the planet, and that correlates with the lowest sea level position. Of course, it's more complicated than that because of movements of the Earth related to the redistribution of the water and the ice. So in any given place, you're only measuring relative sea level, but where the modelers are able to reconstruct the glacial or global sea level by using records like this from all over the planet. And so getting as many records from as many different places is really, really important. And other work that we've done Great Barrier Reef further to the south, we've been able to really capture the exact point of that last glacial maximum period as defined and marked in the cores. But we haven't been able to do it this far north because, as I said, we were only able to drill a few rocks or a few cores. And there's lots of questions about whether there's any significant reef growth during this low sea level position, because we have such an incredibly steep margin that maybe we can find parts or small fragments of that low stand coral growth. If we can't, then we may be able to get deeper into the interior of the older structure of the Great Barrier Reef.
Yeah, just to know how tricky it is completely vertical here, and we're kind of just flying off the edge of this, like a big steep building, and J-Rod and Ben are just trying to find the right position to grab a rock. There we go. Okay, so some of them, of course, crumble. We got a piece, and just having a look. Okay, that looks pretty solid. Marty's having a look. Okay, yeah, we're definitely going to keep that one. She's a keeper. Wow, look at that sort of overhang off to the right there, Jody. So, just you've got to use imagination here, but last ice age, that sea level just dropped down to that 100 odd meters. This is probably, this would have been like the edge of a big cliff, and the waves would have been washing up against the edge of it, and everything above was exposed. High and dry, or it's just been exposed limestone. So if anyone's ever walked across, a cave or karst landscape, they would see this, what I'm talking about here, like limestone that's exposed to the air, becomes all eroded, and get lots of cavities in it. But it's it'd be a karst landscape.
Yeah, great job, guys. Clearly, a nice sample was broken off in situ, which is really what we're after. So, it's really exciting. Hopefully, there's some even a small fragment of the original interior of the fossil coral.
Yeah.
I guess they're going to give it another go.
Yep. So I'm going to try. And of course, there's just so much life in here. You're getting a lot of cover, even though there's still sediment coming down. Oh, we got a fish just hiding in a hole here. Can't quite make out what it is. Very, very pretty. Okay, they're just discussing which piece they might be able to break off. Ideally the bigger the better, but you can't make it too big because it's just too hard. I mean, this limestone is it is cement, and like cracking a piece off is not an easy task.
Oh, that's a good shot.
That face there for a drill.
Oh, yeah. Hey, we're also going to have a go at drilling here, Jody. This is this tool that the guys built over the last few days.
Great.
We used it yesterday to drill the basement rock, that bit of continental crust that's below this fossil reef. Okay, so Great Barrier Reef is growing on old Australian basement rock, and we actually drilled a piece of it yesterday down around about 1,000 metres depth, a kilometre deeper down from where we are now. We're going to try and we're going to pull that out, and we're going to have a bit of a go. And I think that's pretty interesting. It worked yesterday in some pretty hard rock. So we'll see how we go this time.
Yeah, that's a fantastic idea. I think it'll be this material. Should be softer. So it should be a little bit more successful. But,
I hope so. Not always clear. Like remember out in the Coral Sea, they were trying to get a bit out of some of that old limestone rock at the base of those Coral Sea reefs, and it was really hard. Oh, I do like that view.
What's that?
So 130 metres. We're just kind of repositioning ourselves here. Lots of cavities, lots of little caves, little habitats. I see lots of solitary cup corals, probably sponges, brittle stars. Yeah, beautiful life. Okay, we're having a go at grabbing this one. Whoa. This is the wash of the...
Well done.
Yep, well done.
Yeah.
There's a good face.
Something.
Yeah, look at that. You can see a nice fresh face there. So how's that, Jody? What is it? Is that good to you?
Yeah, it looks good. Can we zoom in the camera a little bit? We've clearly broken it off, which is important.
Could you just zoom in a bit, please? Just to have a look at that face there. So thanks, J-Rod. So how does this, how does this sample make it a really great clue for past sea level changes?
Well, we'll only really know when we cut it up and we can look at a cut surface and we actually can determine whether we have sampled corals or algae that is indicative or indicates shallow water environment. And so that's really what we're hoping for here, if we can find some of the original fossil coral or algal material, and then we can date that, and we can argue or prove that it was in situ. By that I mean growing in its position at this depth 20,000 years ago, for example. But it looks promising, and the first step is to identify a good depth, a good location, and then be able to break off a piece from the outcrop, and that's what we've done. So that's fantastic.
Okay, and we're going to give our drill a go shortly.
And what might be ideal with the drill is if they're able to go in close to where they broke the sample off. That they've already broken off the veneer, they may be able to get into that surface and get even further into the reef. So whether they can identify that, that would be great. If not, then we just do the best we can.
Okay. All right, so a different manipulator arm to do this one. That was the piece that we broke off there, Jody.
So I think a strategy here would be to go into that breakage surface with the drill. Because again, our challenge here is to get into the interior. We know from our experience with dredging, for example, and some other work that we've done, is that there's this crust or veneer of bioerosion and boring and infilling of more modern or recent sediments. So the further we can get into that interior, the better chance we have of getting well-preserved fossil reef material.
Oh, you got to love this. This is the just pluck out your tool of choice. So use the right arm to hand over to the left arm, and it's great to watch these guys at work here.
Very skilled.
Yeah, so Ben's got the arms at the moment. They're really unusual how the manipulator arms work. It's not quite what I expected. I was thinking more like a joystick on a PlayStation machine, but they don't look like that. They're almost like a sort of a pistol grip, and there's quite a few buttons on them. But and they're not moving. They're not actually moving their hands around very much at all. It's a very fine movement, here in the control room.
Yeah, very impressive.
Yeah. So they're basically miniature versions of the hands themselves, and because we got lots of cameras here, like 10 cameras all over the ROV, and we can get all kinds of views. Look, downward-looking view, the straight-on one that you've got, views behind, which is important because sharks sneak up on you, and you want to make sure your umbilical cord is clear of any hazards and stuff. So there's cameras for all. Basically, the whole thing is this 360-degree camera.
Yeah, fantastic. I like the shorter barrel this time.
Yeah, it is. Yeah, yesterday's bent. You imagine the torquing going on, like TOQE. There's twisting going on, and and it just like it was hard basement rock. It was like a solid mudstone. So you know, this is the ancient Australian continental rock that's you know, sticking out underneath the Great Barrier Reef is growing, and you know, it was really amazing to see it cut into the rock. We're going to try. So the actual shaft of this has been shortened. Hopefully, it won't bend, and we'll give it a shot. So we might get back to that same rock face that we just broke it from, and see if we can get a piece.
Yeah. Oh, well done, guys.
It's almost like underwater surgery.
I'm amazed at how well they're able to hold station. Are they powering against the outcrop? Is that
Yeah. I think, J-Rod, you're actually leaning against the rock, is that? Yeah. So that gives them a little bit of purchase. You know, there we go.
Really?
So they're stable, but you think this is really vertical, so it's tricky. You know, it's not like yesterday we actually had a nice big flat horizontal seafloor to sit down on. This is
so just
different.
Sorry, Rob. On that image, I can actually see the boring and multiple phases of infill sediments.
Yep.
The camera is so good. You can see the processes that this rock has been subjected to.
Yeah. Now I'm looking at some of the other cameras too. They're pretty spectacular. Looking down, we actually got a downward-looking camera, so we can see the arm now reaching out towards the rock. And there's not much underneath, I've got to say. It's just dark, you know. So it's just its vertical face. There we go. All right. So we're just moving it up a bit. There we go.
Oh, there we go.
Looking at, yeah. There we go. Yeah, we're not talking about going fast here. It just needs to rotate slowly, and it's slipping around a little bit. There we go. Just needs to kind of get a bit of purchase, and then in it goes.
Oh, yeah.
Okay. Pulling back now. Just have another think about this. Yeah, so the yellow cord is just simply a means to grab it, you know. So it's stored over in one of the right-hand boxes. The right-hand manipulator arm can pick it up and hand it to the left arm. So that's all it is. It's just a means of passing it from one arm to the other. Okay, we're going to pull in a little, we're just going to manoeuvre around. Not easy. Parking your car on the side of a skyscraper.
I was jumping around a lot too. I only had one put my feet down.
Yeah, so the problem is, if you can't sit the ROV down and, you know, make it lock solid on the seafloor, it's just that much harder when you're pushing, you're actually pushing, and because the rock, everything's pushing against you, and, you know, eventually the ROV will just push itself out into the out into the water column. So the ROV is, you know, 4 ton, but in the water it's neutrally buoyant, so it's completely float up or down. So very tricky.
Yeah. The challenge on this vertical face, the profiles that Hopley and David Hopley and Peter drew from the previous 80s show sort of a rollover or terrace point at 70 metres.
Yeah, okay.
Brow, which they describe as 70 metre dead reef. And it may be possible if we can't sit the ROV on this form to try further up in the shallower depths.
Yeah, okay. Yeah. Well, they aim
worth another try.
Yeah, they aim to please here, but of course, Joe Rod, if it's just too difficult, we'll may be able to try a little bit shallower up. But he's going to have another go here. But again, it comes down to, you know, getting the ROV in a good position before you can start drilling. So you can see this is the situation cam. This is downward looking, right? There we go. You can see that view. This is what it's like directly under us. It's just a vertical wall. But we've kind of I don't know if you notice at the in the screens there, the sort of legs that have come out, and they kind of just can kind of lock the ROV down on the seafloor. So it's pretty amazing ROV here. Okay, here we go. Let's see what we can do this time. We'll have another shot. There we go. We don't need to rotate fast. We just need to get a good grip. And you can see the teeth at the... there you go. It's starting to chew. It just needs to find its sweet spot, and then in it goes. There you go. That's a great view.
Yeah. Come on, cut.
Because the core you've been playing with in there in your office, it's like 210 meters of this, right? That's quite a job to actually get 210 meter long core out of the middle of a coral reef. It must have been some setup that Peter Davies had, you know, to do this.
Yeah, it was an incredible international exercise. Actually, it was an international project with Europeans and Japanese, and I actually wasn't there. I was in Japan when this was going on, working on some uplifted fossil reefs. But my understanding is it took 6 weeks to actually achieve. It was a pretty mammoth exercise, and to produce such wonderful complete core is a real achievement.
Yeah, I mean, you don't just put like a big long 200 meter long pipe together. You actually do it in sections, don't you? So you can just drill down a little bit, you recover that piece, and then you put another drill bit in, and you push that down a little bit further. And these would be diamond saw, like diamond studded teeth to be able to cut through the rock, right?
Yeah, so diamond rotary drilling bit with a wireline system that sends the drill bit down, takes a piece of core, a meter and a half section, and then another tool would go down to grab that core barrel, the inner core barrel, and pull that out, put on more pipe, put the core barrel back down, and again. It's an incredible feat.
So 6 weeks of just drilling.
Yeah, and similarly with the IODP expedition was even a more difficult technical challenge because with this drilling, Ribbon Reef drilling, it was putting a platform on top of the stable platform of the reef. What we were doing with the ocean drilling program was to drill on the seabed at depth, which was more difficult because you don't have a stable platform.
Yeah, it was a ship, yeah.
So there's all sorts of challenges, yeah. But really with this sort of environment that we have with such steep faces, there's no other way but some technique with an ROV with some drilling setup as we're trying to do here.
Mm-hmm. Boy, this is tough.
Yeah,
this is tough coral. It cut all around it, Jody, but it hasn't broken it off. You can actually see how it's chewed into the coral. This is pretty tough. I mean, look at that.
Can we use the manipulator to break off that piece? Can we use the right manipulator now?
Yeah, I don't know. We're just having a think about it here. Everyone's having a chat. Boy, oh boy. So we're just going to put the little driller Cora back in its box. So our bio boxes come out.
It seems, it's working well. I just wonder, for future refinement, is there a drill bit with a slightly longer barrel? Because the rotating is working well, it's cutting. I'm just wondering whether a slightly longer barrel would cut.
Yeah, but
now you kind of then grab it and cut it off.
Yeah, but you mean like the length of the tube, the main tube? Is that what you're talking about?
Yeah, the actual cutting section of that.
The cutting head of it, yeah. Don't know, we can suggest that. But alright, so I think we're going to bring our claw in and just see if we can snap that off. But man, that was a really solid piece of limestone. So it's deceptive, really. I mean, you look at it and you think it's all just, crumbly limestone. I guess it's got this crust that just kind of can flake off. But the actual inner core of the limestone is just tough as nails. Oh, here we go. We got a piece, maybe a piece. We got a piece, but it's just outside of the clamp and gravity. Oh, yes.
All right, good. See, what I like about this piece, Rob, is how white it is. Right?
Yeah.
The fact that there's a piece there that's white, which is an indicator, hopefully, that is it not going to fall off?
No, we're good.
That's a nice indicator. Yeah, that we've got either potentially a piece of coral, or a thick piece of coral and algae, which are that white colour.
Yeah.
And the darker sediment, the mud, which is quite sometimes liquefied, gets that dark brown colour, which is not what we're after. What we're after is that original coral framework material, the building block of the reef.
Yeah, I'll just pass that to the. Yeah, I'll just pass that to the pilots. Yep. Yeah, we're good here, Jody. Last ice age coral from 130 metres down in front of Ribbon Reef 5. Come on, get that rock. There we go. Oh, a big piece.
There we go. Well done. Oh no, don't you do more.
That might be as much as we can get here, Jody. Oh, it's a bit crumbly.
There we go. Let's grab that.
Hold that and see where that piece went. There's a bigger piece.
Nope, lost it. All just crumbled.
It's a tough business.
Let me see your rock and talk back. Keep your eye on it. Got it. Give me that. Alright, coming in for a park.
The highs and lows of sampling on the reef.
I agree, but I would never, if you told me a year ago that I'd be sitting out here in front of the Ribbon Reef 5 doing this with one of the world's most advanced scientific ROVs, I would never have believed you. Actually, if you told me at the start of the whole COVID drama, when when our big expedition was cancelled, that we would actually be at sea doing this. I wouldn't have believed you, because I was quite depressed by the whole thing. Years and years of planning. I describe it's like, being an Olympic athlete. You would train. Like we had funding lined up, students, equipment. People had been hired. We had the ship time, of course. It was just all that planning effort, and then it was cancelled. And that was. And here we are.
Well, fingers crossed, we'll be back in business. I remember it's the
yes,
long
mate.
game.
It is the long game.
That these guys are going to grab. Well done. I can see it.
Yeah. Good that we had a little ledge there for it just to fall on. You remember yesterday, I don't know if you were watching yesterday, we drilled that piece out of the basement rock, and it just tumbled out, and it was pretty steep, and just rolled down in the mud, and we had to pull off and wait for the sort of cloud of sediment to settle, and then we found that rock. Oh God, I don't know, about 10, 20 meters further down the slope.
Yeah.
So you can easily go to all that trouble, and the whole thing just tumbles off the edge. But it is a long game. I mean, we talk about all that.
Well done.
Yep. Thank you very much from Jody Webster. That is perfect.
Sure.
Well done.
All right. So we're gonna continue on and upwards. We're at last ice age, low stand sea level depth. That's about 130 meters. So everything above us, Jody, would have been exposed to air at the last ice age. Do you want to just talk about that a little bit?
Yeah, so what we know, globally, sea levels are at least 120 metres lower, and on the Great Barrier Reef margin, about 118 metres, based on the work we've done off Cairns and off Mackay. And of course, we haven't confirmed that precise level at this site. Perhaps some of these samples may go some way to helping that. But yes, during that last ice age, that 20,000 years ago, 20,800 years ago, at least based on the lowest the best dates that we have in this part of the world, sea level was lower, and so the entire cliff above us and the reef was completely and utterly exposed high and dry. And so you might have, for those folks that have been to Vietnam, Hulong Bay, with this huge tower cast, that's a larger example of what you would be seeing. But I sort of have that in my mind when I try and visualise what the environment is like during that last ice age.
Yeah, so if you were standing here 20,000 years ago, you'd be looking up at a big exposed limestone cliff. Lots of caves, nooks and crannies, and of course vegetation growing on it. Like, we don't think about that much, but if this was high and dry, there might have been, eucalypts and those kind of plants growing on wherever water could accumulate.
Yeah, we have direct evidence in the cores. In the reef cores themselves, there's evidence of soil horizons that we can see in the Ribbon Reef 5 core. Also, a colleague from University of Queensland, Patrick Moss, who's a world expert in pollen, has been for years reconstructing the record of pollen changes in the deep sea sediment cores in the basin. And you have this incredible record of the vegetation that was growing on the shelf during these different low sea level periods, and those signatures are trapped in the deep sea sediment cores, more deeper and more distal, further away from the tenements. So there's lots of evidence that we can see in the record.
Yeah, still seeing a lot of caves here, Jody. Lots of ledges, overhangs. Really, and I'm looking at the wider camera views. Really big caves Oh, this is lovely. It's almost like this one in front of us. It's collapsed. It's almost like the layer has just fallen down, because we know reefs grow in layers, and there's weakened sections. It just looks the whole thing's just dropped down, and boy, it's just covered.
Very strange.
Yeah.
Strange shapes.
But actually, look at the whole face. It's kind of there's these big buttresses, and then there are these quite sort of cavities, or not cavities, like drainage areas that push into the rock. Presumably where there's been erosion in the past, if this was exposed during the last ice age, this would have been high and dry, and creeks, streams running down them, because they limestone erodes quite well when exposed to rainfall. Boy, isn't this a beauty? This is just beautiful.
Gorgonian?
Yeah, Gorgonian fan coral. But look at the feather stars sort of rimming the edge here. Wow, beautiful. Oh, a big sponge in the background. You can see one of the, like a big vase sponge. But first one I think we've seen. Yeah, and there's a sponge in the centre foreground here. This is this sort of red lobate colony. But yeah, look at this big vase sponge coming up.
Yeah, beautiful.
So Jody, I mean, we talk about lower sea levels, but this is done. We know this because it's happened all over the world. When ice sheets completely covered the North American continent, and a lot of that sea level was locked up in ice, and this was felt right across the globe, here in Australia, Tahiti, North America. I mean, it's a global event. That was the last ice age, and sea levels just were cooled. Thermal, do you want to talk about that? How we, you mentioned soil horizons.
Yeah,
so that's pretty amazing that you can find soil in the coral cores. It can only happen when ground is exposed to air.
Yeah, so the entire configuration of the Great Barrier Reef margin was different. The coast was right here, attached, to this. This is where the water level, sea level was, and 100 metres above us was the gentle coastal plain with vegetation, soil, and trees, and river systems, and savannas, and then of course global climates changed. And sea level began to rise, temperatures were warming, and the sea position then flooded back over the top of the shelf, and then about 8 or 9,000 years ago, and it would have been incredibly disruptive sort of environmental change as sea level flooded very, very quickly, depending on the steep and gentle nature of the slope. And then eventually the Great Barrier Reef turned back on again at its current position. So, incredible dynamic environmental change.
This is really important here, what we've got in front of us. This is Leptocerus. This is a zooxanthellate hard coral. Zooxanthellate because it has zooxanthellae. These are photosynthetic algae living within the flesh. You can actually see the corallites and this ridging effect. This is exactly what Tom Bridge, our colleague, is looking for. And here we are at 110 metres, and we're finding the first of our Leptocerus. So that's this quite large brown one in front of us, and also the little almost mushroom like ones just to the left and at the bottom. So this is Leptocerus, and our first evidence, the lower depth limit of zooxanthellate coral. So this is really significant because now we are in the photic zone. This is where hard corals, shallow water hard corals, are going to grow. And so we're just trying to get a sample of this one. It's quite a big colony. But it's interesting, like it's big plate-like, it's sort of spread out because it's trying to capture as much light as possible. Now, it is the mesophotic zone, so the twilight zone, so, you know, if we turn the camera lights off, it'd actually be, it's twilight, it'd be this sort of dim blue light. And so these depth specialist corals, as I say, specialised in harvesting all available sunlight in order to photosynthesize. You can see a few of them there, right? So this is the first zone that we found them in, and this is pretty important for Tom Bridge, our other colleague. Yeah, so as we climb up, we get a bit higher, we are travelling through the twilight zone and into the photic zone, right, where it's fully sunlit. Typically that sort of 20 to 30 metres depth zone. Now we probably won't do that. We won't get to those depths with this on this particular ROV dive, which would be just way too close to the reef. But we're just slowly moving up out of the twilight zone here. So it's quite significant. We've actually found our first zooxanthellate hard coral, and the genus name is Leptocerus. Fairly delicate though, Jody, isn't it? It's just like a big thin dinner plate.
Yes, yeah, they're beautiful corals, and they're also very important part of. We found them in the Ribbon Reef Falkor, and so we're able to use the currents of, like, for example, we're seeing them in these deep settings to help us reconstruct the environment that the corals were growing in. So, yeah, Leptocerus is a key indicator of deep mesophotic environments, and in the fossil world, we can use that to tell us what sort of setting we're actually in.
You don't know where the pointer is?
Okay, I think we're just going to get...
The point being is when we find this type of coral with this sort of shape, with other sorts of indicators, we can use that to tell us and reconstruct that we were dealing with a deep, low energy sort of setting and environment.
Yep, yeah, that makes perfect sense. Yeah. And, we've done a few of these ROV dives into this twilight zone now, not here on the Great Barrier Reef margin, but further out into the Coral Sea. And we've been really excited to see great fields of these Leptocerus, not just one or two, but literally it's like someone's just cast 1,000 dinner plates all over the seafloor, and we're calling it a Leptocerus zone just because there's so many of them. It's its own little habitat, and as they die, they form another dinner plate, and all the living ones are growing on top of it. So I remember when we first saw this, it was just, we couldn't believe our eyes how dense they were. So pretty amazing animal, really.
Yeah, I've done some work with colleagues in the Big Island of Hawaii, and again, I've seen similar sorts of fields of these things in these deep environments.
The one that's still attached to the best part of that centerpiece.
Yeah, I know Tom Bridge is always pretty... Sorry, go ahead.
No, I was just going to say, whether we see those same fields in the way that we saw out of the Coral Sea is another question, because it may be that the substrate is so steep here that it's not an ideal environment to get that dominance.
Yeah, that's right. I mean, that was a big difference between here and over in the Coral Sea reefs. There was a more gentle gradient as we got closer to the shallow reefs, and so you've got lots of surface area for the sun to sort of to come down on, like a much wider area for the corals to grow. But here it's just so vertically steep. It might be there's just one or just a few coral colonies, just scattered here and there. From where we are now, it doesn't look like there's great dense fields of them, but just occasional Leptocerus colonies just popping out on the seafloor. There we go. Now this is a living one. The actual flesh is just this brown colouring on top. When it dies, of course, the we're going to preserve it. It'll be just this pure white skeleton. But the skeleton itself can tell us everything we want about this animal, and but we'll preserve it, and we will be able to do DNA analysis with the living flesh. We'll be able to take this skeleton, look at it under microscope, and certainly our colleagues like Tom Bridge from the Museum of Tropical Queensland will be able to do a lot with this sample.
It's quite different species.
These quills are so hard to get with this big robotic claw. They're so fragile.
Yeah, Jeremy was just saying, how these are very fragile. You can see, they're like thin dinner plates. And we've got this great big, metal, big metal claw.
Versatile is the word.
Yeah, Jeremy was just saying on his big expedition that he did, when was it? About a year ago? Yeah, about a year ago, where he dived the full length of the Great Barrier Reef with Tom Bridge and Andrew Baird, I think. Yeah, they were looking for black corals and these deep water specialist corals along the whole length of the Great Barrier Reef. They're not really solidly anchored to the rock substrate. They're able to just put a finger underneath them and just kind of flip them up and pull them off the seafloor. Oh, look how soft it is. It's tricky. Yeah, this is at the really lower limit of their ability to grow, and they're making calcium carbonate as skeletons, but we're not talking great big thick robust pieces of limestone here. It's just like thin little dinner plates.
There'll be a lot now. I mean, they're probably going to start dominating soon.
Yeah. I think quite a lot of some of the platey things we're seeing at depth on the slopes are some of the modern and near-modern platey foliaceous things falling off and then being deposited further down through.
Yeah, that's right. I mean, we've seen lots of evidence, both in the 3D mapping that we've done with the Falkor and also just in the vision, the evidence for landslides. If you have a look off to the left, you can see these chutes. This is where the sediment is actually concentrated. So we have these lines of white sediment, and you can actually contrast it against this big, oh my God, that's really spectacular, isn't it? The chutes themselves, the sediment also erodes through time. So if this was already, exposed to air, then, that karst environment you described earlier, the creeks and, streams can actually erode and wear away the rock. And then as it gets flooded by the sea, sand continues to just fall down, and continue to erode in these chutes. You see these brown discs on the seafloor. This is Leptocera. So we're not seeing big patches here, Jody, not like out in the Coral Sea, but they're definitely here, and they're kind of dotted about.
Yeah, quite a bit smaller in size.
Yeah, still plenty of these great big Gorgonian fan corals. I'm looking at some of these other, wow, these other cameras that we've got here. Just everything's so steep. And I think we're just going to fly into one of these ravines. It's amazing. The mapping that we've done kind of shows, it's really deceptive. The mapping sort of shows a steep wall, and it seems to be like a flat, steep wall. But the reality is, it's highly indented. So you've got these big, like big bluffs that push out, and then either side of these ravines, where sand and things are draining down. So it's the 3D mapping really doesn't do it justice how rugged it is down here.
Yeah, you need some sort of sideward looking sonar, high resolution to really map at this sort of level. But maybe the photomosaic may be able to help us understand that in some of this imagery.
Yeah, it's quite possible. Like we can take the still images from the 4K vision and create a photo mosaic. Yep.
Sorry, it looks like there's a clear ledge there.
Yeah. Yep, definitely. But then if you look even further below it, there's another ledge. It's sort of like a whole overhang. This is one of these big bluffs that are just pushing out, and then you've got a ravine on either side of it. So maybe this whole face is just a whole series of bluffs and ravines just moving, in and out. So it's certainly not this big flat steep wall that we thought it was. It's quite rugged. Beautiful soft corals. Look at this. It's just a forest of Gorgonian fan corals.
And it's possible that we at different times, at different generations of reef development, there may actually have been parts of the reef front that have actually sloughed off and fallen down into the slope as large blocks. Because we do see blocks at depth. Whether that's happening here, but certainly we saw that out on the Coral Sea. Clearly, great fronts of the reef.
Yeah, but Jody, if you remember earlier on in the dive, it was around about 400 metres depth. It looked like a scree slope. It was all cemented together, and it just popped out of the sediment a little bit. But that looked like limestone coral, but it did look a bit like a scree slope. But so what we're talking about is how active this all is. Oh, a lovely there's a hermit crab just sitting there. So this is not stable by any means. It might be vertical face, but there's quite a lot of material that's just raining down from above, both the sediment, the finer stuff, and also just big blocks. And you and I, we actually have mapped some landslides. A bit further down from here, a bit further south. But think about that Viper slide. That was right in this kind of depth zone, and it was a decent landslide.
Yeah. So there, the margin's a little bit different, so it's a little bit more gently sloping. But there at the shelf edge, a 5-6 kilometre section of the shelf edge reef front had failed and then travelled, kilometres down south.
That's right. Yeah, it was about 3 kilometres, I think. Yeah. So a decent distance.
And probably, and a really interesting record of how active the canyons and not only the blocked or shelf blocked canyons, but also the shelf connected canyons, the sediment cores that we've taken a little bit further to the north, which have that really interesting turbidite record. So, turbidites, they are essentially coarse-grained deposits that originate from further up the slope and then fall down in this energetic kind of fashion down the canyon system, carrying with it all coarse and fine and deep and shallow material, and then depositing that kilometres down the canyon axis. And so it's a really very active system. So, and particularly during the last 10,000 years, there's lots of turbidite events, little individual layers which represent one of these events. So we have an interesting record of how that relates to sea level change and the shape of the margin.
Yeah, and we've actually, with our colleague Angel over in University of Granada in Spain, we've done some work on, I guess, what makes these really big chunks of the edge of the Great Barrier Reef collapse. And there's lots of different ways you can do this. It can be like dewatering inside the sediments, but despite all that, I think we kind of decided it was more likely earthquakes that might have shaken loose some of these really big pieces of the edge of the Great Barrier Reef that, as they to become underwater landslides.
Yeah, it's a tricky business, trying to figure out exactly the triggering mechanism, and it could be related to earthquakes, but of course these happen in the geologic past. There's some suggestion from other places in the world that during periods of rapid sea level rise, you might actually get increased water loading and so on, and that causes more stress, if you will, to the margin, and could lead to failure. But it's very, very difficult. We need to know more about the composition of the slopes, I think, to really get at this and have a much, much better idea of the timing of more of them. So we need to do more work, I think.
Yeah, that's right. I think of the Gloria Knolls slide. Now that's just a bit south of Cairns, which is 170 kilometres south of where we are now. And we discovered that back in 2007, and it turns out to be globally quite a large landslide. Literally the whole edge of the Great Barrier Reef margin has collapsed, and it's about, we did all that measurement and mapping work. It was about 32 cubic kilometres of sediment has broken off the edge of the Great Barrier Reef and ended up in about 1,000 metres of water, where it's actually what we first discovered were the debris blocks, the remains of it. And over subsequent expeditions, we mapped further and further along the edge of the Great Barrier Reef, and of course we found that big chunk. It was like someone taking a huge bite out of the edge of the Great Barrier Reef, and that one in fact was that you line the two up, it's quite obvious what it was. It was a great underwater landslide. But we remember the age dating we did. So there were some deep water corals growing on tops of the blocks, and those debris blocks, and when we age dated them, we got 300,000 years. So we are talking a pretty old event, but it just goes to show how active this margin is. It's eroding. So we got, it's this crazy contrast. We've got coral reefs which are accreting and growing in the shallows, and then we've got the edge, the deeper edge, which is constantly eroding.
Yeah, very, very dynamic environment with a whole range of different processes, delivering sediment from the shelf to the deep sea, the actual margin or slopes failing and the shelf edge failing, again delivering more material to the upper slope and the basins. And so there's a range of different ways that sediment can be moved from the shallows into the deep sea.
Yeah, I guess you and I, because we look through that deep time history, and you have your understanding of the multiple cycles of birth and death of the Great Barrier Reef, and you can measure this, and we know at least 6 to 8 times the Great Barrier Reef has completely died and then recovered, and died and then recovered, and it's done this about 6 to 8 times, and then that other story about how active the erosion is on the edge, like. I'm always amazed to think that this is really active, like huge undersea landslides that have happened in the geological past. People look at the Great Barrier Reef as sort of serene tropical paradise, and it is, of course, it's beautiful with all of the modern shallow reefs, but when you look at that deep geological time story, not even deep time, I was talking back to the last ice age, just how dramatic changes, sea level completely dropped, 100 metres down. People would have been walking across the landscape. Kangaroos would have been hopping around these limestone hills. Going back further, the whole big chunks of the margin collapsed and have left the remains out in the deep waters. Would that have created tsunamis? Yes, it's quite possible. Certainly when you talk about that much large chunks of the Great Barrier Reef collapsing, then you could create tsunamis. It's just fascinating, this highly dynamic edge.
On surface, how close? Probably about 50 meters.
Yeah, and some of the work that we've done, working with colleagues, to actually try and model how large those tsunamis may have been, and what impact they may have had. Yeah, sorry, I'm getting distracted by the biota here.
Oh, me too, mate. Yeah. Unbelievable.
Yeah.
Like vertical forests.
We should be coming up.
We're about 90 metres now.
Over this. Yeah, from about 80 to 70. It sort of starts to roll over the edge.
Yeah, the shelf break itself. There it is there, Jody. Look, it's just above us. That is the shelf break.
Yeah.
Yeah, this is the brow, right? In David Hopley's profile, drawn looking through the porthole of a garage-built submarine. They would have seen this view. This is the brow is what he called it. But okay, now we've just turned the lights off, right? This is the ambient light. Okay, so we are in the mesophotic zone, the twilight zone. This is the natural light that we see here, and of course, there's lots of we know that there's these hard corals, that are Leptoceras, that we've been sampling. Of course, lots of these beautiful Gorgonian fans. And but this is the natural light that you find here. So it's kind of getting into sort of late afternoon. The sun is setting to the west of us, so we're actually facing the setting sun now. So that brightness that you can see at the top of the screen, that's the sun, setting behind Australia. And of course, we can actually see the mainland from the back of the ship here. But this really, really amazing.
Yeah, and significantly, this brow, this 70-meter feature or terrace, has been interpreted as a drowned fossil reef, the top of it. And so right now, it's a habitat for these deep mesophotic environments, and with all this beautiful biota. But the structure that they're growing on now was probably grown and then drowned between 15 and 13,000 years ago. During one of these rapid rise events.
Yeah, I mean, people were walking across the landscape, and we have lots of evidence of Aboriginals in Australia around that time. They were in Australia, as far back as, say, around 60,000 years ago. So, we've now reached the top of that cliff that they could have stood on, after that, around that last ice age period. And they could have stood here and spearfished, or maybe even, used canoes to, paddle across the shallow water, the coastline as it was back then. But this is really quite amazing. This connection with the Indigenous Australians who, have this. They have their own dreaming stories about talking about the standing on the edge of a steep cliff, and here we are. This is that point. This is the shelf edge of the Great Barrier Reef, at which this cliff that they describe, here we are just coming over the top of it now. Lots more hard.
Amount of life.
Yeah, and lots of hard corals. You see these big plates, really getting more dense here. Big anemone, we're just passing through there. Just need a clownfish to jump into view now. And if you have a look across just above us, you see this halo of fish. Probably antheas, I think, is my, just looking at their profile. Really beautiful.
Just let us give us a 10-minute warning before you leave bottom so I can get it out.
Yeah, and temperatures are getting up there.
Yeah.
24.5 degrees C, so well and truly swimming conditions. And those fan corals keep on going. Sponges, a lot of fish here, really big cluster. I think these are antheas. Big sponges, kind of vase sponges. But yeah, you can see the Leptoceros. They're becoming quite broad now. You know, like a metre across.
As the slope is sort of flattened out.
Yeah, definitely flattening out. We're really at the top of the cliff now. So what are we, 80 odd metres? So I have to remind you of something, Jody. Do you remember coming out on one of those small boats with Richard Fitzpatrick, and he swam down to around 40-50 metres depth and got you a fossil rock from 50 metres? Do you remember that?
Yeah, we were discussing this actually on the coral sea dives that we were narrating together. Yeah, they dove to 58 metres. As we're looking up slope, we're going to come up again to another feature in the seabed, which is this drowned reef at 50 metres. And they dove to the top of that and then went to the base. And so we're kind of coming up from the slope, so going in, past where they dove to, which is quite something.
Yeah, it is. And, we actually won't linger here too long because, in fact, it affects the ROV because it's such a big system. When you're in these very warm waters,
It's almost perpendicular to the vessel.
25 degrees C, internally the ROV starts heating up. So, unfortunately, we can't kind of hang around too long. But, this is really amazing. So we've come up that vertical wall, we're over the brow, and we really are now on the sort of shelf proper. And the shallow Ribbon Reef 5 is literally just like 200 metres away from where we are at the moment. So we're just doing a kind of a slow. Yeah, they're like great ancient rainforest trees, aren't they? Just so big. They're metres across. Oh, look at these fish here. Beautiful fish. I like these little white spots on the sides. I wonder if we got Kai watching here. Kai's one of our fish biologists from Sydney University. It's an antheas, and I'd love to know what that is. Little white tips on the ends of their caudal tail. Beautiful. So Jody, we might wrap up you and I chatting here. I think we've covered the geology as much as we can, and I'd really like to hand off to the biologists. Maybe we've got some coral reef ecologists on board, and I'd really like to give them a chance to talk about what they see, because they've been studying this kind of environment. So, mate, I'll say goodbye, and look, it's been a really wonderful journey heading up this fossil reef zone, and we'll talk again in the future, because you're with us for the remainder of the leg, but from the comfort of your home in Sydney. And we'll keep in touch. Okay, I'll say goodbye, mate.
All right, mate. It's been wonderful. It's been wonderful to share with you. And I will be in touch and enjoy the rest of the dive. And I'll touch base tomorrow with you guys.
Okay. See you, mate. All right, bye.
So introduce yourself.
All right then. Hello, this is Gemma Galbraith. I'm essentially a fish biologist on board this cruise. So this is getting to the exciting bit for me. My PhD is actually looking at a lot of the fish and benthic communities we find on these sorts of slopes on shallow seamounts, pinnacles, and so this is starting to look nice and familiar. So I think the antheas that Robin was so taken by just now, I think was Pseudantheas pleurotinea. But if someone's online that wants to jump in on that, that would be cool. And this is just awesome, you know, how we started this dive at 1,500 meters or so, or some of these ridges go down to 1,500 meters, and just come up this continuous habitat spanning that entire depth range. And you know, now we're coming up to this sort of flatter aspect. You can really see how we've got a lot more fish, corals, the light, all of those abiotic factors as we've come up this slope have changed, and of course changed the communities that we get here. Yeah, some Pictochromis there, those really nice little half and half dotty backs, half purple, half yellow. Bunch of Chromis, maybe Chromis alpha, ambonensis.
Well, that could be off in the distance. This could be 50 meters.
So yeah, this patch is interesting. We've just kind of come off that ridge as we were coming up, and you know, this looks a little more rubbly. So potentially some kind of gully where there may have been some kind of slide. You can see those big plate corals we were looking at are absent. So this is a really interesting change. You know, we're still relatively at a similar depth.
Perhaps it was a really distinct change.
Yeah, I mean, if it were bleaching, I would expect to still see some large plate corals intact, but white. So this may be something to do with different growth conditions going on here. Yeah, there's some Halimeda, lots of Halimeda. Yeah, really interesting.
Right? Isn't this algae on top of everything?
This is
It's Halimeda, yeah.
This is living Halimeda.
So all that, the Halimeda rubble that we were seeing way down the ridge, this is essentially where it comes from, and when it breaks up and dies, it'll tumble down the slopes. Some other calcareous looking algae. Okay, we seem to be transitioning back to a little more growth. Some red algae, some mats, and there we go, some bigger plate corals. Huh.
Oh, we're looking for Acropora branching. Not sure if we'll find it here though.
What do we got here?
That's a mushroom coral.
Got a little fungid down in the corner there.
Oh yeah. So you know Acropora branching hard coral?
No.
Misting up slightly.
Branching hard coral, that'll pop up.
like a
So
plate.
yeah, interestingly, these sort of bigger plate corals are back, but the abundance of fishes that we ran into earlier are
Pachyceres.
not really there. We've got a few
These are all Pachyceres, right?
chromis around and wrasse, but yeah, very flat in terms of complexity.
Yeah, it's about
You launch just outside the dock and head here.
Beautiful.
It is beautiful, yeah. And this is kind of what those photosynthetic corals do down at depth. Their morphologies have adapted to make the most of, the little light that they do get.
coming up here in the next 5 minutes.
So unlike, the branching acroporid stands you get right at the surface, these guys are trying to make the most out of what does reach them down at. We're at 65 meters at the minute.
How the floats like?
Yeah, just fields of them.
Hanging?
This is almost the depth at which we dive.
This is pretty much our sort of survey realms, Jeremy.
Almost all squirrels.
Got lots of nice, squirrelfish and soldier fish. Holocentridae, they like the dark. More lovely antheas. Oh, some gorgeous wrasse down in the corner there. More Pseudanthea. I think that's Pleurotinia, the one with the patch on the side. Rob. It's
Very
one of
pretty.
the bigger Pseudoanthids. Yes.
With the pink
with
finger mark.
the white blotch anthea. But this is a real typical depth for them now, so it's quite cool to see them a little deeper earlier. Yeah, just amazing seeing them all swim through these forests of yeah.
Those are the
Those are angelfish, yeah, just down in the corner, those bigger yellow guys. Dark blotch on the head and the sort of black anal fin. Okay, so we've sort of actually reached scuba diveable depths now, so it's time for SuBastian to make his way up to the surface, and we'll be getting her back on board.
SuBastian can do scuba diving depths. We're out of a job.
There we go. All right. Well, this has been an excellent dive, and we'll see everyone for the next one tomorrow.
Bridge ROV. That's ROV leaving the bottom.
I was going to say, just put one insert.
You can go back to that control.
The 3D.
Yeah, I got ya.
Yeah, you can go ahead and take control and start paying in.
Got that, Ben? Yeah.